two sessions will be held in E-Studio, which is located in the main building, second floor. So here, the first technical session will be on smart breeding in horticulture crops. So in every technical sessions, we have keynote address, one keynote lecture, and two or more than two lead speakers lectures. And after the completion of lead speaker lectures, we will continue with the oral presentations. Initially, oral presentations will be held for offline participants. After the completion of offline participants, the online oral presentations will be continued.
Check this.
चलो that button है sorry
यार
हाँ ಸ್ವಲ್ಪ सर
participants के 5 minutes.
PPT ಇದಕ್ಕೆ ಬರೋಣ level start ಪರವಾಗಿಲ್ಲ ಕ್ಲಿಯರ್
Present.
ಹಾ ಸತೀಶ್ ಅಲ್ಲಿ ಇಲ್ಲಿಂದ ಟ್ರೈ ಮಾಡ್ತೀನಿ ಒಂದ್ಸರಿ ಆಗಿಲ್ಲ ಅಂದ್ರೆ ಇಲ್ಲಿ ಮಾಡ್ಲಾ ಶೇರ್ ಮಾಡಕ್ ಶುರು ಮಾಡ್ಲಾ ಓಕೆ ಆಮೇಲೆ ಶೇರ್ ಮಾಡೋದಲ್ವಾ ಆಗಿಲ್ಲ ಅಂದ್ರೆ ನೀವು ಅಲ್ಲಿ ಹೆಲ್ಪ್ ಓಕೆ ಥ್ಯಾಂಕ್ ಯು
PPT ಗಿರೀಶ್ ಸರ್ PPT introducement
15 minutes ಇರುತ್ತೆ ಸರ್ 15 minutes lead speakers ಇದೆ 5 minutes plus or minus oral presentations as a brief
sir
Welcome to the technical session on smart breeding in horticulture crops. So we have a keynote address speaker today with us, Dr. Sharan P. Angdi. So former head of research Asia Pacific, Nunheim Seeds, Netherlands. And he'll be talking on smart breeding in horticultural crops, innovations for a sustainable future. So before I introduce about Dr. Sharan P. Angdi sir, I would like to request Dr. K.V. Ravishankar sir to take his seat as a lead speaker. And I also request Dr. Lohit Ashwa sir, he'll be the second lead speaker. And Dr. B. Fakrudin sir will be the chair of the sessions. And Dr. Shantappa sir will be the co-chairman. And the rapporteurs for this session will be Dr. Rekha Chittapur and Dr. S. Sir Mulla. I request Satish sir to welcome Dr. K.V. Ravishankar sir and also Dr. Lohit Ashwa sir. I also request Satish sir to welcome the chairman of the session, Dr. B. Fakrudin sir, Director of Research, University of Horticultural Sciences, Bagalkot. And also I also request him to welcome Dr. Shantappa T, Special Officer, Seeds, University of Horticulture Sciences, Bagalkot, as a co-chairman. I also welcome session rapporteurs, Dr. Rekha Chittapur and Dr. S.R. Mulla. So now we will begin the session with the keynote address by Dr. Sharan P. Angadi. So sir is speaking on smart breeding in horticultural crops, innovations for a sustainable future. So I would like to briefly introduce about. So now we will begin the session with the keynote address by Dr. Sharan P. Smart breeding in horticultural crops, innovations for a sustainable future. So I would like to briefly introduce about. So now we will begin the. I would like to briefly introduce about Dr. Sharan P. Angadi, Sharan Angadi sir. So Dr. Sharan Angadi, he is a formerly head of research for the Asia Pacific region with NUNHEMS. It was a part of Bayer Crop Science. And he completed his graduation from US Harvard and later MSc from TNAU Coimbatore. And he served as a research scientist in US Bangalore, and later he moved to the agriculture research scientist and served as a scientist at Director of Oilseed Research, Hyderabad. And he contributed to genetic improvement in sesame, castor, peanut till 1984. For his doctoral research program with ICR Senior Research Fellowship at the renowned Indian Agriculture Research Institute, New Delhi. And he was elected Fellow of Indian Society of Genetics and Plant Breeding, New Delhi in 1994. He served in various positions and different private companies, and he has number of publications to his credit. And currently he is the director of ATP BR Foundation for Advanced Training in Plant Breeding, and is actively involved in designing and conducting training program on general and crop specific commercial breeding programs. He is also the panel expert for global research for development support ventures, GRSV consulting services, and R&D advisor to a few Indian seed companies and KWS, a German MNC. Popular science writing, humor, and karaoke singing are his pet hobbies. He has published several popular science articles in leading dailies and weeklies in Kannada and English. He has spoken on humor in science at the national and international events. All India Radio, national and private TV channels in India have aired his science talks and panel debates. Various scientific and professional organizations have honored him for his contributions to crop improvement and popularization of science. So he'll be delivering the lecture on smart breeding in horticultural crops, innovations for sustainable future. Sir.
Okay.
Sir, over to you, sir. Thank you.
Okay, na?
Good
Good afternoon.
afternoon. The chairman, Dr. Hello? Shall I start? Can you hear
Sir,
me?
Yes, sir. Your voice is audible, sir.
Okay. Would chairman like to make any comment before I start? Or
Sir?
would the chairman of the session like to make any introductory comments?
Chairman of the session, Dr. Fakhruddin, sir.
Dr. Fok, I know him very
Good
well.
afternoon, sir.
Director of Research,
Good
University
afternoon,
of
Good
Dr.
afternoon,
Fakhruddin.
sir.
How are you?
Good afternoon. I welcome all the delegates to this session. I welcome my co-chairman and rapporteurs. And we have the pleasure of listening to Sharan Angadi, sir, and he's a legendary plant breeder and a great speaker. And we also have two more learned speakers, Dr. Ravi Shankar and Lahita Ashwa's lead speakers in this session. Let's start it off. Sir, over to you, sir.
Yeah, thank you. Thank you, Dr. Fakhruddin. Fakhruddin and I, we are old friends. I know him very well. And another person I know in this is Dr. Mulla. So I'm very happy to be here, and thanks for inviting me for this keynote talk. After me, there are two good friends, Dr. Ravishankar and Dr. Lohitashva. They will be speaking on strategies, genomic strategies and pre-breeding strategies, I think. So what I will do is, I'll just give you a brief on, I will make it very general. I will not go into a lot of details. Already that paper which I have sent, it has all the details. I'll try to keep this very light and try to only highlight. How much time do I get, Dr. Fakhruddin?
Sir, about 15 minutes, sir. 15 to
minutes,
20
okay.
minutes. Yes.
In the next 15 or 20 minutes, I'll just try to, I want to keep it like a kind of a dialogue if some of you can participate in what I'm doing. Otherwise, it will be just a presentation. Whichever way suits is okay with me. I'm happy that someone is assisting me with this. I was just thinking of trying to share myself, but then I expected some hitch there or glitch. Thank you very much. I think we can go to the next slide. Hello? Can we go to the next slide, please? Yeah. So when I thought of smart, because since morning I have been listening to all the talks by Dr. Dandin, the chief person from Finland, and Dr. Janaki Ram and others. It was very interesting. It has been a very interesting and elaborate session. So there has been a lot of talk on smart. So when I think of smart, some random thoughts came to my mind. The first thing is smart has mart in it. These days you see a lot of marts, India mart, this mart, that mart. So there is a linguistic connection between smart and mart. Does that mean intelligence is always on sale? Because some people definitely got a discount. Mart offers choices for shopping, and well, being smart means making wise choices. That's what we do in breeding. Smart saves you money, and being smart saves you time, effort, and mistakes. That's very, very important. Because I am in the field of plant breeding since 1974, when I chose plant breeding as my major field of specialization in the final year of my graduation. That was in 74. Those days, breeding was much different than what it is today. We were told its breeding is an art. People who didn't have any basic knowledge of science or whatever, they were able to develop some varieties which are ruling even today. We were told it is an art, then it became a science. The definition of breeding has been changing, and in the past 50 years, things have changed. A huge, enormous change has been there. So then, as I said, if being smart means having a mart in your mind or a selling place in your mind, I hope it is always fully stocked with good ideas and great deals of wisdom. That's a wish, I would say. Yeah, next slide, please.
Next slide.
Hello?
Next slide. Next slide.
Or shall I do it myself? If it is difficult for you. I think let me try. You think you can stop this? I'll try sharing my thing. I think that will be easier for me. We'll go with this, or should I share? Yeah. So as I said, breeding was kind of as an art, and then this I found very interesting. This is from the book which we used to refer as students, Briggs and Knowles, and this is attributed to one Frederick D. Ritchie, and Shattered Dream of a Corn Breeder. I won't read the whole thing. I just put it just to make you aware that this is an old concept that used to, a corn breeder sells 100 corn plants, put each in a cross. Selfing without testing means a heavy loss. So it starts with that. I think I will not go through all of that. One sturdy inbred remains. When it starts with 100 and ends up with one, all alone, it has no sex appeal, can't find a home. That's the effect of the breeding. So if we call today's breeding a smart breeding with so many adjuvants and adjunctant things are available, maybe we can call what we were doing a stupid breeding. So we had no clue of what it was, and then we were going by putting the plants in a field, trying to assess the phenotype, then make crosses, then assess them, think of combining ability, heritability, so many things we did. Next slide. Yeah. So what I will do is I'll just briefly mention what I am going to talk about. I thought I will just list 5 key points. Enhancing crop resilience. That means focus on developing crops resistant to pest diseases and climate stress. Improving yield and quality by integrating modern breeding techniques of genomics, marker-assisted selection, and gene editing. Accelerating breeding cycles, advanced breeding technologies to shorten the breeding time. That's what it is important, because you can't do what you were doing earlier. That used to take a lot of time, 10 to 12 years, but then things won't wait that long now. Sustainable and resource efficient farming, smart breeding contributes to efficient water use, reduce pesticide dependency, and integration of digital and artificial intelligence tools. Combining breeding with AI, big data, and precision agriculture. These are things which I'll briefly touch upon, and I've intentionally put few of them randomly, and then few thoughts may come now and again just to reinforce the thought. Next slide, please. Yeah, yes. Smart breeding is a modern breeding approach that leverages advanced technologies like big data, artificial intelligence, and genomics to make more precise and efficient selections. In my talk, I aim to create a dynamic forum. What to do is stimulate engaging conversation about the latest developments. That would have been possible if I were to be there physically, but then online I have to just be happy with what I share. Provide valuable perspectives from experts pushing the boundaries of breeding techniques. Explore the significant trends, hurdles, and exciting innovations impacting the industry. Because I worked in the industry for a long time, I bring the industry perspective to the smart breeding process, because these things are expensive and only the private sector in collaboration with public sector can make it a success. Connect researchers, policymakers, and industry leaders, encouraging partnerships for a smarter future. That's what I mean by partnerships. Next. Structure is, I am just talking smart breeding. What is it? It combines biotechnology, genomics, digital tools, and sustainable practices, enhances crop resilience. Morning, somebody talked about resilient crops, productivity and nutritional value, technological innovations like genomic selection, genetic engineering, data analytics, and AI. Case studies, I will just go through and list few of the things with success stories in improving crop performance, sustainability, smart breeding integrated with ecological practices, global collaborations, partnership among researchers, industry, and policymakers, and future outlook. This is what I will briefly touch upon. Next. Overview, what is it? Combines genomics, molecular biology, I already said. So just as I said, just to reinforce, advantages are there is precision, there is speed and sustainability in crop improvement. And then core principles are precision genetics. It uses GWAS, QTL, mapping for trait targeting, then high throughput phenotyping, imaging, robotics for real-time data, and genome editing, CRISPR-Cas9 for precise gene modifications, and data-driven decisions. AI and ML, it is machine learning for predictive breeding. Innovations, again, we have omics integration, genomics, transcriptomics, proteomics, metabolomics, example, grape genome sequencing for drought tolerance and aroma. I'll just go on giving few relevant examples as and when I come upon those. Next. Yeah, this is how it is. I'm just taking it from modern agriculture. I have put the reference there. So previously, as I said, the breeders were doing artificial selection. Molecular breeding happened then using molecular markers. When we go for smart breeding, we have AI, artificial intelligence coming in, and so many things which I already mentioned in the previous slide. Next. Yeah. So here I have to, this is from Trend. I have put the reference there. Those who want more details, they can easily access this. This is from last year, October, Trends in Genetics. This is AI-enabled plant breeding, artificial intelligence-enabled plant breeding. In the first panel, it is source of germplasm from a gene bank and then used directly are subjected to omic analysis to generate big data. That when we come to the next panel, then that could uncover genes and predict breeding values. That's very predicting breeding values. That's very big progress, I would say, which would characterize the gene editing targets and optimal lines for developing next generation cultivars. You see there between the last panel and there are 4 panels here. In between, I have put the smart breeding platform. That's where things start happening. Next. Yeah, designing crops I have put here, genotyping and NGS, SNP detection, genome and gene editing, epigenetic analysis, plant microbiome engineering, precision breeding, phenotyping, directed evolution, all those things I have just listed there. I'm not going to read all those, just for putting things together. This is how future crops are being designed. Things are much more different now than what they used to be previously. Next. Yeah, we'll come to that. With that, we come to marker estate selection. That's, of course, a precision breeding. It helps in targeted selection, helps to select superior individuals without directly measuring the trait. Increased efficiency, speeds up breeding by selecting at seeding stage and improving accuracy for traits with low heritability or difficult measurement. As breeders, we know what it is to improve traits with low heritability, but here marker estate breeding can help in that. Accelerated improvement facilitates faster integration of beneficial genes into elite lines through MABC. Enhanced outcomes contributes to development of improved crop and livestock varieties with enhanced productivity, resilience, and quality. Yeah, next. Genomic selection, what it does, utilizes genome data, considers thousands of genes simultaneously, eliminates the need for phenotypic evaluation in early generations. That was the previous trend. Employs high density SNP arrays to accurate genomic predictions. Genome editing, you are all aware, and then this is being applied in several crops already. The CRISPR-Cas9 is genetic scissors. These two ladies who developed the technology, they were awarded Nobel Prize, you are all aware. Applications of genome editing, enhancing pest resistance in tomato and potato. As I said, I will give few crop-specific references in each of the techniques. Enhancing pest resistance in tomato and potato, it is being possible by genome editing, improving shelf life in strawberries and mushrooms, and boosting nutritional values such as higher lycopene content in tomatoes. And then there are CRISPR-edited bananas with improved resistance to Panama disease. Next. Yeah, this just I have tried to put what I am saying in a pictorial form. Genomics-assisted breeding. There you have marker-aided breeding, genomic selection, genotypically, genotyping by sequencing, and speed breeding. I think someone is going to speak about speed breeding. De novo domestication, accelerated genome editing, novel CWR is crop wild relatives. Integration can happen. And then genome editing and phenomics and artificial intelligence. Yeah, next. This is called climate smart breeding. Double haploid is another technique, offers speed and precision in breeding. DS lines are used for disease resistance breeding and hybrid seed production in tomato, capsicum, and cucumber. It enables rapid generation of homozygous inbred lines for hybrid development. DHS are ideal for genomic studies and marker estate selection due to genetic uniformity. And they combined with speed breeding, DH can further accelerate the breeding cycle. As I said, most of these are trying to help you in predicting the performance and making things faster. That's why we call it as speed breeding. Next. So I talked about speed breeding and DH. They can work complementing each other. What is speed breeding? Many of you are aware. It very pioneering work has been done in Australia in wheat, basically. What it does is accelerates plant breeding by creating optimal conditions for rapid growth and reproduction. It involves growing plants under controlled environments with artificial lighting, temperature, and humidity. Using 22 to 24 hours of light promotes continuous photosynthesis, shortening the time between germination and flowering. Induces speedier flowering, often combined with MAS, facilitates quicker production of inbred lines in crops like tomatoes, peppers, and cucumbers. Of course, goes without saying, it saves space, water, and inputs in controlled environment. Integrating speed breeding with precision phenotyping and genomic tools is a cornerstone of future-proof horticulture. Next. I'm just trying to go through quickly so that I leave some time for discussions if needed. Yeah, speed breeding, it is more explained as a kind of pictorial thing. In conventional breeding, what we do, line development takes 8 to 12 years, field testing 3 to 5 years, and then trials and another 1 to 3 years. That doesn't happen. Here, speed breeding, you see the comparison, how it speeds up things. Line development takes, instead of 8 to 12, it takes just 2 to 4 years. Then field testing, of course, that has to happen, how to do that elaborately in the different environments, and trials and other things, it will save. I think basically maximum time is saved in line development because of speed breeding. Next. Yeah. Digital agriculture and smart breeding, high throughput phenotyping platforms. What they do, capture real-time data, phenotyping examples, hyperspectral imaging. I don't want to go into all those. I think those interested can refer to those. I'm only trying to list all the possibilities, what all are possible. AI and machine learning, AI algorithms analyze large data sets to improve crop breeding, AI-driven predictions, AI breeding line performance prediction, that's most important. AI optimization, it optimizes genetic crosses for maximum gain. So it will tell you what are the crosses you need to make based on the prediction. Next. Sustainability. Now we talked about how does it help in sustainability? Resource efficiency. Development of drought-tolerant and salt-resistant varieties minimize water usage, but done with smart breeding, that can be done faster and more effectively and efficiently. It reduces chemical usage because when we talk of breeding for disease and pest resistance, that definitely reduces the use of chemicals. Climate resilience, crops bred for heat and flood tolerance are better equipped to thrive in adverse weather. Nutrition enhancement, biofortified crops address micronutrient deficiencies, improving global health concerns or outcomes. Next. Innovations across technologies. I thought I'll just put a small brief on this. These are the things I have elaborated in my paper, which is there already. I have sent it. Genomic technologies, genome editing, precision modification using CRISPR in tomatoes, extended shelf life and improved flavor. I'm trying to put the crop and the traits which have improved. RNA interference, papayas, mealy bug resistance they have developed. High throughput phenotyping, drones, sensors, and hyperspectral imaging enable real-time trait evaluation for drought resistance. AI and machine learning models predict trait environment interactions, accelerating breeding decisions in grapes and citrus. Those crops, I think that's very, very valuable because they're long-term, long-duration crops. Omics approaches, transcriptomics unlocks stress-related gene pathways in apples. Metabolomics enhances flavor and aroma in strawberries and melons. Next. Challenges and opportunities. Because when we talk of things, we know everything is not as it is claimed to be. Challenges are there are regulatory barriers. Genome-edited crops face inconsistent approval processes globally. Initially, it was said genome editing really does not require. CRISPR-Cas is free of all regulation, but then there are inconsistent approval processes. Costs are high. Advanced technology remain inaccessible to smallholder farmers or breeding communities or companies. Public perception is ethical concerns and skepticism about biotech crops still are there. They are the ones which hinder adoption. Opportunities are for expanding collaborative research and technology sharing initiatives. That's what I meant. Unless the collaborative research and technology sharing happens, these things cannot make much progress. Developing cost-effective open source tools for genome editing, educating consumers on the safety and benefits of biotech crops. That's very, very important. Future directions are enhancing genome editing, expanding applications of CRISPR and synthetic biology for complex traits, integration with smart farming, combining breeding advancements with IoT, precision agriculture and automation, and policy harmonization, establishing international standards for genome-edited crops to facilitate global trade and adoption. But this is a long-term thing that has to happen globally. Next. Future perspectives. The integration of smart breeding with vertical farming. I think someone in the morning spoke about vertical farming. That's the thing that is coming up now. Aquaponics and other innovative cultivation systems hold immense potential. Future research should focus on enhancing genome editing tools for polyploid crops, which have not been explored so much as diploid crops, expanding the use of synthetic biology, and opportunities for global collaboration. They are plenty. In what areas? Scaling innovation, expand public-private partnerships to disseminate improved varieties, capacity building, invest in training programs for breeders in developing regions, policy support for regulatory frameworks that promote genomic tools and IPRs. These are very, very important. Next. Yeah. So with all that, this I should have put in the beginning. What does smart breeding mean? I'm trying to emphasize again the partnerships here. it is, as I said, initially it was said plant breeding is an art. Then they said plant breeding is an art as well as science. Then they said plant breeding is an art, science, and business of improving crops. So I thought, let me try to put a comprehensive definition. This is what I have tried to put together. The purposeful modification of plants for a speedier creation of innovative varieties with desired trait blends, combining conventional methods with advanced science and techniques of marker-arrested breeding, genomic selection and gene mapping, genome editing, speed breeding, precision phenotyping, and marker-arrested selection to develop crops that can better cope with emerging pests, diseases, and climate changes. Please note that combining conventional methods with advanced science. Conventional methods, there is no replacement. Whatever you do, ultimately, these have to find application in the conventional breeding. That point, I think we should not forget. Plant breeding and breeding research are a collective responsibility of public organizations and private enterprise, working together on market-driven plant breeding. We call any plant breeding we do has to be market-driven. It has to be driven by the market, the traits required, the people, consumers' preference, the traders' preference. But
Check,
unless
check.
we do that, the products will
Check.
not be. Yeah. Next.
Hello.
Am I running out of time, Chairman? Or I'll hello.
So one or two minutes there.
Okay, I'll wrap it up. Case studies I mentioned already. Bananas, CRISPR to combat Panama. That was already mentioned. Citrus, CRISPR-edited varieties resistant to Honglongbing. It's a disease. It's also known as citrus greening or yellow dragon disease. It's a bacterial disease. Marker-arrested selection for resistance to scab and fire blight in apple. Avocado breeding for higher omega-3 content. Selection for increased flavonoid content in cocoa. Lettuce varieties bred for vertical farming. Genome-edited grape vines resistant to powdery mildew. AI and genomics, heat and drought-resilient vegetable crops. That's being done in companies in Netherlands, which I have personally seen, and that is going on at the moment. Next. Conclusion. Smart breeding is transforming horticulture, addresses critical agricultural challenges, promotes sustainability and resilience, fuses innovation with eco-friendly practices, ensures equitable agricultural progress, unites researchers, policymakers, and industry stakeholders. That's very, very important. Fosters global partnerships, utilizes cutting-edge science, and protects our planet while feeding a growing population. Call to action is cross-disciplinary and cross-border collaboration is essential. Realize the full potential of smart breeding for a better future. That has to happen, again, I am saying this, through public-private partnerships. I think this is the last slide I have. Thank you. Is the last one? Yes, thank you. Thank you, Dr.
Thank you, sir. Thank you, sir. Thank you very much.
Yeah.
Quickly, couple of questions.
Yeah.
If anyone has.
Yes, one or two.
Yeah, there is one question. Please reach the mic over there. Sir, there is a question for you.
Okay.
Very good afternoon. Am I audible,
Good
sir?
afternoon. Good afternoon.
Yeah, quickly, my name is Amit Kulkarni, and I am a sound-based energy practitioner to my plants. One thing, there is a certain amount of changes which we've been observing in the plant during the growth cycle. My question to you, sir, is you were referring to something called spectrometer analysis. Do you have any such example to showcase here on this screen today? If it is possible, I would like to understand the differences between the pre-application of the genome sequencing and post-genome application. I would like to see the spectrometer analysis or hyperspectral analysis graph. That's what my question is. Thank you.
Yeah, good question, Dr. Kulkarni, and I'm happy you asked it. Yes, there are indications or examples to say, like you said, the plants responding to sounds. I also was reading some time back a very interesting paper saying if you play a particular music, plants grow very well and happy and healthy. But the spectral part, I have references. I can share them with you. Let me admit I am a conventional plant breeder, but out of my own interest and interest to know things, I have tried to study that. I have references. I can share them with you. If you can please share your phone number or mail ID, organizers can send it to me. I can share those responses to you.
Thank you so much. The point is like I would like to understand, or have you got any opportunity to see the changes in the spell, the cell level changes? What I mean to say is, did you observe any changes in the cell size or the structure altogether when you applied the science?
No,
This is purely because
Yeah.
I tried it in onion. We have something called teen patti onion. I tried it on the onion. The cell structure totally changed. I did the microscopic observation, but I did not get an opportunity to have a look on molecular level. Probably scientists like you could help me get there to understand the cellular level changes. My name is Amit again. My number, probably if you have pen and paper, probably I can
I
share
can
it
note it
with you.
down. Tell me.
Yeah, 99
99
72
72
59
59
10
10
14.
14. Amit Kulkarni, Dr. Amit
Yes,
Kulkarni,
yes,
right?
sir. Yeah.
Thank you.
Thank you.
Thank you, Dr. Amit.
Thank you.
I think in
Thank you,
looking to the
Amit.
time and management thing, we can discuss individually later. Yeah, thank you.
Thank you. Any other
Honest
question?
honest answer is I don't have any such personal experience, or I have not conducted any studies, but I can dig out things and know from people whom I know who have been conducting such studies and pass on that information to you, Dr. Amit. Is that okay?
One other quick question if anyone has. Okay. Sir, thank you very much for your lucid lecture as usual. Very comprehensive lecture covering all aspects of the topic. And
Thank you, Dr. Fakhruddin. I think
national level trainer in the plant breeding domain. And it was our fortunate to listen to him. Thank you, sir. Thank you very much.
Thank
We'll
you,
move
Dr.
on to the next lead speakers. Now we have I have an opportunity to call Dr. K.V. Ravishankar, Principal Scientist at IAHR, to deliver his lead talk. Dr. K.V. Ravishankar is a very experienced principal scientist at IAHR. Yeah, he combines multiple domains, crop physiology, breeding, genomics, and data analytics. He had an opportunity to train dozens of PhD students cutting across universities, a large number of JRFs and SRFs and research fellows and in his laboratory. It's in fact a breeding ground for the breeders and molecular biologists. And there's a lot to say. He's number one in many things. His work in this mango is type work for India, and a lot of projects from basic work to advanced genomic applications. Thanks for accepting our invitation for this lead speaking. Sir, over to you.
Thank you very much, dear Dr. Fakhruddin and my colleagues and my student friends. So, what I am going to tell another, whatever the slideshow. Yeah. So, what I am trying to. Okay. So in whatever the given time, allotted time for me, I just give the glimpse of the methodology, analysis, and few example which has been done in the fruit crops. The major problem with fruit crops is the long juvenile phase and the long generation time and high heterozygosity. And also it lacks a lot of genomic resources like molecular markers and system biology approach. So the main aim of these functional genomics or the genomics in this area is to identify functional markers linked to fruit trait. That is essential for speed breeding or genomic selection or marker assisted selection. Here, what the approach is, we need to use multi-omics modeling based approaches to identify candidate genes, QTLs, and predict the markers for important fruit traits like the pale color, pulp color, or quality aspects like aroma, nutrient content, and the shelf life, etc. To give the nutshell, what are the genomics or the multi-omics used in fruit crops is one is genomics, transcriptomics, metabolomics, proteomics, and phenomics. From this, we can use these methodologies on the mapping population or the diverse germ plasma. And then the analysis part, we can do genome association studies or modeling to identify the QTLs, genes, markers, and regulating elements like transcription factors or the promoters for the fruit quality. And these functional markers or the genes which is identified can be used in marker-assisted selection or transgenic or in CRISPR-Cas or to improve the cultivars with desired fruit quality. So one of the most uh, the advanced technology, I would say, in this all of marker-assisted building is in next generation sequencing method. See, this next generation sequencing method has helped both the proteomics and metabolomics, because all the whatever the genetic variation which we find in the nature all come from the differences in the genotype. That is basically differences in the sequencing. So there are three methods generally used in fruit crops. One is genotype by sequencing or whole genome resequencing. Second one will identify whole genome sequencing itself, and another RNA-seq. So using this, we can develop linkage map or association studies and draft genome or expression profiling to identify QTL marker gene and of interest and marker assisted selection or whatever there. But what is these are the basically next generation sequencing opened up a lot of the other areas, both the bioinformatics and marker assisted selection, breeding, and also other omics. Like without sequencing, you can't do good proteomics and good metabolomics also. So this is the basis of whole lot of things which we call now genomic selection or speed breeding, whatever possible now. So the two strategies are being employed in food crops. One is we have a mapping population, which you can see here. Either you cross contrasting genotypes and develop F1 mapping population, because most of the tree crops are heterozygous in nature. So F1 population itself is segregating. And other go to the natural population and look for the variation. So either you do QTL mapping or genome association studies, identify the markers or traits which is for the fruit quality, and then use that in genetic engineering or molecular breeding or phenomena or new phenomena, whichever is possible nowadays. You have so many things, CRISPR-Cas and knockout genes, all those things. So expression of profiling, that is RNA-seq. So I'm not going to deal with much. Here we are using contrasting genotype and to try understand what are the genes or the metabolics which are responsible for that particular trait. To give you the example, you can see here, like either you have a colored passion fruit pulp or colored peels. See what the people have done. I'm just giving the example. They used both RNA-seq and proteomics to identify the genes, proteins involved in anthocyanin pathway and sibyllinated flavonoid pathway and polypropylene pathway. So using this, you can identify what are the genes which gives for the red peel color in the pomegranate or the white peel color in the. So coming to the similar, some example, you can see in the what is the dragon fruit, the betalin and white pulp. So you can see that you can see the pathway which are involved in anthocyanin and betalin pathway. See, for example, in RNA-seq in mango. So here, that whatever we see, three colors we generally see in the mango: green color, yellow color, and red color. So green color is basically because of the chlorophyll, and yellow color is basically because of high amount of carotenoids, and the red color is because of both carotenoids and anthocyanin. And coming to understand the shelf life of the banana, so there is one hexanol, new natural, the anti, this is called, which increases the shelf life of the banana. They just saw that what are the genes which are involved in increasing shelf life. So they could identify some of the whatever the hexanol, the new natural compound which increases the shelf life. Here the result shows that increase in decreases the ethylene biosynthetic pathway and decreases cell wall degrading enzymes. So that you can see the first row, you can after 24 days of hexanol treatment, you can still the bananas are slow ripening and they look good. And there are several examples for the peel color and pulp color which are involved in, you can see that they are involved in carotenoid biosynthesis pathway or genes involved in anthocyanin pathway. Yeah, you see that these are the things. So Bain genotype by sequencing is one of the most widely used genotyping assay. Here, what is happening is the reduced representation sequencing at low cost, and genotyping without reference genome is also possible because most of the pre-crops don't have the reference genome, and thousands can be detected. For example, when you do SSR markers, we are trying to do it with few hundred markers. Here, thousands of SNPs are detected between the parent and also in the mapping population. See, application is here the SNP, QTL mapping, or haplotype maps, or one can use for the QTL mapping and also in and also you can identify genomic regions which are responsible for the particular trait. Using these traits, using this genomic region, we can identify genes which are residing in those QTL regions and look for their mode of action or functions in that particular QTL region. And also we can do the diversity analysis. So coming to the QTL, here we do two types of QTL mapping. One is QTL mapping using, we do genotyping whole the recombinant inbred lines or whole mapping population, or we take sometimes QTL-seq. Here extreme phenotypes is used in QTL-seq analysis or bulk segregant analysis. The major genes are identified in this particular QTL-seq, but in QTL mapping, you can identify both major and minor QTLs. For example, one of our studies, we had QTL mapping for the guava fruit, peel color and pulp color. We could identify 15 major QTLs and one minor QTL for the both pulp color and fruit color. Fruit pulp color and peel color. And the similar studies were done by the Dr. Kuhn in US. They could identify 19 QTL for many of them, including embryo type, both and beak color, pulp color, branch habit, linkage map. These are the very few examples which you can find in the tropical fruit crops. And similar way they did, see the papaya is used for both ornamental purpose. This is the purple papaya and also for the using the field also. So you have two types, green petiole and the purple petiole. They could identify one major QTL, and that major QTL has they identified 4 genes, 3 MIB transcription factor and chalcone synthase. All are involved in anthocyanin pathway. More. Okay, this is drought genome assembly. I'll not get into that because okay. So there are whole genome sequencing data is there for most of the molecular breeding, because once you know the whole genome sequence, you can identify what are the genes, because there is a synteny. See, all the animals or plant, they use same amino acids, and most of the genes, there are a lot of synteny. The functions are known. So you can go and identify those genes and you can work on that. So one of the other technology or methodology used in fruit crops is the genome-wide association study, identification functional SNP. Here there is this is basically to identify non-random association of SNP marker which is linked to some particular traits. So these are the methodology or technology used. Here you can take either GBS data or whole genome sequence data. Earlier people used to use SSR marker, but now it has become old fashioned because we need high density markers and the markers should be distributed throughout the genome. So people use GBS or whole genome resequencing. So some of the example which I can give you, in the whole GWAS in fruit quality improvement in banana, they could identify marker for seedlessness. So there they identified the one QTL region that contains 7 genes which are involved in seedlessness. One of the one is they are involved in most of them are involved in hormonal signaling and also the gametophyte biosynthesis. So that way one can identify the reason or physiology or biochemistry behind the seedlessness and also identify genes which one can mutate or do editing in the future crop improvement program. So some more the genome studies in the leaf wax content. Here they identified fatty acid genes which are responsible for leaf wax content in apple. So multi-omic approaches, which generally people started working on that. One is do genotyping, transcriptome, and also do the metabolomic. Like the color might come from carotenoid and also color might come from the anthocyanin. We need to know whether the peel color is because of carotenoids or because of anthocyanin or flavonoid. So then you integrate all those into the analysis. Then you identify the functional marker, and also that functional markers can be tested using WIX, that is, virus induced gene silencing, and also using CRISPR-Cas. So some of the examples for multi-omics approaches you can see here. The people have done fruit color and aroma in apple. See, most of the examples, unfortunately, from citrus and apple, that is temperate crops. And these are some of the multi-omics then. And also now there is a pangenomics that is similar to the genome association study, but here the whole set of the germ plasma is resequenced to identify the domestication, loss of genetic diversity, and also identify the structural variation in the germ plasm. So coming to the genomics selection, which people want to use that. Okay, I'll come to this. See, what is happening in genomic selection? You have a training population, and you have to do the training population, either maybe your germplasm collection or mapping population. So one has to do genotyping and phenotyping. Then you have to do association studies to connect both genotype and phenotype data, and then you have to do selection of what you call genomic breeding estimated values, and then selection of individuals maximum with GBEV and make selection. See, you can go to the conventional breeding. Generally, you take for mango to develop a variety, it takes almost 28 years. For example, you need to do 4 years process to have a good number of population, and it's very difficult to do it. And you have to evaluate 20 to 25 years, and then you have to multiply and release as a variety. So it takes 28 years. So what happens in the genomic selection, one can do the crosses 4 years, that you can't reduce it, but that can also be reduced in the speed breeding nowadays. And evaluate for 1 to 2 years. How this is possible? Because we have the GB value, that is genomic estimated values, breeding estimated values are there. Based on that, you can do the evaluation and propagation in 4 to 5 years. So instead of 28 years, one can breed the tree crop within less than 10 years. So that way you can reduce the time almost 1/3. And you can still reduce if you have a speed breeding in that first crosses which you make. So advantages of this genomic selection is reduces the selection cycle duration, increases selection accuracy also, and intensity, efficiency, and gains per unit time is very high in the genomic selection and enhance the rate of genetic gains. So whatever the data which we used in GWAS or QTL mapping, one can identify the functional marker. So one can use these functional markers in the CRISPR-Cas, that is in genome editing. So you have different methodologies like CRISPR, TALEN, JALEN, FN, JFN. They are the different systems in genome editing. So the only disadvantage with genome, so you can use any trait like abiotic, biotic stress, or nutrient enhancement, everything. But some disadvantages also there. There's off-target effects will be there, and to do the CRISPR-Cas or genome editing, one has to have the good tissue culture, high frequency transformation method should be there. So these are the two backdrops in fruit crops, which we don't have the tissue culture developmental protocols, and the delivery system is also not there for the fruit crops. So some of the example for the CRISPR-Cas is already there. So here they have edited the beta carotene biosynthesis gene, and you can see the high carotene. And also another one in apple, they have edited the CGN2 silencing, improves the resistance to bacterial the disease. Okay, there is some of the one they have developed a pink pineapple by improving the carotenoid, and also they have edited the apple. They have silenced whatever the polyphenol oxidase enzyme, so you can have the non-browning apple. Okay, this I already discussed this. And also there are crop-specific databases for tropical fruit available. One can see what are the genes and what are the methodologies and what are the markers available for genome editing or marker-assisted selection. So one can use these crop-specific databases for tropical fruit. To summarize what I would say, like the most important is development of cost-effective functional marker that is possible using either germplasm or diversity analysis or QTL mapping. And once you identify the functional markers, you need to see allele-specific variations in this functional markers. And also GWS QTL analysis are complementary approaches when can combine, inform genetic correlation with phenotypic traits. Different omic approaches like GWAS, genomic selections, and all the multi-omics, phenomics, everything will help in understanding genetic grain and speed up crop improvement in programs. And we need to use all the new breeding technologies like gene editing, cisgenic, epigenetic approaches to enhance or improve the fruit quality.
Nice. Thank you, sir. Any questions? Quick couple of questions. Anybody has any question? Okay. Thank you. Thank you, sir. Thank you very much for your nice talk, starting from biparental mapping to multi-omic approaches with the clear case studies and examples. It was very good talk. Thank you very much. I now request Dr. Lohitashwa, the second lead speaker of our this session, to take over the session and deliver his talk. Dr. Lohitashwa HC is a principal scientist, a seasoned plant breeder who started working from his early in the pulses breeding of chickpea, pigeon pea, and later on wheat, rice, maize crops. He has a history of releasing a lot of varieties. His varieties have been released both at state level and national level, and many of them are in the seed production chain. And he's responsible for guiding a lot of students in his laboratory. The beauty of his work is he combines conventional plant breeding and molecular biology, the genomic approaches. That way, besides releasing a lot of varieties, he has also benefited a lot of students. Over to you, sir.
So thanks to Dr. Fakhruddin. So good afternoon to all of you. So hope you will be able to spare 10 or 15 minutes of yours. Huh? So it should be interactive more of, and I expect at least a few good questions from student side. So of course, my area is genomic selection, but Satish wanted me to speak on pre-breeding. So I have prepared my presentation for 30 minutes. Unfortunately, I received a call from Satish and restricted my presentation to 15 minutes. So we have replaced several slides, and then I'm just going to present all of my topic in just 15 minutes time actually. So this is pre-breeding strategies for improvement of horticulture crops. So you all know that climate change is imminent. So we are all experiencing ill effects of climate change globally. Okay, so some areas which never used to receive rainfall have received the severest form of downpour, especially Doha during World Cup, Soccer World Cup, and then Sharjah. So they all received the heaviest possible rainfall, and some African countries which never witnessed rainfall in their lifetime actually witnessed heavier downpours. And then, in some areas actually which used to receive the normal rainfall are experiencing drought. So, and then, you just talk about America and then Europe. So severest form of drought in the recent past, those countries are experiencing. So this is imminent. We need to somehow manage these ill effects and produce food to meet the nutritional requirements of the ever growing population, not just human beings and also animals actually we need to take care of. Yeah. And this climate change has also resulted in emergence of several insects, pests, and diseases in highest possible proportions. And new pests and diseases are also emerging. So we need to somehow breed varieties which are climate resilient and without compromising with the yield levels or profitability. So that is the utmost requirement in the present day agriculture. So considering these effects actually, these effects and several requirements, so pre-breeding is the most important aspect of plant breeding because, so it sources novel genes or useful traits from several other genotypes like local varieties or wild species, wild relatives, and then several distant relatives from primary gene pool, secondary gene pool, tertiary gene pool, and quaternary gene pool to breed varieties which are climate resilient. So, okay. The term pre-breeding was coined by Dr. Rigg during 1984. So essentially, pre-breeding aims at transferring useful genes or traits from otherwise unadapted genotypes into intermediate genotypes. So from the wild relatives or wild genotypes, and these intermediate types are distributed to several other breeders or stakeholders to use them as new varieties or use those resources or intermediate types in their breeding programs to transfer useful traits into the genetic background of varieties. So, and then, it also results in useful genetic diversity, not just transfer of useful traits, but also it addresses issues related to genetic diversity. In one of the slides, actually, you can see the kind of genetic diversity that resulted in just one wide cross in the case of tomato with respect to leaf structure and leaf shape, fruit structure and fruit shape. So much of genetic diversity, just one cross produced. You can experience, you can feel it in one of my slides in the later stage. So especially pre-breeding, when we talk about pre-breeding, so it encompasses the steps or stages before release of the variety. Before release of the whatever we do, so it all comes under pre-breeding, and then essentially it links it links, the relationship between genetic diversity that is existing in any crop plant with the breeding programs. I'm just showing this particular slide. You can see the kind of genetic diversity that is present in wild relatives of pigeon pea. So some of the genotypes you just cannot say that they are the relatives of pigeon pea. I'm just showing this one, and then I had some slides on tomato also. I have just removed. Fortunately, Dr. Dandin showed the enormous genetic diversity that is existing in different vegetables, fruit crops, and in some ornamental croplands. So fortunate actually. Then Dr. Ravi Shankar talked about, and also Dr. Angdi talked about modern day breeding programs, which we can essentially make use of in pre-breeding also. I'm just going to touch upon those, and then I'm not going to enter into details of all those breeding aspects. So when we talk about modern day plant breeding program, actually they are all constrained by the availability of lesser genetic variability. Modern plant breeding means exhausted the amount of genetic variability that was existing earlier actually. Either you take wild species or wild relatives actually. So plant breeding, so made use of all those modern day plant breeding, made use of all those genetic resources. And then, we are experiencing limited genetic variability. And then, we are also experiencing problems with respect to development of varieties with resistance to different diseases and insect pests. And then essentially we need to think of, germplasm diversification to address these issues. And then, we need to target useful genes from wild relatives and wild species.
Turn this side. Turn this side and yeah.
Okay. Just, this particular slide is with respect to, comparison between traditional breeding and pre-breeding. So essentially traditional breeding, so talks about the profitability. So it's about, development of priority varieties or hybrids, whereas this breeding is with respect to enhancement of germplasm, that is addition of useful traits into the genetic background. It leads to the development of improved varieties in the case of traditional breeding, whereas pre-breeding talks about broadening the genetic base to address several of the issues like climate ill effects. End product is used as a commercial product like varieties or hybrids, and then end product is used as a parent for developing new varieties. And it's a short or medium term breeding program, traditional breeding is, whereas it takes around 10 to 15 years essentially. Sorry.
I'll turn that side.
Okay. So let me talk about steps in pre-breeding. The first step is transfer of useful traits, that is making crosses actually. So first of all, actually, we need to know which of the wild species or wild relatives or land races house the useful genes for several biotic and diabetic tests actually. So we can employ both morphological and molecular markers to to locate whether useful genes are present in any particular species. Second step is integration of these new traits into the genetic background. So that is through repeated backcross breeding program and congruity backcross. In the case of a repeated backcross breeding program, we don't talk about the fertility because it essentially involves the primary gene pool genotypes. Whereas in the case of congruity backcross, we involve secondary and tertiary, and sometimes, quaternary gene pools also in the breeding program. So essentially there we face a lot of problems with respect to fertility. So here we need to talk about selection of fertility restoration also. So since we give importance for fertility restoration also, so here is just transfer of useful traits. Transfer of useful traits is with respect to congruity backcross. So in the case of congruity backcross breeding program, so these are the 4 different steps which are involved. First one is we need to hybridize the wild species or wild relative or local landrace with the existing genotype, which is used as a recipient parent in the breeding program, and then backcrossing to the cultivated parent. So, and then these hybrid crosses are repeatedly backcrossed to the recipient parent with the selection for partial fertility restoration and useful traits. In all the stages, actually, we need to concentrate on retaining that useful trait, which is being transferred, and also we need to select for partial fertility. So they are the 3 important criteria which we make in the process of recurrent backcrossing. Is one is increased fertility we need to achieve, and then we need to retain the intraggressive trait that is received from the wild parent, and then we need to look at the agronomic similarity with respect to that recipient parent, and then we need to produce these backcrosses on a large scale and give it to the stakeholders. These are the steps essentially we talked about with respect to pre-breeding. So you can just see it takes around 5 to 80 years, right from hybridization to selection, and then making available the intermediate types to different stakeholders for use in the development of new varieties. So I am just going to talk about some pre-breeding efforts in horticulture crops. Primarily, I'm going to discuss on several efforts that are available in tomato and other vegetable croplands. So first of all, you just can look at the use of Solanum pimpinellifolium, and then Avocates and Peruvianum for transferring resistance to bacterial wilt and late blight. And this Hawaii 7996 was the resistant variety with the bacterial blight. So developed through pre-breeding program essentially. And then the second one which you can talk about is transfer of tolerance to heat and insect pests. So that is aphids and thrips tolerance from Capsicum baccatum and Capsicum chinense and Capsicum frutescens genetic background into cultivated genetic background. And the next one is about the transfer of resistance to shoot and fruit border from Solanum torvum and Solanum virum into with resistance to shoot and fruit border. Coming to the citrus, from Citrus latifus and Poncirus trifoliata, we have transferred resistance to Huang Long Ping, that is HLB, otherwise called citrus greening. And then coming to strawberry, from Fragaria chilensis and Fragaria virginiata, so we transferred resistance to or tolerance to cold and resistance to fungal diseases. So these are the 2 genotypes, Sanioi and Albion, were the genotypes or varieties which were developed through pre-breeding program in strawberry. Coming to banana, GCT-CB2180 was the triplet developed in banana with tolerance to Physarum wilt. So we made use of Musa acuminata, a subspecies Malacensis with resistance to tropical race 4 of Physarum wilt. And in the case of cucumber, the resistance to downy mildew and powdery mildew was transferred from Cucumis cysticus and Cucumis hardwicki. And then Genian number 4 was the variety hybrid released in China with resistance to these diseases. So coming to pumpkin, resistance to viruses like CME, pumpkin variety, pumpkin virus, and then Juchini yellow mosaic virus So we sourced genes from Cucurbita eucadorensis and Cucurbita okichobiensis. So these are the 2 wild species which were used essentially to transfer resistance to these 3 different viruses. And this is the one actually, Menina basilaria is the variety actually, which is carried resistance to these various diseases, developed using pre-breeding efforts. And then coming to challenges in pre-breeding program actually, so these are the essential or main challenges with respect to pre-breeding actually we need to essentially address. The first one is genetic barriers to hybridization. So this is imminent because we are employing wild relatives, wild species, and then distant relatives, from sometimes from tertiary gene pool, quaternary gene pool. So we face problems with respect to fertility. So in that sense, actually, barriers are there, but there are several ways present to overcome these barriers actually. And that is also, there is also one problem with respect to ploidy levels. So many of the wild species and wild relatives have higher polyploidy. So sourcing genes from such high polyploid genotypes and transferring those resistance genes or some useful genes from so higher ploidy genotypes to genetic background is a bit difficult. So coming to the second problem is with respect to linkage drag. So essentially, useful genes are associated with some undesirable genetic background. Of course, we have marker set selection to help us in this particular process. So we can address this particular issue. Things are happening, and then especially in the case of tomato, so linkage drag has been addressed to the significant degree. And then, essentially, it involves long breeding cycles because, so we are employing wild relatives, wild species. Distant relatives are being used. So naturally, it takes 10 to 20 years sometimes. So next is phenotypic challenges. So it's there actually. Of course, Dr. Rangdi talked about several phenotypic approaches available, like and then coming to the next one is I already talked about this, the polyploid nature of wild species. It's again severe problem in the case of transfer of useful traits from wild species to the cultivated genetic background. And then, let me talk about the use of or application of genomic assisted breeding in pre-breeding. So these are the steps actually which are essentially involved in application of genomic assisted breeding in pre-breeding. So first we need to evaluate the potential of donor lines for the presence of useful traits. And then we need to identify the genes in terms of QTLs or something like that. And then in the QTL region, actually, we need to source the candidate genes. And then we need to develop markers for these candidate genes and validate these genes. And then use these markers in the breeding pipelines to transfer useful traits. Let me talk about the first step, that is genomic characterization of wild relatives. Actually, we have whole genomic sequencing, next generation sequencing efforts are made available. And then, we have several papers right now recently emerging, wherein which we are addressing pangenomes. In the case of pigeon pea also, so pangenomic efforts are underway. So recently, Rajiv Varshney published one paper in pangenomics. Several subspecies are considered, and then useful genes are traced from several genomes, related genomes. So that effort is also being made in several croplands to source unique alleles from wild relatives. And then we are also making use of IRD marker selection is being essentially used in all the vegetable crops and also in some fruit crops, as Dr. Ravi Shankar listed. So the best case of transfer of physarimid resistance in the case of watermelon. So from Citrullus calisthenicis into the commercial varieties. And then, we are also employing markers that backgrass breeding program in the transfer of useful traits. So wherein which, we employ both forward and backgrass process actually. And then identification of novel alleles in the wild crosses, employing genome editing, site-directed mutagenesis, tilling, eco-tilling, and other approaches. And then since, across the entire genome of any other cropland, as of now, we have markers to trace the presence of useful traits actually. So we can essentially make use of GWAS and genomic selection in the breeding process, as Dr. Ravi Shankar talked about. And then genome editing is another area actually which can be efficiently employed in breeding efforts actually. So several of the wild species actually, so which can be made or which can be converted into cultivated form by changing the nucleotide sequence. So those efforts are also being using CRISPR-Cas9. So we are sourcing useful genes, and those useful genes are being altered using CRISPR-Cas9. So there are some significant developments with respect to different horticulture croplands actually. In all the 6 different crops actually, products are ready. So they are available in the market. So in both China, America, so Europe, so the genome-edited croplands are available in tomato, citrus, cucumber, apple, grape, watermelon, kiwi fruit. So coming to other techniques that are available to address several challenges with respect to pre-breeding or somatic hybridization. So speed breeding and DS technology, as Dr. Angdi talked about, so we can essentially make use of speed breeding and DS technology, so to advance generations rapidly. So rapid generation advancement techniques like DH, it's made available in several of the fruit crops and also in the case of vegetable croplands. And then another issue that is we need to address is with respect to precision phenotyping. So, non-destructive phenotyping with respect to root traits, it's being made possible actually. So we are making use of these things actually. And then we are also employing drones and also GA-based phenotyping to identify the trait of interest and advance the generations more quickly now. So this is one particular actually I just want you to appreciate, that is employing monogenic alien addition lines in transferring useful trait. You just see here. So they employed DNA markers, so which were present on all tomato chromosomes at an average interval of pi megabase. So they concentrated on 28 mass and then came down to 8 actually, and then from that actually they came down to only one genotype with essential trait. And I just want you to appreciate just in one wide cross, so much of genetic variability that has resulted in terms of leaf structure, leaf shape, and then fruit structure, fruit shape. You just see just one cross. And just see here actually, these two were the ones actually which carried drought tolerance transfer from wild species actually. And then you can continuously see the genotypes that LA 3122 and the one actually are consistently showing drought tolerance over the existing genotype actually. So showing highest water content. And just see the control with respect to the 2 lines actually which they came out with from this particular breeding program, that is MBL2 and MDIL 11.3. So there are several, it's because of time constriction actually, I'm just stopping at this stage. Actually, these are some of the important traits which we addressed with respect to several vegetable and fruit crops available in the literature actually. So I'm going to leave this particular presentation. Anybody can take it and use it for your purposes. So see, so many classical success stories you can notice in these cases actually in grapes. And then we have made use of NGS and pre-breeding. So, and there are successful examples of employing NGS to address several issues in some vegetable and fruit crops. And then just talk about the pre-breeding in tomato. Actually, several of the useful genes have been transferred from several wild species into cultivated genetic background. Just see the dwarf tomato seedling actually, Solanum plenelli carries useful genes for dwarfing nest actually. That dwarfness has been transferred from penally into cultivated genetic background. Just see, look at the kind of productivity that you can see in one particular genotype. So these are all the some of pre-breeding efforts actually in tomato. So coming to challenges and future aspects, challenges are with respect to inadequate characterization. That is one thing. Just because I already told you, they are essentially wild relatives, wild species. Characterization itself is a difficult task, and then there are most of them are polyploids, and then addressing such issues in polyploids is again a serious problem. And then funding. So for these issues, only at only at few centers actually you can think of addressing these issues. So pre-breeding efforts can be made only at few centers, that too in international institutes like CIMMIT or ICRISAT. And then coming to future aspects, we need to make use of genomic assisted breeding program in transferring useful traits. So coming to the key takeaways from this particular presentation is so we can not just transfer the useful traits into the local genetic background or existing genetic background. We can also increase the genetic diversity in different crop plants, which we noticed with respect to that tomato breeding effort actually. So thank you. Thanks for your particular time. Hope I was able to address several issues with respect to pre-breeding. So I'm open for questions from your side.
Thank you very much. Any questions?
Yeah. Thank you, sir. Thank you for elaboratory explanation about your research work. I have 5 different questions to you. Hope time permits me. I have 5 questions to ask.
One important question he asked.
Yeah, these are a couple of questions associated to the general topic. One is something associated to the ploidy technique, which have been applied in your studies. The question is like, has any studies or tests done on the native breed, be it vegetable or fruit, where we've been able to fix the genomic deficiency? Let's say, for example, we have our native chiku variety here, which is bred here in this region, let's say from Bagalkot. Was there any such example which is considered, or rather which is scientifically picked fruit or vegetable which is being considered for genome sequencing? That's one first question for ploidy technique. Second question to you is post-production of application of this genome sequencing or modification. Was there any such tests on human, were there any trials on human beings in case if there is any psychological balance or imbalance which is happening after consuming this particular food? Because we need to ensure that the necessary amino acids are being present in each of these food. This is a very generic question to you. Post application of genome transformations. These are 2 mixed questions. And third question to you is, as a
Sir, can you keep
yeah.
the question short? You can meet him.
These are all questions. These are all questions to the science world, basically. Probably
answer
anyone can pick up this question and ask.
He's available.
As a layman farmer, how do I know that post-production or post-application of this genome sequencing, will it be fruitful in my soil? Because I have not tested my soils according to your research. How do I get my yields properly aligned to your research? When you say this germination will happen, post-production germination of that seed will happen 96%, surely. Now, if I get 70% success in the germination, probably there is a gap in between my soil and your research soil, so on and so forth. How do I fix that post-genome application or ploidy technique, application of ploidy technique? Thank you.
Okay.
These are 4 concepts which were running in my mind during your session. Thank you.
So first thing with respect to genome sequencing. So genome sequencing is just knowing, the sequence of nucleotides that is present and employing that genome sequencing efforts in breeding varieties. So there, problem what you talked about, that is with respect to only genetic engineering. And in the case of genome editing, that is not an issue at all, but still, so we don't have permit to release the varieties as such, even from genome editing also. Although it is devoid of any issues that are that are available with respect to transgenics. So transgenics highly impossible in India. Genome editing products can be readily made available, but it takes some more time. So that's being taken care of with respect to several fruit crops and also with respect to several vegetable crops. Actually, people are addressing. And then coming to pre-breeding, it is nowhere different from traditional plant breeding. So we are employing just primary, secondary, tertiary, and quaternary gene pools in the breeding program. That's it, actually. They are all plant types only, essentially plant types only. And then local land races, actually, they contain useful genes. We are transferring or sourcing useful genes from such sources into cultivated genetic background. So no issues are available with respect to that. And then with respect to, if at all, you want to involve in these kinds of breeding programs, there are people. So especially Manish Pandey in ICRISAT, actually, you can join, you can discuss with him sometime. So RN, you can discuss with me anytime. So we can discuss over these issues. Actually, not an issue. It is no way different from traditional plant breeding.
Short question.
Sir, as you said, pre-breeding includes linkage drag is a major problem. If the gene is tightly linked with some undesirable trait, can you please mention what are the ways to overcome this problem? If it is really very tightly linked.
Okay, tight breeding, trait linkage is one issue actually. So, in several of the mark-casted backgrass breeding programs, that particular issue is being solved, addressed. But with all that, so we need to grow largest F2 population or largest F3 population to overcome that particular problem. That's being addressed through growing largest segregating population from such white crosses. It's happening. And other way is mark-casted breeding programs. Selection of the trait through the linked marker. So things are happening, no issues. Any
Okay.
other?
Thank you, Dr. Lohitasha.
So, thank you.
Yeah.
Thanks for your time.
He is available here. You can meet him and discuss with him.
So I'm available here till tomorrow evening. And then, thanks to Dr. Satish, thanks to Dr. Balaji, thanks to the chairman and co-chairman, the rapporteurs for their time. I'm so grateful for inviting me to present this particular issue before all of you. Thank you. Thanks for your time. Thank you so much.
Thank you very much, sir, for your wonderful lecture. So now we have another lecture by a very renowned person, Dr. A.V.D. Daroji Rao. He is now Associate Dean of YSR Horticulture University, Venkatagudalam, main campus. So he is basically professor and head of floriculture. Now he is heading the Dean chair. So he has a rich experience of teaching, extension, and also education. So he's guided more than 25 MSc students and 8 PhD students. He published nearly more than 150 research papers. So we are happy to have you, sir. Now I request Dr. A.V.D. Daroji Rao, sir, to deliver a lead talk on nanotechnology application in horticulture.
Sir, you have 10 minutes.
Thank you, sir.
Yeah.
Thank you for your concern. So I start my talk with a sacred saying that Patram Pushpam Phalam Toyam Yome Bhaktya Prayachati Tadaham Pratyupahritau Ashnami Prayatatmanaha. This is the sacred saying that you give either fruit, leaf, flower, or even water, but with devotion. That is more important. For a student or for an expert, the most important thing is we should have love and devotion towards the subject. So we, the horticulturists, we deal with all these commodities. Either we produce them or we utilize them. And I am very much privileged to stand before you, before such a senior people like Dr. Sharan Angadi, who happens to be the classmate of my guide, Anand Mukashi, and Dr. Balaji, Dr. Satish, Dr. Fakhruddin sir, and respected chair and co-chair of the session,
faculty members, and dear students. So my talk is again a smart talk to fit into this smart conference, nanotechnology applications in floriculture. Next slide, please. So nano, the word nano is literally means dwarf. And the experts of the units and dimensions, they have hide that word to mention 1 billionth part of the a meter. So science at nanoscale is exploiting. So that's very magical, and the nanoparticles in any molecule, it is there's a lot of difference. That's where the intervention of nanotechnology comes. Some of the renowned people in this particular area, Richard Feynman, he is renowned as father of nanotechnology, and father of green nanotechnology being Kattesh V. Katti, father of Indian nanotechnology, CNR Rao. So the size varies from 1 nanometer to 100 nanometers. So we deal with the particles which are in the size ranging from 1 nanometer to 100 nanometers. They have unique properties in terms of shape, in terms of surface, in terms of size, in terms of material. So actually the key or science of nanotechnology applications are embedded into this. For example, if you take dendrimer, which is a branched molecule. So the dendrimer can be anything, a protein or any metabolite, and that will be influenced by nanomolecule because of the branches and because of the space that it is oriented. So even shape, it has several unique shapes which can influence any biomolecule which the other molecules may not be able to do. The nanoparticles have a unique properties like high surface volume ratio, small size, high charge density, and better penetration into the cell. These properties are linked with one another. That's why their effectivity is dependent upon. So if you see the aspect ratio, high aspect ratio means width is less and width is more and height is less. So nanowires, nanoparticles together are made into wires. Then they are called nanowires, nanofibers, etc. And there are different states of nanoparticles, and there are different compositions, chemical compositions of nanoparticles, which are applied in various uses, either in floriculture or in general in horticulture or agriculture. The advantages of nanomaterial over conventional particles are high solubility, high diffusion rate, improved stability, slow degradation, and compatibility biologically, and controlled release. These are the advantages of nanoparticles over the traditional particles. Different types of nanoparticles are inorganic nanomaterial like silver, gold, etc. Organic nanomaterial, and particularly carbon nanomaterial, which comprises of graphene, carbon nanotubes, etc. and etc. These are the different material or molecule which are useful in the applications of nanotechnology side, either in floriculture or horticulture. The nanoparticles are synthesized in three major means, that is physical means, biological means, and chemical means. So physical means like electrochemical means or ultrasonication, etc. Biological means that is termed as green synthesis, wherein different parts of the plant can be utilized. I will tell in detail about it. And chemical means include mechanical milling, etching, etc. The basic methods are classified in other way, that top-down method where larger compounds are broken to nanoparticles, and bottom-up method where smaller particles are composited to larger particles. So nanoparticle synthesis is graphically represented in this chart. So green nanoparticle synthesis, as you know, either flowers or leaves, they contain diverse compounds. And these compounds are made to feed. That means the plant is made to feed a media wherein the targeted salt is included, and then it will be nano materialized within the plant biologically, and afterwards it will be extracted. So the chart also I will show in brief. The that entire process is called green synthesis. The green synthesized nanoparticles can be obtained through easy method and efficient method, and this method is named as eco-friendly method. Different phases of green synthesis of nanoparticle include initial phase, activation phase, growth phase, and termination phase, the details of which are self-explanatory, but whatever are there, this green synthesis include intervention of a plant material, be it a flower, be it non-flower. So here comes our main important part. Silver. When silver and gold are kept, what is chosen? Gold is chosen. So gold particles, gold nanoparticles are prepared by intervention of Plumeria alba, Clitoria terniata. Clitoria terniata is popularly known as Senku Pushpa. See, green synthesis of nanoparticles, how it is done. So this is one schematic representation where silver nanoparticles are prepared using Catharanthus roseus flower extracts. And this flow chart is also self-explanatory. But after including nanoparticles into the plant, they are also carefully extracted from that. So that is how these nanoparticles are prepared or synthesized through green method. Because our interest is mainly green method, I have detailed green method, but other methods, physical methods and chemical methods are also there. So these nanoparticles are applied, this technology is applied in various dimensions as indicated in this graphical diagram, from nutrition to plant protection, even from monitoring, where AI-based applications are there, and even for herbicidal preparations. One by one, the propagation aspect. So propagation aspect, it is used in vitro for disinfection process, for differentiation morphogenesis process, to include in the media by involving the salts based on nanoparticles or nanomolecules. So these are the different applications wherein you can see Gerbera jamsoni silver nanoparticles at a concentration ranging from 25 to 200 milligram per liter. It was found to reduce microbial contamination more effectively. In germination, zinc oxide nanoparticles and carbon-based nanoparticles were found to be more useful. Similarly, silver nanoparticles were found to possess or exhibit more efficient antimicrobial properties compared to conventional silver nitrate particles. So positive effects of nanoparticles are very much proved on plant growth and development by various workers, and it promoted seed germination, it enhanced photosynthesis, and all of them were proved scientifically with a large amount of data in various universities across the globe. Nutrition also. Nutrition aspects, nanofertilizers were found to be more efficient compared to the conventional molecules. The uptake efficiency was also found to be good with nanofertilizers. Nanofertilizer experiments were conducted by all horticulture people like fruit science, vegetable science, plantation crops and flower crops, medicinal aromatic crops, etc. So they have characterized, they have also studied microscopically that there were improvements by means of nano-urea treatments or any other molecule in its nano form was found to be more efficient compared to conventional particles. And even in crop protection also, the nano-based pesticides were found to be more advantageous, and they were found to confer systemic acquired resistance. So this is an outline depicting superiority of nanopesticides over traditionally used chemical pesticides. Here you can see a larger molecule, it covers a particular area. So if something is there, some spot is there in that particular area, then only it detects. Whereas nanoparticles, which are spread all over the leaf surface, can spread over that and it can give signals more efficiently. So this is, if this is understood, the applications of nanoparticles is very much understood. Even in post-harvest also they are useful for ethylene scavenging ability, etc. and etc. So nanoparticle, by means of having high electric charge, by means of these properties, it was found to enhance floral quality posterior to harvest, enhancing the vase life. For plant monitoring systems, nano sensors were found to be more useful. And these are all different nano sensor based technologies like single walled carbon nanotubes, fluorescence resonance energy transfer, etc. and etc. So nano biotechnology is also useful in genome modification. Precise genome editing was said to be possible by means of CRISPR-Cas technology, wherein nano-based products were being used. Gene silencing also made successful applications of nanotechnology. These are the leading institutions over the length and breadth of the world where nanomaterial are being researched. So these are the modes of entry of nanoparticles into the plant, and uptake and translocation of nanoparticles is possible by both apoplastic and symplastic pathways. So this is one example where insect is subject to different kinds of actions by nanoparticles in various organs. The advantages of nanotechnology are summarized as increased productivity, improved soil quality, and smart monitoring compared to traditional particles. But there are as well limitations which leads to nanophytotoxicity, reactive oxygen species accumulation, toxicity in beehives, etc. But these limitations can be overcome by some of the remedies like doing more research focused on these deficits and maintaining proper distance between beehives and to maintain buffer zones, etc. And some of the conclusions out of this presentation would be it hold a transformative and smart solutions because it happens to be the sixth revolutionary technology after mechanization, then smart farming, precision farming, use of drones, artificial intelligence based technologies. In that series, this becomes the revolutionary technology. And we hope that more and more applications will come ahead, and floriculture in specific and horticulture or agriculture in general will be much benefited by nanotechnology applications. Thank you. Thank you, sir.
Thank you very much. Any one question?
Yes, sir.
You spoke that nanoparticles will induce somoclonal variation.
Yes, yes.
So, like, is it proved in any other crops? What is the molecular mechanism? Is there anything on that?
In that research paper, they have not discussed about molecular mechanism, sir, but they found to have a significant change, significant improvement in somoclonal variations when they used nanoparticles over the, but they did not get when they used traditional particles. I can quote that
Thank
reference
you.
also.
Thank you, sir. I'll contact.
Thank you, sir.
Thank you, sir. Thank you for this very informative message on role of nanotechnology in horticulture crops. Now we have completed the lead speakers and also the guest note of this session, that is theme 2, smart breeding in horticulture crops. Now it's time to felicitate our lead speakers. So for this, I request Dr. Shantapati, Special Officer Seeds UHS Bagalkot, to felicitate Dr. K.V. Ravishankar, sir, Principal Scientist, IHR, Bengaluru. I request Shantapati Special Officer seats to felicitate our today's lead speaker, Dr. K.V. Ravishankar Sir, Principal Scientist, IHR, Bengaluru. Now I also request Dr. Lohitaswa HC, Principal Scientist, US, Bengaluru, for his lead speaker on the topic pre-breeding strategies for improvement of horticultural crops. I request Dr. Shantapati, Satish D. Sir, and also Vijay Kumar Sir to felicitate Dr. Lohitaswa HC, Principal Scientist, US, Bengaluru, for today's lead speaker on the topic pre-breeding strategies for improvement of horticultural crops. Thank you, sir. Now, I also request our today's another lead speaker, that is Dr. Daroji Rao, Associate Dean, YSR University, Andhra Pradesh, for his lead speaker or the guest lecture on nanotechnology application in horticulture crops. I request the organizers of this conference and also Shantapati sir to felicitate Dr. Daroji Rao sir, Associate Dean, YSR University, Andhra Pradesh. Thank you, sir. Thank you, sir.
So now welcome back to Smart Breeding. Now offline presentation will start now. There is the okay. Yeah, we have the time constraint. I think all the speakers should restrict to 5 minutes, 4+1. Please don't elaborate on methodology. Come for the results and the conclusion. Okay. So first speaker is Vidya. She's going to speak on evaluation of sponge gourd, genotypes for summer season, performance and adaptability.
A very good evening, everyone. I'm Vidya, a PhD scholar from University of Horticulture Sciences, Bagalkot. Today, I'll be presenting about minimize market by screen mini. Today, I'll be presenting about evaluation of sponge gourd genotypes for summer season performance and adaptability. And sponge gourd is a monoecious cucurbit, which is botanically called Lupha cylindrica, having a diploid chromosome number of 26. As we all know, it belongs to the family Cucurbitaceae, and which is originated from tropical Asia. And it is also commonly known as Ghiathori or towel gourd, smooth gourd, and Chinese okra. The crop is mainly cultivated in some parts of UP and Punjab. And also in Nepal, every backyard of the home will be having this sponge gourd crop. In spite of its less economic significance, the crop has got a very rich nutritional significance where it is nutritionally richer than ridge gourd, and its seed oil is commercially extracted. And one of the major byproduct of the crop is sponge extraction. And the sponge gourd is also recommended for the treatment against arthritis, leprosy, and it also has got anti-HIV property. And coming to the actual concept, sponge gourd is basically a short day crop which comes up well in the Rabi season and also in the Kharif season for some extent, but it produces very less flowers and fruits during the summer season. And when coming to the female flower production, it is inversely related to the increase in the temperature, which in turn affect on the drastic reduction in the yield. So with this background, the study was conducted to identify the sponge gourd genotypes suitable for summer season cultivation in order to identify the genotype suitable for the longer photoperiodic conditions, in order to identify the genotype which is tolerant to high temperature conditions, which is a way towards a breeding for abiotic stress tolerance. And coming to the details of the experiment, total 20 genotypes which were performing in the Rabi and Kharif seasons were evaluated in the RCBD design during summer season of 2024. And these are the list of genotypes that were used in the study. And during the tenure of experiment, a maximum of 42 degrees Celsius was observed, and the minimum of 18.32 degrees Celsius was observed. And this table 1 shows the mean performance, where among all the genotypes, Bagalkot local 3 was exhibiting the highest vine length. And coming to the earliness parameters, the genotype SG3 was recorded the minimum number of days to first flower and the first fruit harvest. When coming to the yield parameters, among all the genotypes, the genotype SG4 recorded minimum sex ratio, that is 17.54, and the same genotype was found to have the highest number of fruits per vine and fruit yield per vine and fruit yield per plot of 17 kg per plot. And this Pusa Sneha was the earliest variety in the study, and SG4 was the high yielding variety. With this, Pusa Sneha was found to be having earliest variety exhibiting the minimum number of days to female flowers and first fruit harvest, which can be used for the genotype to escape from the high temperature conditions. And another genotype, that is SG4, which is high yielding. So it is like exhibited as high temperature tolerant, and it can be recommended for summer season cultivation. And also it can be further used in breeding program for the development of high temperature tolerant varieties. Thank you, everyone.
Thanks for completing within the time allotted. We have a question. We have time for one question. Anybody would like to ask? Okay, no one. Thank you, Vidya. Thanks. Our next speaker is Ms. Mahalakshmi M. She is going to speak on evaluation of snake gourd genotypes for qualitative and quantitative traits.
A very pleasant good evening to one and all. I'm Mahalakshmi M, PhD scholar from COH Bagalkot, here to present the title on Evaluation of Snake Gourd Genotypes for Quality and Quantitative Traits. Snake gourd, scientifically called as Trichoxanthus sanguina, belongs to the Cucurbitaceae family, with holding the diploid chromosome number of 22, and it is an Indian originated crop known for its diverse morphological traits, and it is distributed throughout the world in tropical and subtropical regions. In India, it is highly famous in Tamil Nadu, Kerala, and some of the northeastern states. The crop holds several benefits. It is one among the cucurbitaceous crops which has the potentiality to yield more yield per unit area. It is rich in nutrients, bioactive components, and it is used in the traditional medicines for ages as an detoxifier, diuretic, and purgative. Besides all these benefits, this crop faces some productivity issues, as there is a less number of released varieties has been developed, and also the less researchers has been forced on this crop. With this, an experiment was conducted to study the qualitative and quantitative traits. The randomized block design with 26 diverse genotypes replicated twice with a spacing of 2 into 2 meter in the season Kharif 2023. These are the list of genotypes which are used in the study, collected from all over India, from Tamil Nadu, NBPGR lines, Kerala and Coimbatore. These are the morphologically diverse characters has been identified in the crops for specifically for the fruit shape and fruit color, as well as the fruit length, like elliptical, oblong shaped fruits, elongate elliptical fruits, and the color varies from cream, dark green and light green color fruits. These were the pictures exhibiting the different in shape, color, as well as in the stripes of the fruit. So the per se performance of the snake guard genotype shows that the genotype G3 produces early seedlings at 4 days. The genotype T1 produces its male flower in the 3rd node itself, and in the 11th node, T3 produces female flowers at the 11th node. Maximum number of female flowers were identified in G24, and also the G24 is recorded as early genotype for days to male flower, female flower, days to first fruit set, days to first harvest. And the genotype G18 has a potentiality to produce the fruits for 143 days. And the fruit length, which is ideal and as per the consumer preferences, several genotypes noticed from 25 to 30 centimeter, G2, G3, G4, G10, G23, 24, 26, T3 as well. And for fruit width, the G7 has been recorded highest fruit width. And an average fruit weight of 300 to 350 centimeter, 300 to 350 grams were noticed in G3, 8, 9, 11, 18, 20, 21, 30, T1 and T3. The maximum number of fruits were identified in genotype G24, and the maximum yield per plant and per hectare was recorded in the G7, and maximum number of seeds per fruit was recorded in the G6 as well. Moving on to the quality parameters, the most trimmed fruit is recorded in the G6, the maximum TSS of 5.5 in G10, maximum total phenols of 103.45 milligram in G6, and 0.4% of magnesium in G9, 1% of calcium in G25, and 0.34% of phosphorus in T3. These were the high-yielding genotypes, which is white and green in color, with average yield of 13.61 kg per plant and 9.95 kg per plant. With this, it is concluded that the genotypes G7, 24, G2, and T3 were identified as the best performers for the yield and other yield-related traits. These genotypes can be further evaluated for the crop improvement program and can be released as a varieties, or it can be selected as a parents for heterosis programs. Thank you.
Any questions? If there are no questions, thank you, Mahalakshmi.
Thanks.
So now I call next speaker, Ms. Rajashri. Heterosis study. She is going to speak on heterosis studies in brinjal.
Good afternoon to everyone. I am Rajashri, PhD scholar from the University of Horticultural Sciences, Bagalkot. I will be presenting on the research topic of heterosis studies in brinjal. As we all know that brinjal is one of the most important crop worldwide. It is having a botanical name called Solanum melanogaena and belongs to the family Solanaceae. It is believed to be originated in India. It is having another name called eggplant and aubergine. Brinjal is one of the nutritious rich vegetable. It's consumed in various ways. It's also beneficial for diabetes and for liver health. Apart from this, it provides the dietary fiber, vitamins and essential nutrients. Due to its diverse medicinal effects, it acts as a variable component for the human diet and natural healing properties. Consumer preference for brinjal may vary among the regions with respect to fruit size, shape, color, and spines, which influences the market demand. Given the growing population, the high-yielding hybrids with a regional taste is beneficial. Heterosis is the easiest method to develop the hybrids. Hence, heterosis signifies the increased or decreased vigor of the F1 hybrids over the parents. The expression of heterosis may due to factors such as heterozygosity, allelic interactions such as dominance, over dominance. The heterosis can be estimated in 3 ways: over mid-parent, better parent, and over commercial check. The objective of the study is to study the heterosis for growth, yield, and quality traits. The experiment was conducted at Kitturani Channamma College of Horticulture, Arbavi during the Kharif season 2023-24. The experiment followed a RBD design with a line into tester mating design. The experiment includes 12 lines and 3 testers with inclusion of 2 checks with the 2 replications. The spacing 60 by 45 centimeter is followed for proper growth and maintenance. This table depicts the list of genotypes used in the study and their source of collection. These are the, this figure indicates the list of genotypes used in the experiment. The experimental results revealed that the crosses KRCCH11 into Bangalore local exhibited the highest positive significant heterosis over better parent, best parent, commercial check 1 and commercial check 2 for plant height, number of secondary branches per plant, and plant spread at east, west, and direction. And the crosses KRCCH12 into BL exhibited the highest positive significant heterosis for the trait plant spread at north-south direction. And the cross KRCCH11 into Gangavati local exhibited the highest positive significant heterosis for the trait stem girth. This is due to its effective vegetative growth and effective utilization of the resources. For earliness parameters, the cross KRCCH10 into Arbavi local, KRCCH11 into Bangalore local exhibited the negative significant heterosis for the dates to first flowering, and the crosses KRCCH11 into GL exhibited the negative significant heterosis for the days to 50% flowering and days to first fruit among the parents. And for the yield and its attributing traits like fruit diameter, the cross KRCCH10 into GL exhibited the highest positive significant heterosis, and the cross KRCCH11 into GL exhibited the highest positive significant heterosis for the number of fruits per plant, average fruit weight, and fruit yield per plant. This is due to genetic diversity, dominance effect, and hybrid vigor among the parental lines. Additionally, for the qualitative parameters, the KRCCH11 into AL and the KRCCH3 into BL exhibited the highest positive significant heterosis. These all hybrids hold the promise for high yielding and quality parameters. With this, the pairings KRCCH12, 11, and 10 among the lines, and the Gangavati local and Bangalore local testers are good combiners for all the characters which can be used in a future breeding program. And the crosses KRCCH11 into GL, 11 into BL, 12 into GL, 12 into BL, 10 into GL were superior hybrids. These hybrids, 11 into GL and 3 into BL, expressed superior quality parameters. Thank you.
Thank you. So our next speaker is Dr. Mahantesh B.N. Nayak. So he is going to talk on discovery of candidate genes and pathways associated with essential oil production in Indian coriander cultivars using transcriptomics approach. Okay, ಇವ್ರಿಗೆ ಹೇಳ್ಬಿಡಿ ಅವ್ರಿಗೆ. Mayur, okay, there is a small change now. ಎಲ್ಲಿದೆ ಇಲ್ಲಿ ಮಯೂರ್ ಪಾಟೀಲ್?
Most respected Chair Madhu Session, Co-Chair, and respected delegates, I am Dr. M.D. Patil, Scientist Plant Planning from All India Coordinated Research Project on Mular, University of Agricultural Sciences, Dharwad. I am standing before you to present our work on genetic profiling and molecular diversity and population structure analysis in pea germplasm. Once we hear the pea, you know that this history dates back to 1866, the experiment of Gregor John Mendel. And we know that this is one of the very important crop of present era also because it is very nutritious and it is a main source of vegetable protein, and that especially for the large population of vegetarian populations of countries like India. And it also contains a lot of other nutrients. And nowadays, it is also known to be a low glycemic index food, which is very important for the countries like India, which is the capital of diabetes. Genetic profiling of the traits is very important to isolate superior cultivars and also trait-specific donors. And once we use the modern tools, we can have a more informed and precise decision-making in breeding. And also, it also helps to isolate a hybridization, for hybridization, to create genetic variability through hybridization, and also to isolate the better parents for hybridization. With this objectives, what we use is we used 160 genotypes sourced from different institutions like NBPGR, New Delhi, IIPR, Kanpur, and also we collected some local cultivars in and around Dharwad of pea, which are varying in so many different traits, and this were evaluated in augmented design, and we could record the observations on 28 traits, both including qualitative and quantitative traits, and we used R Studio for data analysis and also structure software to analysis of population structure. And to identify the which important trait, we use the guidelines of PPFRA, that's guidelines, and the experiment is conducted in Agriculture College, Vijaypur. And these are the wonderful variations we got for different traits like anthocyanin pigment, leaflets, flower standard petal color, leaf axil color, foliage color, rabbit ear stipules, number of pods per axil, pod curvature, pod distal part shape, and then pod color, and so many different seed cotyledon colors. And we know that wrinkled and smooth is one of the important traits of Mandel experiment, tester modeling, seed shape, and also hilum color. And once we see the frequency, we had, total 160 genotypes when we grouped, for different traits. We had a wonderful distribution of the genotypes for various traits. This graph shows that distribution of those all qualitative traits among this 160 genotypes. And then when we did a cluster dendrogram, we got a wonderful diversity panel of all 160 P genotypes we got. And I've clearly marked 2 genotypes about which I'm going to tell in further slides. So, and when we did cluster analysis, there were 8 clusters were formed, and cluster 1 was highest number of genotypes. And these 2 marked genotypes were also in different clusters, what we saw. And the contribution when we saw to the diversity, the flowering time was the factor which was contributed highest to the total variability present in the this germplasm lines. And then when we did cluster grouping, and cluster 3 had wonderful genotypes with the highest, lowest score and with ranking first. So this is a cluster where we can get the wonderful genotypes for different traits. And then we also did the scree plot explaining to know how many principal components we have to isolate. So we did the rotated matrix to know the correlated variables in the principal component analysis, and also the direction of direction also. And then we did molecular diversity analysis using Chiaxal advanced automatic capillary electrophoresis, and this is one of the representative picture of AA446 marker. And then we had a very good peak value of more than 0.5 explaining this one. And then we got 25 clusters for molecular diversity analysis. And the population structure analysis, we got 4 population subgroups. Maximum allele frequency divergence between 1 and 3, and minimum was 3 and 4. And this is this one showing. And so this is a wonderful scheme of this work. We got 3 genotypes which are high yielding, high protein, and also early maturing. And this we have taken to breeding application, where we have crossed those 2 genotypes which I have marked. We were extremely, different for so many different traits, and we have now isolated, very good transcriptive segregants in the F2 population, and we are taking them forward and stabilizing to isolate the best genotype out of that, which is early flowering and also high yielding, which is needed, which is a need of the work. Thank you, one and all, for this opportunity for presentation. Thank you.
Thank you. Next speaker is Dr. Mahantesh Naik. Discovery of candidate genes and pathways associated with essential oil production in Indian coriander.
Good afternoon to all, respected chair and co-chair and rapporteurs and dear participants. So I'll be presenting about, in fact, our focus of the work was to identify the qualitative trait. So in that, I'll be presenting the genes which we have identified mainly for essential oil responsive genes. So coriander, why coriander? Because of course, this is a global crop. It's a dual crop known for spices and vegetable. Looking into its important, we know that it will going to give us a wonderful essential oils, which is important with respect to the medicinal perspective in terms of looking into its anti-diabetic, anti-hypersensitive, apodresic, and again, of course, it has many other medicinal importance. Having its all essential oil, which are going to help for the medicinal perspective, what we thought of is like looking into the genes which are responsibility to contribute in terms of the qualitative traits. And that's how we started looking into the identifying the genes with respect to, because many of the papers it just talk about the either leaf or the seeds, but there is no such studies where completely talks about seed and leaf. Even if it just talk about in one genotype, but nowhere it talks about the comparing with the different genotypes. In that section, we have selected 3 cultivars, which are national cultivars. So we thought of that, yes, sequencing and transcriptome could be the one of the best approach to really discover the genes which are responsible for essential oil synthesis. So what we did is we choose the 3 cultivars which are very popularly grown. That is AGCR1, that is Azmir green coriander, CO2 and Pantharidma. So this talks about the phenotype of this particular cultivars. Then what we did is we took the tissues here, leaves and seed. The mature seeds we took, and seed is leaf we took for 3-month-old leaf we selected. And after that, the general flow, what we generally do is once we extract the RNA, we prepare the cDNA library. After preparing the cDNA library, yes, we choose the Illumina platform, which is Next 6 500. In fact, within the Next 6 platform, they have different plats, like, but we chose the 500 because that yields the best library. Having into that, what we did is we took all the raw reads of the leaf and the seed, and we utilized the thymotic tool, wherein again we obtained the high quality reads after trimming the adapters. Obtaining into that, then what we did is we did the de novo pooled transcriptome, because we wanted to really look into how exactly these genes are there in the seeds and the leaf. So we pulled it, and after that we identified the unigenes, we predicted the unigenes, and then we validated. And after that, ultimately we wanted to know that, so how many of the coding sequence or ESTs are there into that. Then in that, once we predicted into that, we validated. And of course, ultimately, in order to identify their function, in order to identify the level of expression, in order to identify these genes into the associated pathway, what we have done is like we looked into the functional annotation. Of course, since none of the genomes so far, we don't have the 100% annotation information, still we are really finding to obtain the annotation for the whole genome, the plant-like model system, Rhodopsis thaliana, still we are not able to completely do the annotation. In such case, we have known functions and unknown functions, and we got the differential expression genes in leaf and tissues of the different genotypes, and ultimately we could able to identify the pathways which are associated to synthesize these essential oils by using Cas. So this talks about the overall outcome of the transcriptomics data, where if you look into if it is single tissue, single sample, that's fine. But we had an excitement clearly looking into the different tissues, because it was a challenge, because handling into the big data, it's not simple. We really need to look into the number of genes, and we have to really, our focus was to, of course, we mainly focus for the qualitative traits, but one of the aspects is to identify the essential oils responsive genes. So if you see here, these are the different tissues, and these are the different reads and bases obtained, and data is available in the NCBI, it is submitted. And so looking into our objective of research was to identify the essential oil synthesis genes. So here we could identify the metabolism of terpenoids and polyketones. Here we could able to identify the differential expression genes which are associated with the biosynthesis of essential oils. Yeah. So here it comes. Ultimately, what we have seen is in terms of looking into the leaves and the seed. So the very important gene which is present in the coriander is especially in terms of its aroma, that is linolol. Linolol is the one which is high, because if you look into its biochemical composition, nearly 70 to 80% of the linolol is present in the coriander. But interestingly, what we have discovered is we also identify the genes which are there in these leaves and seeds. Apart from the terpenoid synthesis, biosynthesis genes, we could able to identify other genes also which are equally important in terms of their contributing for aroma. Now, if you see the number of genes which are there in the leaves, again, one interesting outcome what we got from here is we have noticed that there are common gene and there are unique genes. So now the next interesting is if you look into the especially myrisine synthase, it is only found in Pantharidma seed, but it is not found in any of the tissues of the cultivar. So that could be the another novel molecule where we can take further and look into its, of course, pathway we know, but further look into its composition and further it's really utilized into the medical industry, agriculture industry or horticulture industry. So just to conclude, we have got a very promising lead molecules and this could be further effectively utilized for the crop improvement program. So with this, I conclude my talk. And since it is a collaboration research with ICR NRC Seed Spices, I would like to thank Dr. Sharda Chaudhary for their constant support and collaborative work. And finally, I would like to thank UHS-Bagalkot for the infrastructure facility. So thank you all. Thank you for your patience hearing.
Any questions? Okay, if there are no questions, thank you, Dr. Naik.
Thank you, sir.
So I invite Sinchana Jain. She's going to speak on strategies for speed breeding of mango through morphological and color-based pre-selection markers.
Good evening, respected dignitaries present over the stage, the audience, and my dear friends. Myself, Sinchana Jain from ICR IHR. India is recognized as mango in many foreign countries. So mango is the national fruit of India, and it is regarded as the king of fruits owing to its luscious taste and delicious flavor. And India is the largest producer of mango in the world, and there are like Uttar Pradesh is the leading state in the mango production. There are more than 1,000 varieties do exist in India with respect to mango, and only few are competing in the international market. The major thing that is contributing for the international trade is its attractive peel color. So this color is coming from two pigments, that is anthocyanins and carotenoids. These pigments not only contribute for the visual appearance, they also have some nutraceutical properties. So coming to the breeding of the mango varieties for color, it takes more higher year, as one of the lead speakers says, it takes 28 years to develop a new variety in case of fruit crops. It is true in case of mango as well. So some of the, with this background, we have formulated our research in such a way that we have taken Amrapali as a female parent. The major thing is it is having a good quality fruits. The major lacuna in Amrapali is its yellowish green peel color. So we try to transfer the redness from male parent Vanraj to the Amrapali. We have generated around 208 mango progenies, and in that 168 progenies which bears both the seasons were considered for further evaluation. And we tried to identify few morphological markers so that we can reduce the conventional breeding years. Since this work is based on color, we have evaluated the color of emerging leaf, peel, as well as pulp from RHS color chart and Minolta colorimeter. And the pigments were analyzed using spectrophotometer, like chlorophyll, carotenoids, and anthocyanins were analyzed. These are the variations that we have observed in peel, pulp, and emerging leaf. So this RHS color chart, actually, it didn't differentiate both male and female parent. It showed a grade orange color group, but the code was quite different. So this RHS color chart didn't give the accurate results in terms of emerging leaf color. Whereas in case of peel and pulp, we have identified variations with respect to both the parents and progenies. So for the accurate measuring, we have gone to colorimetric values, where we have evaluated Hunter color scales L, A, B, C, and H values. We have identified much variations with respect to emerging leaf color in all the Hunter color parameters. We have also evaluated the biochemical parameters like chlorophylls, carotenoids, and anthocyanins. Amrapali recorded highest chlorophyll, and Vanraj being a male parent, it has recorded highest anthocyanins of 29.74 milligrams per 100 gram. And we haven't observed any transgressive segregants in the progenies. So in order to check the morphological markers, we have studied the correlation of emerging leaf color and colorimetric scales of peel. We have identified this Hunter A value showed a negative, marginal, significant negative correlation with all the other parameters like L, C, and H. And then Hue showed a positive correlation with peel L. Moving on to the correlation between the leaf and pigments, we have identified this leaf A parameter is showing a significant marginal negative correlation with chlorophylls and carotenoids. And Hue value showed a positive correlation with carotenoids. Moving on to the correlation between the color coordinates of peel and pigments, we have identified this color parameter A. It is having a significant positive correlation with anthocyanins and negative correlation with chlorophyll. This is vice versa in case of Hue angle. Unfortunately, we haven't observed any correlation between the color coordinates of pulp and pigments. So based on these correlation studies, we have generated the regression models, and with this regression models, we can easily medium to good predictions based on these color parameters can be done. So this serves as a non-destructive way. In general, extraction of pigments it's a laborious process. So many chemicals were involved and it takes 1 to 2 days. So with this method, we can easily identify conclude the pigment concentration non-destructively. And the major drawback, major identification of our research is these color parameters of leaf can act as a morphological marker so that can be incorporated into the breeding program and we can fasten the breeding program. Thank you.
Thank you. Any questions? Okay, if there are no questions, thank you, Sinchana. So now I call upon Dr. Chaitra Kulakarni. She is going to talk on transcriptome dynamics between Western and Eastern carrot gene pools and identification of candidate genes for vascular cambium root patterning.
Very pleasant good evening, everyone. I'm here to present my PhD research work on transcriptome dynamics of Asiatic and Western carrot gene pools to decode candidate genes for vascular cambium patterning. Carrot is regarded as the second most important vegetable considering its nutritional and economic value with many health beneficial factors. Globally, the cultivated carrots is divided into two major gene pools. One is Asiatic gene pool, another one is Western gene pool. The Asiatic carrots are tropical adaptable with very least beta-carotenoid content. Western carrots are adaptable with highest beta-carotenoid content. The tropical carrots are considered to be annuals, but that they don't require any vernalization treatment to switch from vegetative phase to reproductive phase, whereas biennials are Western types that require typical vernalization treatment to switch from vegetative phase to the reproductive phase. On the other hand, you can see the matured storage root of the carrot, wherein there are three layers. The outermost layer is phloem, the innermost layer is xylem. The layer that separates the phloem and xylem is called as vascular cambium. Vascular cambium acts as a typical key regulator to determine the xylem-phloem patterning in the carrot. Both vascular cambium patterning and vernalization are two domestication traits in the carrot. That is the reason why, to identify the vascular cambium responsive genes, we performed the transcriptome profiling between Asiatic carrots as well as Western carrots, then identified the differentially expressed genes and candidate genes responsible for the vascular cambium. Then the functionally annotated genes were validated using the qPCR. In this figure, you can observe the typical differences between the Western carrots and the Asiatic carrots. The Asiatic carrots at the center, they do have the typical xylem patterning. The Western carrots do not have the xylem patterning. They are having uniform phloem patterning. These two materials, these two are the materials selected for the present research work. They were screened by using that 146 germplasm accessions. By screening 146 germplasm accessions, these two cultivars were utilized for the present research work. Then to identify the typical differences between the xylem phloem patterning, histological study was also carried out between these two cultivars, wherein in the tropical cultivar, that is Asiatic cultivar, you can observe the three different layers, xylem, phloem, and the vascular cambium. The xylem was typically, protoxylem at the center was typically with the xylem loaded cells only. But in the phloem, you can observe the three layers fully loaded with the phloematic cells only. So these two cultivars were subjected for transcriptome profiling to identify the vascular cambium responsive genes. Reference-based assembly was constructed, differentially expressed genes were annotated, and then the gene regulatory network was constructed. The mapping rate, reference-based assembly mapping rate was 73.87%. A total of 3544 differentially expressed genes were identified. In the volcano plot, you can see the significantly expressed genes, red color dots for the Asiatic cultivars and blue color dots for the Western cultivars. The annotation percentage was 43.49%. So the annotated genes were used for further construction of the gene regulatory network by comparing with the homologous genes with the Arabidopsis thaliana crop for the vascular cambium responsive genes. So further, the gene ontology terms were identified for biological process, cellular component, molecular function, and KEGG pathway between these two cultivars. All the four pathways identified confirmed that the Western carrots were enriched with geoterms for responsible for photosynthesis, carbon metabolism, and secondary metabolism factors. And the Asiatic cultivars were having stress-responsive genes like MAPK signaling, salicylic acid, brasinosteroid, and many other components. Further, the gene regulatory network analysis was carried out using the Arabidopsis thaliana responsive genes in comparison to our annotated genes. Then in that also, gene regulatory network also, at the center you can observe, represented by yellow dots, all the genes were representative of photosynthetic response as well as carbon fixation responsive genes. Importantly, three vascular cambium responsive genes were identified, TL29, APE2, and NPQ4. They are known to inhibit the xylem differentiation in the Western carrot. On the opposite, in the other hand, in the Asiatic cultivar, we could able to identify the gene regulatory network comprised of many of the stress-responsive genes only. But on the other hand, interestingly, in the Asiatic cultivar, the vascular cambium responsive genes like NAG03, XTH7, XTH9, and other two were involved in development of the xylem. You please understand, they were involved in the development of the xylem. So this is the result showing the congruence between the RNA-seq results as well as the qPCR results. So this is the globally, this research is of first of kind in our lab. Recently, we published these two articles for vascular tissue patterning and carotenoid content and flowering pathway deciphering using this assembly, what we generated. This is the first of its kind globally. With this, I would like to conclude saying that whatever the vascular cambium responsive genes we have identified in Asiatic cultivar, that would be further genome edited and uniform phloem patterning can be brought, and candidate gene variants can be further used for genomic assisted breeding and QTL mapping. Thank you.
Hello.
So there's one girl, she has a question, sir.
Yeah. Oh, no. Wait.
Please take it.
You can talk.
Yes.
Okay, my question is Asiatic carrots and Western carrots. You were specified about Asiatic carrots contains less beta carotenes compared to this Western
Yes, madam.
carrots. So for a human consumption, which you refer, one question. The another question, which is the commercially viable for the farmers? So whether Asiatic or this Western.
Madam, okay, thanks for your question. Actually, both the cultivars has their own advantage to the consumers. I would like to say that beta carotenoid content, if consumer want to have enriched with beta carotenoid content, definitely Western carrots are only having the highest beta carotenoid content compared to Eastern carrots. And the cost price is also, farmer will get more from the Western carrots only. Eastern carrots price is less, and that cost benefit ratio will also be less for the farmer to the Eastern carrots. carrots. But eastern carrots, not only the or light orange color, there are different colors of eastern carrots like black color, purple color. They do have their own advantages of health benefits. Black carrots are having anti-aging properties, having cardioprotective activities, and the black color powder is also exported to many countries from India. So both the cultivars have their own advantages. East Asiatic cultivars are having many stress responsive genes. Very importantly, Asiatic cultivars are soft rot resistant, whereas Western cultivars are highly susceptible to soft rot. Actually, after nearly 80 to 90 days, the cultivar in the field will start getting rotted if it is not correctly promptly harvested. So, but many stress responsive genes are there in Asiatic. For breeding purpose, Asiatic cultivar gene pool is a very excellent gene pool for the breeding cultivar to any breeder of the carrot.
For the farmer's sake for the farmer point of view, I'm just asking the question because it's commercially available.
Farmer point of view, definitely. For income oriented farmer, definitely Western carrots are only the best. But the Western carrot seed rate is very high. The F1 hybrid seed rate of Western carrot is nearly about 35 to 40,000. F1 hybrid. OPVs are only 1 kg of OPV only costs about 15,000 to like that.
Even I feel that compared to Asiatic carrot, Western carrots germination percentage is very low, like 40 to 45%.
No. If you at viable seeds, if you promptly at right stage, if you seed in the soil, definitely uniform seedlings will come. There is no problem of germination, but there is a problem for the stress responses, biotic as well as abiotic. For that, Eastern carrots are excellent gene pools. They do not suffer from such stresses.
Thank you.
Yes, madam. Thank you.
Thank you, Dr. Chaitra. So,
thank you, sir.
Our next speaker is Dr. Raghavendra Gunnaya. He is going to speak on CRISPR-mediated editing of bacterial blight susceptibility genes, a sustainable approach to enhance pomegranate resistance to Xanthomonas citrui punicae.
Okay, good evening, everyone. Respected dignitaries and the dais and the delegates. It's my pleasure to present my present research on CRISPR-mediated gene editing of bacterial blight susceptibility genes. So coming to the pomegranate, so it's commonly called as a dollar earning crop for the farmers for multiple reasons. It is nutritionally as well as medicinally highly valued. It can sustain very well in marginal soils. Hence, farmers are growing very well, are adopting this crop widely, even in non-conventional areas. So India is the largest producer, cultivating about 2.6 lakh hectares and producing 23.15 lakh hectares. So it is commercially cultivated in Maharashtra, Karnataka, Andhra Pradesh, Gujarat, and Rajasthan. So it's called as a dollar earning crop because India has exported around 62,000 tons of pomegranates valued at around 58.36 million US dollars. It is expected that by 2030, its market is going to be around 367 million US dollars. However, this crop is severely affected by disease called as bacterial blight. So I think it is the first most disease among all the biotic stresses, which accounts for almost 45% of the biotic stress diseases. It affects all parts of the plants except the flowers and the roots. And the severity is up to 70 to 80% yield loss is so seen in the farmer's field. So the severity is documented in various newspapers. So the farmers are cutting down the orchards or even they are burning. So that tells about the severity of the disease, which is affecting the pomegranate. So it is caused by a gram-negative bacteria called as Xanthomonas citrui punicae. So currently it has been managed by field sanitization chemicals such as copper-based bactericides, antibiotics, as well as some of the biologicals. However, they are not efficient, and moreover, antibiotics like streptocycline are banned in India. To breed for the resistant, all the present-day cultivars are highly susceptible. So although we screened some wild cultivars from Himalaya, derived from Nanu and Daru, we found good tolerance. However, these are associated with a lot of non-desirable agronomic and yield quality traits. So in this scenario, so I'd like to present this as gene editing using CRISPR tools as an efficient tool, mainly because of high precision, and we can breed it. And since it is a perennial crop, gene editing will help us in breeding at a very short time. It is highly efficient. Unlike our genetically modified crops, it does not have any foreign genes or the element in it. And most importantly, Government of India has already approved commercial release or maybe field trials of SDN1 and SDN2 trials, site-directed nucleus 1 and 2 trials, which does not have any foreign material. So coming to the steps, major steps in gene editing, the first and foremost is the selecting a specific target gene which can confer the resistance. Second one is the designing the guide RNA, construction of Cas9 and guide RNA, construct delivery using different methods, in vitro regeneration, validating, and finally regulatory approvals. So towards our first step, so to identify the target genes, we started studying about the pathogenesis of the Xanthomonas bacteria, and we found that transcription activator like effectors, which are secreted by a type 3 secretion system, are most important factors in the causing the pathogens. So what that does is, so once the Xanthomonas secretes these tails into the cytoplasm, so with the help of nuclear localization signal, they enter into the nucleus and bind to certain genes. Mainly we call them as the susceptibility genes, like sweet genes, and activate. So these genes are mostly useful for deriving the nutrition, such as the sweet genes, which are the sugar transporter genes. So coming to the structure, important part is the repeat domain. So each repeat is made up of 34 amino acids, and each tail can contain around 17 to 23.5 tails. So these among these repetitive, the 12th and 13th amino acid, you can see HD is red labeled. The 12th and 13th amino acid very important. It's because they determine which nucleotide should it bind in the promoter region of the susceptible genes. So with this background, so we started identifying the susceptibility genes by sequencing the whole genome of Xanthomonas using a long read genome sequencing, because these are all highly repetitive elements. Illumina sequencing will not help. Hence, we went for PacBio sequencing. We predicted the tails. Then we identify the tail binding regions. Oh. I'm sorry. Where did it go? So we identified there are 6 tails in the Xanthomonas genome. Among them, 6 are functional. So then we identified the targets using different bioinformatics tools. We found like 95 to 195 tail targets are bound by each tail. Then among them, we selected 195 tail targets, which can be targeted by either of one of the tails. Then we did the differential expression analysis of these tails. So among the differential expression analysis, we found that age65 genes have the tail effector binding elements. Based on the differential expression analysis, we identified 5 candidate genes which are expressed at 3 days post-inoculation as well as 9 days post-inoculation. Further, we did a molecular docking to find the tail and the target binding. And we shortlisted 2 targets with very good significant RMSD scores. And we also tested them, validated them by targeted gene expression analysis using qPCR. And we found that only in the susceptible cultivar, these genes are expressed, whereas in the tolerant cultivar, they are not expressed both at 3 days as well as the 9 days post-inoculation. So now we constructed the single guide RNA. So further, we have also shortlisted, standardized the tissue culture generation protocol. So we would like to further transform these vectors into them and develop transformants and screen for them for the phenotypic and bacterial blood resistance as well as growth and yield. Thank you.
Thank you, Raghavendra. Abhishek is there? Okay. Okay. So now Mr. Abhishek is going to speak on study of transgressive segregants in the F4 population of okra for yield and its contributing traits.
A pleasant evening to everyone present here. I am Abhishek from the Department of BCI. I am here to present my research article on transgenic segregant studies in F4 population of the okra. Okra, which is scientifically known as a Abelmasca esculentus, belongs to the family Malvaceae with a chromosome number of 2n equal to 130, which origin is in the tropical Africa and which is having a high nutritional value. Transgenic segregant refer to an individual in a segregating population that exhibit a trait value beyond the range of its parents, either higher or lower. This is the image which showing the transgenic segregant. And the concept behind is because transgenic segregant have high recombination, as it has additive gene action, epistasis, over-dominance. They are environmentally influenced as genotype into environment interaction can be modified phenotypic outcomes. The breeding value, which refers to the individual's ability to pass desirable trait to the next generation. The transgenic segregants often have high breeding value, making them excellent candidates for developing superior crop variety through selection and hybridization. Evolutionary role. The transgenic segregant contribute to adaptive evolutions, which mechanism like crop domestication and diversification, natural selection, and enhancing genetic variation within population for future breeding programs. Key causes of the transgenic segregants are complementary gene action, epistasis, over-dominance, recombination of additive alleles. Before going to the F4 populations, we go back like hybrid evaluation, which has been done in 2018 to 2022. We selecting about the 54 lines. And after in evaluation of that 54 lines, we have developed the hybrids. Among the 36 hybrids, these are the top 5 performing hybrids, and these F1s were taken to the for the next F2 generation. In F2 generation, we taken the one of the cross that is OK2017010 cross with the Varsha Upar in an experimental design of unreplicated design. This is the top 10 transgenic segregant which were found in the F2 populations, in which the yield per plant which has been ranged from 280 grams to 303 grams when comparing to the both the parents. And these are the top 10 transgenic segregants selected from the cross. And after that, we gone for the F3 studies with an augmented block design of the same population. In this, the same crop, the same plant, what are the 10 plants which have been selected in the F2? There is a variation. They like the yield per plant has been gone up to the 622.40 gram when comparing to both the parents. And these are the transgenic segregant which were seen in the F3 population. And after that, the final which are done is F4 population, which has been done in the randomized complete block design at the College of Horticulture, Bengaluru, in the Department of BCI. And these are the top 5 promising transgenic segregant which were identified. These were the same as this, in which we have seen that the highest yield is about 1 kg 303 grams when about the 64% highest when comparing to both the parents. And after that, the combining of the different characters for fruit yield, in which we found that plant height, number of nodes, and number of fruits have the highest number of transgenic segregants. And after that, the plant height and number of fruits also exhibited the highest number of segregants. And in the total simultaneous transgenic segregant, in F2 we identified 108, in F3 93, in F4 84. By this, I want to conclude that the identified top lines were gone for the F5 generations for further evaluation. And these lines can be taken for further disease and insect resistance. Thank you.
Thank you, Abhishek. So next speaker is Revati Bannatti. She is going to speak on screening of moringa genotypes for nutrients and seed oil content.
Good evening, everyone. Myself, Revati Banati. I'm working as a young professional one under DUS Center for Drumstick Project at the Department of BCI. Today, I'm giving an oral presentation on the topic of screening of moringa genotypes for nutrients and its oil content. Coming to the introduction, the botanical name of moringa is drumstick is Moringa oleifera, which belongs to the family Moringaceae with a diploid chromosome number of 2 is equal to 28, and it is native to the sub-Himalayan region. Coming to the present scenario, Andhra Pradesh is the leading state, which constitute an area of 15,665 hectares, which is followed by Tamil Nadu and Karnataka. And total annual production in India is about 1.3 million tons of pods. Coming to the importance of that moringa tree, here, every part of the plant, moringa tree has a nutritive values. The leaf extract has anti-diabetic properties, and also leaf, flowers, and seeds, which are richer source of vitamin A, E, and C, vitamin A, and vitamin C, and vitamin E. And also the chemical composition of leaf, seed, and flowers are quercetin and camphorol, and also the root of the moringa constitute ursolic acid, which could possess hepatoprotective, anti-inflammatory, and anti-allergic properties. And come to the nutritional and therapeutic values of the moringa. Here, the niamycin, which is present in leaves, which has the antibiotic properties, and beta-cysterol, which is present in case of leaves, which could possess cholesterol reduction, and vitamin C and peroxidase, which is present in case of leaves and seeds, which could possess antioxidant and anti-cancerous properties. Coming to the concept here, recently the cultivation of the drumstick is become very much popular because of its anti-diabetic and nutritional values. And also the moringa seed oil is very much similar to that olive oil because of it is the richest source of palmitic acid and stearic acid. And the people suffering from that malnutrition and poverty, which found to be the moringa act as a superfood, and the proximate nutrients, which plays a very important role in case of fighting against that malnutrition. So the attempts has to be reveal the nutritional composition and its suitability as a vegetable in the available genotypes are important. With this, the two objectives are there. The first one is nutrient profiling in the drumstick collections, and second one is to genotypes. Coming to that, the program of research, there are 21 genotypes are moringa are collected from the different parts of India, and the experimental design was RCBD with a number of replication is 3. And some of the observations are recorded during the research. The first one is nutrient profiling. The first one is moisture percentage, and second one is ash, protein, crude fiber, fat, and total carbohydrates. And second objective is the moringa seed oil content, ml per 100 gram of the seeds. Coming to the procedure, the first one is the moisture content was measured by the hot air oven method, and second one is the ash content was determined by burning the samples in muffle furnace at 500 degrees Celsius. And the third one is the protein was estimated by the method. Similarly, the crude fiber was estimated was done by using the instrument Fibra Plus, and the total carbohydrates content was determined by 100 minus sum of that all the proximate nutrients. And second objective is that oil content. This oil content was estimated by using the oil pressing machine. Here 100 gram of these seeds can be used for each genotypes. Coming to the experimental results, this table one shows that per se performance of that the proximate nutrients in the different tissues. Here the highest leaf crude fiber was present in case of Carvar 1 genotype, which is about 13.77%, and the lowest was recorded in BG1 variety, which is about 4.93%, and highest fat content was observed in case of the seeds, which is about 26.27%, and the lowest fat content was recorded in case of Badami 1 genotype, which is about 5.87%. And coming to the moisture percent, the highest moisture percentage was recorded in case of leaf tissues, which is about 11.27%. And the carbohydrates, the highest carbohydrate was present in case of leaves, which is about 54.38%, which is followed by that pods and seeds. And this is a graphical representation of the tissue-wise comparison of the proximate nutrients among the moringa genotypes. And this table 2 shows that variation in the moringa oil content. Here the highest seed oil content was present in case of Badami 1 genotype, which is about 22.5 ml, and lowest was recorded in case of Mysore 2 variety, which is about 8.17 ml per 100 gram of the seeds. And this is a graphical representation of that moringa seed oil content among the genotypes. And finally, the conclusion, among all the 6 proximate components, carbohydrate followed by the protein found to be the present highest in concentration in all the tissues, and also the information about the nutritional status of the different parts of the plant that is helps to explore that various germplasms accessions in the breeding program for further development of that varieties, so that which is containing the specific minerals to combat against the major diseases. Thank you.
Any questions? What is the difference between fat and oil percent? You use it both fat and oil percent, no? Oil percent and fat percent. Do you have any? Oil percent and fat percent.
Sir, fat percent and oil percent. Sir, the oil is consisting of majorly the palmitic acid and stearic acid, but fat is content both the unsaturated and saturated fatty acids, sir.
Okay, thank you.
Thank you, sir.
So our next speaker is Dr. Vijay Kumar Rathod. He is going to speak on genetic inheritance and mapping of genes related to fruit quality traits in bitter gourd using microsatellite marker.
Dignitaries on the dais and dear friends. As you know, India is the most populous country in the world. At the same time, India is achieving or booming like a best economic and almost occupied the 6th position in the world with respect to economics. That makes the consumer preference from the traditional, where we are focusing much on the quantity, to change to the quality because of improvement of the economy. And when you talk about the quality purpose, especially food purpose, the fruit and vegetable plays a special position because of its nutritional value. And among the vegetables, cooker beets are majorly considered as important vegetable crops, with the many number of species are available, of which the bitter gourd is one of the important crops which occupies a special position because of its nutritional value. Especially bitter gourd is considered as a vegetable insulin. It is a anti-inflammatory, anti-HIV, anti-diabetics. So there are many medicinal value of the bitter gourd. So because of its value, from last 1 decade, the crop improvement has been tremendously utilized in the bitter gourd, especially for the quality purpose. And when you see the quality, especially in bitter gourd, we mostly focus on the quality, especially people are thinking in the different ways. The quality may be vary from the one person to another person's. And in bitter gourd, major quality factors, which includes the fruit tubercles, shape, color, size, etc. Among them, in this study, I have focused much on the quality, especially the fruit quality, where the fruit tubercles are morely focused, as well as the richness. And so in this experiment, we taken 3 contrasting parents, which having complete smooth surface with the continuous ridges as well as discontinuous ridges with the tubercles. So these are the characteristic of the genotypes or parent we used in the study. And we use the mortal F2 population for the mapping purpose as well as for inheritance study purpose. So in one population, almost 110 F2 population. In another study, almost 111 population we have used for the genetic inheritance and the mapping purpose. And these are the markers which is used for the screening purpose. Almost 534 markers we use, out of which hardly we got 10 and 11 respectively for the different characters. They are polymorphism. When it comes to results, so these are the parents we use for the study. One with a complete smooth surface and the ridges, and we have developed a population of F1 backcross population and the F2 population. And with respect to the inheritance study, here we must mostly focus on the tubercles characters. In F1, we got the tubercles indicate the trait is controlled under the dominant traits. With this phenotypic data, we also screen the population for the genotypic, genomic or marker level. By using these 2 data, we constructed a genomic map, linkage map by using the map maker. In spite, this is the first work where we much focus on the quality, especially for the tubercles. And the marker is MCSSR20, which is almost linked, not that much close, at least 12.5 centimorgan. And in another population where we mostly focus on the ridges, continuous ridges as well as discontinuous ridges, the same inheritance we observed. The discontinuous ridges are recessive in condition compared to the continuous ridges. And here also, the markers, almost 111 markers we are used for the screening purpose, and single marker analysis we use. We identify the marker JY004, which is linked to 19.6 centimorgan. And the same marker, especially because of the marker second, JY, which is almost far away from the linked marker. So that's why we skip that one. And only the earlier marker, which is for tubercles, which is revalidated on the different genotypes used. And where we are clearly seen there is a difference with respect to that marker for the tubercles as well as non-tubercles segments. So with this study, we conclude that this is the first work we observed. We reported, especially in case of bitter gourd. In spite of bitter gourd have been sequenced, but we don't have the complete genome sequence. It is a drop sequence available. So this study will be helpful for the future work. Though the marker distance was a little bit far away, the few more markers we can screen and we can identify the closely linked gene to that particular marker. Further investigation need be to undertaken to especially to identify the marker almost closely linked to that particular genes. Thank you.
Thank you. Any questions? Okay, thank you, Rathod. Now I call upon Dr. Deepak Kore. He's going to talk on morphological and molecular characterization of in vivo mutants in banana cultivar Elaki Bade.
A very good evening to one and all, respected chairman, co-chairman, and all the delegates of this conference. My name is Deepak Kore. I'm PhD scholar from Department of Fruit Science, Bangalore. Today I'm worked on the research on morphological and molecular characterization of in vitro mutants in banana cultivar Elaki Bade. So as we know that basic introduction of banana as it is, scientific name is Musa species. As there are only, we can see there are two different species like Musa acuminata and Musa bulbiciana. And we can see the origin is Southeast Asia. So coming to the main research of this, totally 96 mutants were selected for the early screening. Among them, 76 were treated with physical mutagens and 20 chemical mutants were used as a chemical mutagens. So among them, based on the morphological characterization, only 50 best mutants were selected and screened for molecular characterization. So these were the treatments like from M1 to M19, they were the chemical mutagens, and from M20 to M96, they were the physical mutagens such like gamma irradiation of different dosages. So the objective is to molecular characterization based on the phenotypic variation. So we can see the UBC series primers as they shows a polymorphism and we can see the diversity. So we can see the totally 81 loci were present among the 15 markers. Among 15, only 13 showed the polymorphism. So we can see the marker UBC-866, it has showed around 50% of the polymorphism and followed by UBC-809, that is around 47%. So coming to the characterization from the physical mutagens, we can see the marker UBC-856 to 900, it has showed the difference in the allele frequency, band frequency, heterozygosity, everything. So coming to the chemical mutagens, we can see the UBC series 864 had showed the highest band frequency and allelic frequency. So coming to the band patterns, we can see that in chemical mutagens, the mean heterozygosity that was around 0.024. And when coming to the physical mutagen, it was around 0.013% of heterozygosity was observed. So these were the bands which were observed of the different markers. So UBC 809, UBC 840, we can see the in the ladder genotype number 4, 5, it has showing the difference in all the markers. We can see the from UBC 809, 840, 856, and 864. Similarly, in the primer UBC 866 and 846. So the other further comparison, we can see the principal coordinate analysis. We can see that majority of it was divided into 3 clusters. Nearly first cluster comprises around 43%, second cluster around 25%, and third cluster around 70%. We can see that the mutant M4 and M5, it was the most diversity among the chemical mutagen treated. So coming to the physical mutagen, it was also categorized into 3 clusters. So here also similarly, mutant number 21, 22, 20, they showed the highest variation compared to the mutant plant. So coming to the dendrogram, we can see that it was divided into 2 clusters. The cluster 1 mainly comprises of chemical mutagen. Cluster 2 comprises about physical mutagen. Among the cluster 1, they were divided into 2 subclusters. Among them, we can see the chemical mutagen M38 and M32 show the diversity. When compared to the cluster 2, the physical mutagens such as mutant number 4, mutant number 3, and mutant number 8 were the present. So this shows the diversity among the population, and so we can conclude that mutant number 4 and mutant number 5 had the highest diversity compared to the all remaining pattern. So I would like to conclude saying that mutant number 4 and 5, that is the chemical mutagen, having the highest frequency, that is the highest diversity, and compared to the physical mutagen, that is EMS. So these were the plants obtained at different dosages. This is a Alaki Bale that is 25 gradient, has shows the very good compared to the, all the morphological characteristics. So thank you.
Thank you. Any question? Did you check any sugar acid ratio?
One second, sir. Not on.
Sugar acid ratio of the mutant fruits?
Sir, only a morphological characterization was taken, sir, not for any biochemical parameters.
Okay, okay, thank you.
Thank you, sir.
So I think with this we are concluding all the physical presentation. Now there are 5 online presentations are there.
Now the online presentation starts. So there are around 7 online presenters. So check ಮಾಡಿ sir ಅಲ್ಲಿ. Soumya, are you there, Soumya? Soumya. ಇದು ಕುಕುಮಿ ಸ್ಮೆಲ್ ಆಸಿದೆ. Next, Anjineya. Soumya, are you there? Anjineya? He's here. So, offline or online? Okay, Anjineya, you can present now.
Anjaneya, please.
If he is not there, Sandhyarani, Dr. M. Sandhyarani. Sorry, Harshita CB. Harshita CB, are you there?
Yes, madam, I am here.
Okay, you can present now. You can share your slides. Please share your slides. Others, please be ready for next presentations. Harshita, next is Dr. M. Sandhyarani.
See, host disabled participant to share the screen. So just kindly enable us to share the screen.
Okay, now we can share. It's a Arisha.
I think screen is visible.
Yes, you can start presenting.
Yeah, thank you very much. So my presentation topic is on selecting drought resilient or moisture deficit resilient fennel. So insights from stress indices and photosystem 2 efficiency. So fennel is one of the important major seed spice crop after cumin and coriander. So it is mostly cultivated in 1.2 lakh hectares and production is from 2.6 lakh tons, mainly from 4 different states, western states. So due to moisture stress or lack of irrigation during the critical growth period, that is flowering and reproductive stage, there will be yield loss from ranging from 30 to 50, 60%, depending upon the cultivar that is being grown. So in most of the cases, what happens when we go for simply for yield selection under normal growing condition, we go for a better yielders. But in abiotic stress condition, so we always look for a better yielders with a stable performers in both the conditions, stress as well as non-stress conditions. So in this case, simply depending upon getting high yielding varieties which are giving high yield in stress condition, but very huge reduction in the yield is not suitable criteria for selection. So in that case, depending upon stress indices calculation and some other physiological parameters by imaging technique, so better genotypes can be selected. So with this objective, we made identify stress tolerant phenol genotypes using stress indices and PS2 efficiency, that is photosystem 2 efficiency, and to calculate the water use efficiency of these phenol mutants under stress as well as non-stress conditions. So what we did is we developed very stable mutant lines, 43 mutants, along with 2 check, that is mother parent varieties, AF1 and AF2. We did it in a split plot design in 3 replications. 2.5 3 plot size with a spacing of 50 centimeter by 30 centimeter. Using drip irrigation, we raised the crop and we gave the irrigation an ETO approach. During the critical phase of moisture stress, this thing, we gave irrigation 250 mm and 413 mm in stress as well as non-stress plots. We withhold the irrigation during the flowering phase to seed setting. So approximately for 30 to 40 days duration. And we calculated 10 stress indices, which is available in the literature. And we developed a ranking system to calculate the ranking. So we did some PCA and clustering to identify the better genotypes. See, these are the indices that we calculated: stress tolerance index, susceptibility index, tolerance index, yield reduction ratio, mean productivity, geometric mean productivity. So several other things. And this is the field view of our experiments, stress as well as non-stress plots. So this is how the plant was affected with the stress just before the termination of the stress and non-stress plots. So this is the yield of genotypes under stress as well as non-stress condition. So here FM2, AF2, 43, 39, 32, 35, these are all the better performers under non-stress condition, that is stress condition. But there are several other genotypes which are performing good under the non-stress conditions, but they are poor yielders under the stress conditions. So there will be a huge gap in the yield. So difference is there. So with that, if we go for identification of genotypes, there are several genotypes which are omitted or excluded from the selection, which is having a very least difference. So that has been omitted. So what we did is we developed a stress indices and we ranked them. So according to that, like mean productivity, so harmonic mean, stress susceptibility index, stress tolerant index, we ranked them, and average rank we have taken, that is AR. So whichever is having a low highest ranking, that is means less number, so that we selected as a tolerant genotype, that we used as a to use it for selection purpose. Here you can see this is a seed yield under stress condition. So here what we found is several the stress indices like stress tolerance index, which is higher the value, better will be the tolerance index, and mean productivity, relative efficiency index, mean relative performance. These kind of stress indices we can rely upon to select for particularly for abiotic stresses. And for susceptible genotypes like susceptibility index, partial yield reduction rate, susceptibility index can be calculated. Here we made some cluster analysis. So here in cluster 1, the genotypes which are tolerant to stress with the least reduction in yield compared to non-stress conditions are there, like FM2, AF2, FM43, 32, 39, 35, these genotypes. Mostly they are bushy in nature. So they matured, synchronized maturity and flowering will be there. That is why their yield reduction is very less. When compared to a completely susceptible ones in cluster 2, they are susceptible or very least yielders in stress condition. Whereas in cluster 4, we can see they are poor performers in both stress as well as non-stress condition. Their yield level is very, very less. The yield range was approximately from 15.2 grams per plant to 26.5 grams per plant. So here we made some infrared imaging to see how these genotypes, tolerant genotypes, top 5 tolerant and susceptible genotypes in stress as well as non-stress conditions. In susceptible one, you can see that the plant was completely dry, so that the image is having a very bright pink color. Whereas in case of tolerant one, we can see some yellow colors because they have stay green types. So if it is see the photosystem 2 efficiency in tolerant
Dr.
ones.
Harish, please conclude.
Yes, madam. Yes. So in tolerant one, so photosystem 2 efficiency was retained, but in susceptible one, because of loss of photosystem apparatus damage, so they lost the efficiency. These are the genotypes under tolerant genotypes on stress as well as non-stress conditions. This is the susceptible genotypes on stress and non-stress conditions. The interesting to see is the water efficiency of the tolerant ones. They are having
Harish, you can conclude.
So these are the genotypes, that is FM243, that is check AF2, and FM30, 31, 39, 37, 29, 41 are the better genotypes. They give very less reduction in yield, and some certain imaging techniques like fluorescence imaging as well as the infrared imaging can be relied upon to select the stay green types especially for the moisture deficit in fennel. Thank you, sir.
Thank you very much.
Thank you. Next, Dr. Murali, he will be presenting on collection and evaluation of indigenous and wild brinjal germplasm as rootstock suitability for abiotic stress tolerance. Dr. Murali, please share your slides. Okay. The timing is 4+1.
Yes, good afternoon, everyone. I am Dr. Murali. So is it clear, my screen? So I am presenting.
Please make a slideshow.
Murali,
please.
Slideshow.
Go for slideshow.
Right, madam. Good afternoon to everyone. It's my work on a collection and evaluation of indigenous and wild brinjal solanum germplasm as a rootstock suitability for abiotic stress tolerance. So grafting susceptible scions on resistant rootstocks is a valuable tool for managing various biotic and abiotic stresses. So currently Solanum torvum, wild brinjal, is commonly used rootstock for solanaceous vegetables, brinjal, chilies and tomatoes. So Solanum torvum seedlings raising is a challenging task because of poor.
Your slides are not moving, Dr. Murali.
Yes, madam. Yeah.
Your slides are not moving and they are not in the slideshow mode.
It's on slideshow on my side.
No, it's not visible here.
It's
Please close and rejoin once again.
Okay.
Please leave and rejoin again.
Okay, madam.
Next person is, the next presenter is Dr. Dr. Praveen Nayak. Praveen Nayak.
Yes, ma'am. My voice is
So you can please
ma'am?
share your slides. Praveen Nayak, he is going to present on studies on floral morphology, pollination, and seed setting behavior of hibiscus. Please share your slides, and your timing is 4+1 minute. Slideshow mode.
1 minute. It is visible, ma'am?
Yes, please start.
Good evening, dignitaries. My name is Praveen Nayak Keti. I am PhD research scholar from IRI, New Delhi. I am here to present my MSc research work on hibiscus. The title of the work is Studies on Flora Morphology, Pollination, and Seed Setting Behavior of Hibiscus. As we know that hibiscus is also called China rose, and it is commonly visible in South Karnataka and also in South India. It is very rich in nutritional values also, and it is used in Ayurvedic medicines also. Hibiscus is a complete flower and bisexual flower, but still it is unable to breed the hibiscus because of their morphological characters and also their polar morphologies. That's why we conducted the research work with an objective of studies of floral morphology of selected varieties of the hibiscus rosa chinensis and pollination and seed setting behaviors of the selected varieties of the hibiscus. This research work was done in BCKV, West Bengal, during the year of 2021 and 22. For this research work, we selected 10 varieties and we did the pollen morphology. That method is proposed by the Shenin et al. In 2009. And also pollen characteristics, we observed that pollen characteristics like shape, size, diameter, and spines of the on the pollens. This experiment is analyzed by the randomized block design, and these are the different 10 varieties. And out of these 10 varieties, according to the morphological characters, we selected 3 male parents and 3 female parents, and we did the one-line crossings. These are the 10 varieties those we selected for these morphological studies. Here we can see that floromorphological characters of the 10 genotypes, and each genotype shows the different characters, like number of flowers per plants is more in versicular pinwheel variety, and that and the calyx, epicalyx numbers varies according to the varieties. Some are the double varieties, like a double peach. It has a epicalyx more as compared to the other varieties. These are the parts of the varieties, like petals, calyx, epicalyx, styles, everything. Coming to the androecium parameters, brilliant versicular pinwheel and cherry glow shows the better characters compared to other varieties. Those parameters like number of pollens per plant, number of anthers per flowers, and the stamen length and number of spines per plant, per pollens and pollen diameters, and the spine length per pollens. These are the 10 pollens of the different genotypes varieties that we are selected, and we did their morphological characteristics like pollen spine count, and the pollen diameter, pollen area, and also the spine length also. Here we can see that pollen tube germination on the stigma also. Coming to the gynoecium parameters, double peach, sigma, and cinnamon girl, and agni, these varieties shows the better characters as compared, like spine length is less, style length is less, and the ovary length is more, tail arm length is less, and ovary diameter is also more. After the morphological scrutiny, we selected 3 male parents like brilliance, versicular pinwheel, and cherry glow, and we selected 3 female parents like agni, cinnamon girl, and double peach. We did the one-line crossings, and we found that the more percentage of capsule set is seen in versicular pinwheel when it is crossed with a cinnamon girl, and the seed set percentage is also more in these lines. That weight of the 10 seeds per pods, it is more when cherry glow is crossed with
Dr.
the
Praveen, please conclude your time up.
Yes, ma'am. These
Please
are the capsules and the seeds. It is concluded that experiment highlighted that this experiment highlighted the certain obstacles for the seed production after the crossings of the studies, and it guides the breeders and cultivators to making successful hibiscus choices, admiring future efforts in the field. In field. Thank you, ma'am.
Thank you. Next, Shreya Bhosle.
Hello,
Next
ma'am.
presenter, Dr. Sandharani, please be ready for the next presentation.
Am I audible?
Yeah. Please share your slides.
Is it visible?
She'll be presenting on biochemical characterization and studying the influence of plant phenology on the quality parameters of Gymnasium vestris.
Is it visible, ma'am? The slides are visible?
Yes, please continue. Your time is 4+1 minute.
So just to give an introduction about this particular crop, this is an important medicinal crop which is commonly known as Madhu Nashini. As the name indicates, this is an anti-diabetic medicinal herb which originated in India and is distributed prominently in the Western Ghat region. It is a perennial climber, and the bioactive constituent responsible for the anti-diabetic property is the gymnemagenin. This is propagated both through the seeds and the cuttings. The hypoglycemic effect of this particular herb is through different modes, like inhibition of the glucose absorption, increases the secretion of insulin, promotes the regeneration of the islet cells, and increases the utilization of glucose. And in this way, this controls the obesity as well. And why this study is because there is a great demand for this particular herb, both in the domestic and the international market. And as of now, there is no commercial cultivation, and still it is harvested from the wild, which is threatening the survival of this particular herb. So when the medicinal crops are concerned, the content of the natural therapeutic molecules is more important, because the extraction cost of the metabolite influences the market price of the drugs. And hence there is a necessity for assessing the chemical diversity for identifying the most efficient chemotypes in a particular herb. And the phenological analysis of the bioactive content is very important, because a lot of variability is created by the growth stages of the crop on the phytochemical content, and the therapeutic efficiency of any drug, it depends on the amount of the bioactive which is present in the drug. With these objectives, with this background, the objectives were the collection of the Gymnema sylvestre from the diversity-rich areas, the biochemical characterization with respect to the gymnemagenin and other proximates, and studying the influence of the phenology on the biochemical traits. So as a result, we have collected 25 accessions of this particular herb, mostly from the southern Indian states of Karnataka, Tamil Nadu, Andhra, and Telangana. And gymnemagenin was estimated using the HPLC technique, which was standardized by Dhanani et al. And the other parameters are estimated as per the methods given. The results indicated that there was a significant variation in the gymnemagenin content when it was analyzed during the vegetative and the flowering stage. The accession IHR GS44 exhibited the maximum content of gymnemagenin, that is 2% during the vegetative stage, and it slightly dropped down to 1.92% after the flowering stage. So this, therefore, it was evident that the vegetative stage represents the most favorable time for the maximum recovery of the gymnemagenin content. This was the HPLC chromatogram of the standard and the sample. When the phenol content was analyzed across the accessions, there was a significant variation, and the highest value for the phenols was reported in the accession IHR GS45. The other promising lines were also identified in the analysis. Similarly, for the flavonoid content also, during the vegetative stage, the flavonoids were found to have a higher values as compared to after flowering. The PCA analysis of the phytochemicals during the both stages have revealed two factors which was contributing to almost 70 to 80% of the variability between the accessions. And this biochemical, the gymnemagenin, was the major driver of the variability, contributing to the variability or the diversity between the accessions. The analysis of the macro mineral content revealed that the leaves were rich sources of calcium and potassium, and in terms of the micro mineral, the leaves were rich sources of the iron. In terms of after flowering also, there was a higher content of calcium and potassium reported, but the contents were reduced when compared to the vegetative stage. So as a conclusion, this is the first attempt to collect and conserve the large number of accessions of this particular herb from South India. Significant biochemical variability was observed for the phytochemical traits, and gymnemagenin was the major driver contributing maximum to the variability between the accessions. The phenological studies revealed that the vegetative stage represents the most favorable stage for maximum recovery of the phytochemicals, and few of the promising chemotypes which were identified in this study, they can be used for commercialization, for the commercial cultivation of this herb, and also for future breeding programs. Thank you.
Thank you, Shreya. So the next presenter is Dr. M. Sandhyarani.
Yes, madam.
Yes. Your time is 4+1 minute.
Okay, madam.
Please share your slides. Next presenter will be, please be ready, Dr. V. Murli. Dr. M. Sandhyarani. 9. Serial 9. If your internet is not good, you just check it once. We will call Dr. V. Murli.
Okay, ma'am.
Okay. Dr. V. Murli.
Okay, madam, I am ready. Audible?
Yeah, you can please share your slides.
Yes, madam. This time, okay, madam? Screen?
Yep. Slide presentations mode.
Right, madam. So, good evening to all. I am presenting collection and evaluation of indigenous and wild brinjal germplasm as a rootstock suitability for abiotic stress tolerance. This is the non-plan.
Slides are not in a presentation mode.
Oh, it is from my side. It is showing.
Slide. It's not in the slide presentation mode.
Slides are showing up, madam, in the other screen. It's okay.
It is not visible here.
Oh.
Check your internet.
Internet is okay.
Slideshow.
It's on slideshow from my side. Slides are moving. I don't know what's going on here.
Okay, please continue with your normal slides.
Right, madam.
Now it is not visible. Slides are not visible now.
Okay. So we did, madam, what we did, we collected indigenous germplasm from local Adilabad farmers.
On your presentation, please.
Yeah, it is
Open
there,
the
madam.
presentation, PPT. It is not visible.
Okay, I will get it. Share screen. Window.
Okay, we will share your presentation here. Please present.
Right, madam. That's okay. So, oh. It's opened up, madam, there.
Okay, you can continue. Slides are visible to you?
Oh, yes, madam.
Okay, you can continue.
Right. Good afternoon, everyone. So this is the non-plan workstation at Horticultural Research Station, Adilabad, on collection and evaluation of indigenous and wild germplasm at tribal areas of Adilabad district. Next slide, madam. So grafting susceptible rootstock on tolerant ones is a major outcome to go away with abiotic stresses. So Solanum torvum is currently used as a common rootstock. And so in place of that one, if you have the indigenous and wild collections, if they found it better, so that will be a replacement for Solanum torvum. Next slide. Next slide, please, madam. Move on. So we got, we have done a plant exploration survey in July 23. We got indigenous collection Pandiri Vankaya. It is a beautiful one. It the crop, the plant lives up to 8 to 10 years in a backyards. It is widely grown and it is resistant for abiotic and biotic stresses. So farmers say that it is a perennial type. It can overcome the borer problem and they can choose it for tasty also. So Solanum virum, nightshade or soda apple is common in our area. They are present in Mavala forest range. We brought them. We conserve them at Horticultural Research Station, Adilabad. Screen for abiotic stresses, waterlogging, drought, and salinity. This is the preliminary work we did. So next slide. So waterlogging, plants were withheld for water was kept for the week and scored for their symptoms like plant is healthy if it is 1 and 2, wilting, same like that up to 5 points score we utilized. Similarly on drought 11 days in the pots withheld for water, 0 is no symptoms and 9 all plants died. Salinity, artificially introduced salinity with 25 millimolar sodium chloride. So when the plants are showing up symptoms of salinity injury like leaves retarded and small rolled out, so symptoms were scored. Next slide, please. Next slide. So germplasm with the germination percentage, Solanum tarum is having very low low germination percentage. It took 63 days to retain the grafting mean of fifth true leaf stage. It took 63 days. Whereas these local collection 45 days we got. So nearly 3 weeks time we can say with indigenous collection. Seedling length is also very good and within a quicker time. So seedling vigor index, a product of multiplication of germination percentage vigor index is also very good with indigenous collection. Next slide, please. So regarding waterlogging tolerance, Bhagyamati indigenous collection has the lowest score, means it has a better tolerance. Drought is also same. It is having Solanum tarum is having higher tolerance. Salinity, with regard to salinity, this is a good one and Torworm. So cumulative score accordingly lowest score I had with IC 7.5. So we can say that Torworm followed by indigenous collection can be used for breeding purposes. Next slide. So based on germination percentage, we can say that with quick growth, we can use Solanum Pandiri Vankaya followed by Solanum Vairam in case of germination and other things are concerned. In other cases, we can use Torworm for salinity tolerance. So these germplasms are very valuable. We are trying to register with NVPGR germplasm division. So we are planning for establishing a physiological and molecular basis of various biotic stresses. Thank you, madam. Any questions?
Thank you. Thank you. Next presenter is Miss Sandhya Rani. So we will share the presentations here. Just present your slides. Dr. Sandhya Rani. Dr. M. Sandhya Rani.
Hello, madam.
Yes,
I'm here, madam.
Okay, please present your slide.
Is that visible? My screen is visible, madam?
Yeah, slides. We only shared your slides here.
Okay, madam.
Just
Thank you,
present
madam.
it now.
Okay, madam. Myself, I'm Sandhya Rani from Fruit Science Department. I'm PhD scholar. Coming to the title, today I'm here to present my PhD work, that is studies on extent of genetic diversity among the mango germplasm by using quantitative characters. Coming to introduction, mango is the most nutritive and delicious fruit crop belonging to Anacardaceae family and originated in Indo-Burma region. Due to its popularity and importance, mango is often named as king of fruits for its excellent flavor and taste. The variation among the genotypes for quantitative characters were important for considerable crop improvement in mango, and it could be achieved in most of these characters by selection. Hello, madam. Coming to the objective, to study the extent of genetic diversity among the mango germplasm by using quantitative characters. Coming to the materials and methods, the research was conducted at Mango Research Station, Nujividu, Elur District, Andhra Pradesh during the period of 2021 to 22 and 2022 to 23. The experiment was laid out in augmented block design consisting of 36 genotypes and 4 checks. These are the different treatment details that have used 36 genotypes and 4 checks. The checks that have used are Banganpalli table variety, Chinnarasam juicy variety, Jalal pickle variety, and Suvarnareka colored variety. And these are the different observations that have recorded. Coming to the first character, height of the mature tree, the minimum height was 6.33 meters and the maximum is 13.58 meters. 8 genotypes recorded the best among the checks. Next, the trunk circumference ranged from 0.53 meters to 2.63 meter. Among the checks, Banganpalli 1.770 meters. 13 genotypes recorded maximum than Banaganpalli. The leaf blade length varied from 15.34 centimeters to 26.96 centimeters. The Suvarnareka recorded the best among the checks. The 15 genotypes recorded the best than the check Suvarnareka. Next, the leaf blade width ranged from 3.52 centimeters to 6.49 centimeters. Among the check, Banganpalli recorded 5.26 centimeters of leaf blade width, and the 12 genotypes recorded maximum than Banganpalli. The petiole length ranged from 1.41 centimeters to 5.36 centimeters. The Chinnarasam recorded the maximum petiole length, that is 3.74 centimeters, and 4 genotypes recorded maximum than the best check Chinnarasam. Coming to the inflorescence length, it is ranged from 15.12 centimeters to 34.61 centimeters. Among the checks, Chinnarasam recorded maximum inflorescence length, that is 27.69 centimeters. Inflorescence width ranged from 8.08 centimeters to 26.06 centimeters. Among the checks, Banganpalli recorded the maximum, that is 16.76 centimeters of inflorescence width. The number of hermaphrodite flowers ranged from 27.54 to 175.44. Among the checks, Suvarnareka recorded the highest number of hermaphrodite flowers in the inflorescence, that is 167.50. The fruit length ranged from 9.74 centimeters to 21.35 centimeters. The Jalal among the checks, the Jalal recorded the maximum fruit length, that is 16.62 centimeters. The fruit diameter ranged from 17.25 centimeters to 27.79 centimeters. Among the checks, Banganpalli that recorded the maximum fruit diameter, that is 27.79 centimeters. The fruit weight ranged from 135.03 grams to 605.35 grams. The Banganpalli recorded the maximum fruit weight. The pulp weight ranged from 40.27 grams to 270.31 grams. Among the checks, Banganpalli recorded the maximum pulp weight. The pulp percent ranged from 25.66% to 68.70%. Among the checks, the Jalal recorded the maximum pulp percentage, and the pulp to stone ratio ranged from 1.60 to 6.60. Among the checks, Suvarnareka recorded the maximum pulp to stone ratio. The peel percent ranged from 10.93% to 34.43%. The Chinnarasam recorded the maximum peel percentage. The number of fruits per tree ranged from 30.11 to 989.49. The Banganpalli recorded the maximum number of fruits per tree.
Conclude your timer. Please conclude.
This is the diversity among the trees. Among the genotypes and checks, this is the diversity among the leaves. Here you can see the diversity among the inflorescence. And this is the diversity among the fruits. Of 36 genotypes and 4 checks. Coming to conclusion, in present findings, the quantitative characters that have studied showed significant variation. The variation among the genotypes for quantitative characters were important for considerable crop improvement and could be achieved in most of these characters by selection. Coming to future line of work, accessions E6 and H49 was appeared to be the promising donors for fruit yield, showing the maximum fruit length, fruit weight, and pulp weight. The promising genotypes, which are superior in different desirable characters, can be used in breeding program to incorporate those characters in high yielding genotypes. Thank you.
Thank you. So we have come to the end of this session, technical session 2, smart breeding in horticulture crops. We sincerely thank keynote speaker, lead speakers, and all the oral presenters of this session. And also I sincerely thank on behalf of organizing committee, chairman and co-chairman and rapporteurs of this session. Now it's time to honor our chairman and co-chairman of this session. I request Dr. C.N. Hanchinmani sir, Dean COH Shirsi, to felicitate our chairman and co-chairman of this session. I request Dr. B. Fakhruddin sir, Director of Research, University of Horticulture Sciences, Bagalkot, to please come on the stage. Another session, session 3, smart horticulture farming. The session will be continued in E-Studio in the main building. Second, DOE office, Director of Education office in the main building, second floor. I request Dr. Balaji Kulkarni sir to join for this event. Thank you, sir. Now I request Dr. Shantappa sir, co-chairman of this session. And I request Dr. Balaji Kulkarni sir to felicitate co-chairman of this session, Dr. Shantappa. Thank you, sir. I request Dr. Rekha Chittapur and Dr. S.R. Mulla, session rapporteurs, to receive the certificates from Balaji Kulkarni, sir. Thank you, sir. Thank you, one and all. Now the cultural program will continue in this auditorium, and the second session theme 3 will be continued in the Director of Education office at main building. So those who are having presentations, please attend the session in the Director of Education at the main building, second floor, first floor.
So dear participant, please note the theme third, smart horticulture farming presentation, oral presentation will continue in the main building DOE office. Those who have oral presentation, kindly move there and make your presentations. So now this hall will be open for cultural programs. Government. Okay. And NGT awards will be distributed here only. And those who have already notified for the award, please stay here and receive the award. Shortly, they will announce the list.
Madam, you
ಒಂದು ಅವಾರ್ಡ್ ಕೊಟ್ಟು ಬಿಡ್ತೀವಿ ಒಂದು 5 ನಿಮಿಷ ಅವ್ರು ರೆಡಿ ಆಗುತ್ತಾನ ಕೊಟ್ಟು ಬಿಡ್ತೇನೋ ರಟೋಡ
ಒಂದು ಜಾಸ್ತಿ ಸರಿ
ಬರ್ರಿ ಎಲ್ಲೋ
Dear participant, please note, theme third, smart horticulture farming presentation will be continued in the DOE office main building. Please, those who have a oral presentation, kindly move to the DOE office auditorium, please.
So all are requested to come to DO office meeting hall. It is at main building. No.
Hello. Check, check, check.
Good evening to everyone. It's a beautiful evening and we are thrilled to have you all here.
Yes, and the energy in this place is already electrifying. We can see those excited faces waiting for a fantastic evening ahead.
Absolutely. And why not? We have an incredible lineup of performances that will make this night unforgettable.
But before we dive into this excitement, let's officially welcome you all to this cultural evening where talent meets celebration.
Now, let's take a moment to extend a warm and heartfelt welcome to the esteemed dignitaries who have graced this occasion with their presence.
It is my privilege to first welcome our visionary leader, Dr. Balaji S. Kulkarni, Dean, College of Horticulture, Bagalkot. We welcome you, sir.
Been complete without all the dignitaries on and off the dais, including our teaching and non-teaching staff.
Moving forward, a warm welcome to all the heart and soul of this event, delegates and attendees of the global conference, and all our incredible students.
So, with all these remarkable personalities gathered here, we are all set for an evening filled with vibrance, culture, and celebration.
Now, what's a celebration without a classical performance which embodies our pride, Bharatanatyam? And to do just that, we have a very talented dancer, Pranavi. I now request her to take the stage and enchant us with her performance.
तेरे दर पे हम भरे तूने दर पे आ तू देवा तेरे दर पे हम आए हैं तू देवा तेरे दर पे हम आए हैं
Now we welcome RM Hiremat sir to provide a certificate to Pranavi.
Engineers for an electrifying.
ಸರ್ ಇನ್ನೊಂದು ಪಿನ್ ಇತ್ತಲ್ರಿ ನಿನ್ನೆ ಅವ್ರು ಕೊಟ್ಟಿದ್ ನೋಡ್ರಿ ಎರಡು ತಂದಿದ್ದೀವ್ರಿ ಒಂದೇ ಇಲ್ಲಿ ಒಂದು ತೊಗೊಂಡಿಲ್ರಿ ಇವತ್ತು ಇಲ್ಲಿ ಕೊಟ್ಟಿದ್ರಲ್ಲ ನಿನ್ನ ಒಂದು ಇನ್ನೊಬ್ರು ಇದ್ರಲ್ಲ ಸರ್ ಅಡ್ರೆಸ್ ಅವ್ನ ತಗೊಂಡಿರ್ಬೇಕು ಯಾಕಂದ್ರ ಎರಡು ತಂದಿರಿ ನಾವು ಸರ್ ಈ ಲ್ಯಾಪ್ಟಾಪ್ ಇಲ್ಲ ಅಂದ್ರೆ ಶಟ್ ಡೌನ್ ಮಾಡ್ಬೇಕು ಇಲ್ಲಿ ಬರ್ಬೋದು ಸರ್ ಈಗ ಸರ್ ಈಗ ಎಲ್ಲ ತಗದೈತಿ ಬರತ್ತೆ ಇಲ್ಲಿ ಸರ್ ಬರ್ಬೋದು.
Respected Chairman, Co-Chairman, Deputies, and moderator. Now we are going to start theme 3, that is Smart Horticulture Farming. So in this session, we are having one keynote speaker by Dr. Director of Research with Bagalkot, and then we have two lead speakers for different topics. So to start today's program, so first we will start with a keynote address by Dr. P. Sir. Sir, it's a great pleasure for me to introduce you to this session. Sir has almost 35 years of experience in research and architecture. Sir is art and also research in different space. Sir is involved in raising many hybrids and varieties, especially brinjal, tomato, bean, etc. And Sir has guided more than 11 PhD students and more than 40 students. And Sir is also working as a mentor for students. Sir is also having a credit with more than 20 national funded projects and NPI. And Sir is actively involved in the institutional building efforts, having more than 200 publications in journals, conducted many training programs, and Sir is also involved in and a member in the different institutional societies. So with this short introduction, now I request Dr. Sir, Director of Research, UHS-Main, and also the Dean of College of Horticulture, Engineering and Food Technology for this today's keynote address on the topic disruptive smart farming technologies for sustainable production. Welcome you, sir.
ಚೆಕಪ್ ನಾಳೆ ಟೈಮ್ ಕೊಟ್ಟೆ ಮತ್ತೆ ಅಲ್ಲ ಅದಕ್ಕೆ ಮತ್ತೆ ನಾಳೆ ಟೆಸ್ಟ್ ಮಾಡಂತ ಅಂದ್ರೆ ಅವರು ಸ್ಕ್ಯಾನಿಂಗ್ ಲಿವರ್ ಅಥವಾ ಬೇರೆ ಏನ್ರಿ ಅದ್ರಲ್ಲಿ ಆಗ್ಯದ
Participants are requested to be ready with your PPTs.
Respected Chairman, am I audible? Yes. Okay. Respected Chairman, Co-Chairman, all delegates, good evening to you all. I'm happy to make this keynote presentation on advances in disruptive smart farming technologies for sustainable food production. This is the kind of talk I thought is fitting to this theme of this conference. So basically, we have a couple of challenges. I take you through a couple of challenges and then see what kind of advances are there in technologies that are coming up. Challenges in agriculture industry in kilos, elevated increase in demography that basically deems the, boosts the demand for the food. The kind of scenario that is prevailing right now is we have 7.6 billion people. From there, it's likely to go to 10 billion people by 2050. We all know about it. But actually, expected rate of growth is 33%. And given the fertility and other scenarios, it could hit even 16.5 billion by 2050. That's humongous population. So that's the biggest challenge, and since morning everybody is talking about it. But there is another perspective for that it's not actually 10 billion, it's also 19 to 16 billion, which is going to be even more tough. One key thing that we all can see is urbanization is increasing. A net addition of 2.4 billion people to towns and cities is happening. This trend generally raises income. The moment you move to urban, income levels rise. The meaning of that is what? Demand for food increases, and that also leads to the crisis in child obesity kind of thing. These are all new things that we will have. So net increase in, net higher increase in 70%, higher increase in production need to be achieved. And even meat production is likely to increase its consumption from 36.4 kgs per person to 45.3 kgs. This means what? An effect on increase in meat production, and that eventually will have environmental concerns. So this is first challenge I feel is one important big challenge. And the second challenge would be current use of natural resources are highly stressed. The natural resources that are in front of us, they are all highly stressed, and how they are stressed. World's farmland is becoming increasingly unsuitable for production. That's happening. About 25% of the farmland is already rated as highly degraded, and about 44% of it is moderately or slightly degraded. And water resources are highly stressed. More than 40% of the world's population is living in water-scarce area. So these are all very big problems. Land shortage has resulted in small farmhood. At the moment we have smaller farms, and the rest of the things are, we know, everything are the poverty, and this is the biggest problem. If you carefully look at all these things, the primary cause is agriculture. The primary cause is agriculture for all these things. Other few perspectives in this is that direct and indirect cause of land degradations are related to agriculture in any manner if you look at it. Deforestation of unsuitable land, overcutting of vegetation, inadequate fallow periods, overgrazing, improper crop rotation, unbalanced fertilizer use. All these things are in one or other way related to agriculture, and that's responsible. The third biggest challenge in front of us would be climate change, and this climate change is reducing productivity. And this is clear, we are seeing it in all spheres of our life. Agriculture is one of the primary causes of greenhouse gases, and we very clearly know about it. And there is 2 times increase in greenhouse gas production by in the past 50 years, and it's likely to increase unless we take care of it. And what is the net result? It results in reduced production. That's going to be the cause of this particular thing. The fourth challenge is food wastage. Food wastage is a massive market inefficiency and an environmental threat. And the kind of thing that is happening is about 33 to 50% of the food we produce is never eaten. That's really not a good thing at all. And it is equal to about 25% of the potable water, fresh water. So that's the kind of thing in front of us. And if food wastage were to be a country, it would have been third largest country to contribute to the greenhouse gases indirectly. So that's the alarming kind of situation, and that would have been the next to China and US, which are contributing highest right now. So what is the outcome of all these things? Actually, is poverty and hunger. So this is the challenge in front of us, and we need strategies to produce more food, to have more productivity, and also have efficiency in delivery mechanism. So when you look at these things, this conference really addresses this very issue, and that's what in front of us is disruptive production systems, really a new technology such as Agriculture 4.0, which in other words we also call it as digital agriculture or smart farming, all the same actually, all words are same, one and the same. And this particular technology realm is likely to give some solutions. So traditional agriculture is undergoing a fundamental transformation over a period of time. First technology revolution in agriculture made very impressive strides, as all of us know. And we have something called agriculture 1.0. That's the agriculture our forefathers did. Possibly from about 12,000 years to until say Mendel's last rediscovery, beginning of this century, last century. Until that time, we can call it as agriculture 1.0. And then from there onwards, like from 1920 onwards, you can say, would be agriculture 2.0, where all these hybrids, production of hybrids, application of principles of genetics happened to the agriculture. And that's also actually responsible for our green revolution. The agriculture 3.0 is actually agriculture 3.0 is something that happened in 1990s, and typical agriculture 3.0 is the signature for that is GPS signals, application of GPS. Basically, application of GPS in agriculture marked the beginning of agriculture 3.0, and this is something somewhere early 1990s. And this used aerial sprayings using GPS. People started using aerial sprayings. And the first automatic steering in the late 90s, tractors with automatic steering kind of thing that happened in late 90s, that's because of the GPS. They are linked to one another. And during 2000, guidance accuracy increased to 1 centimeter. The GPS guidance increased to 1 centimeter. That made all the big difference in bringing accuracy in digital realm, and that's how it happened. Now, other things that if you see, the coming agriculture revolution is going to be the agriculture 4.0 or digital agriculture. And what it is, science and technology at its heart. It's all science driven, invariably science and technology driven. And a new boost in precision farming would happen, and that would be because of, look at that, cheap and improved sensors and actuators. They are the core of this digital realm. And then low cost microprocessors. You need microprocessors which are low cost so that you can apply heavily. Then higher bandwidth cellular communication. You need communication, and that makes all the like your 4G, 5G, and all of that. And then cloud-based ICT applications, cloud computing and other things. What we do, what we right now trying to use, that makes a difference. And then big data analytics. In the past maybe 6-7 years also, the big data analytics is becoming impressively important and becoming a reality, and that's at its core of precision farming. The aim of all this is to improve. Aim is to improve needs of consumers and re-engineering the value chain. See, needs of the consumers and re-engineering the value chain. It's also production and then supply of food. Everything has to be looked at little differently. So with that, what would happen? Agricultural operations will work more differently than what it was, and we all know about it now, right now. And the advancements in sensors, devices, machines, and information technology would be at its core. Then this involves targeted application of water. With all this, what happens? It would result in targeted application of water, fertilizer, pesticides across entire field. Targeted application within field. It's not that whole field you would apply, but it would be targeted application. Use of abundant and clean resources of energy, like sun energy or sea water to grow food. This kind of completely different perspectives. Farmlands in deserts. You have a lot of deserts. Now we will see farmlands in deserts. Somehow we have to have our food being grown in farmland, deserts. That would be the likely outcome. Then next is like vertical space. Or vertical space will be used to produce more food. In the food production realm, vertical space will be used. You are growing in one layer, now you would grow in multiple layers. So that would add to more food. So these are the technologies that are like that have to come in the time ahead. Reduced food wastage. See, reduced food wastage through technological interventions should happen. That's what is Agriculture 4.0 all about. Now, this means what? This means expansion and intensification. You can achieve all these things either by expanding or by intensifying. So, what do we mean by this? We have a very good picture in front of us. Look at here. This is capital, money. And down here, x-axis, land area. Look at these geographies. You have Australia. There's plenty of land out there, plus plenty of technology. And they are expanding land also, technology-wise also. That's why they are moving like this. Look at Europe. If you look at Europe is not interested in expanding, but technologically they are growing. That means they are using more and more new technologies to produce more. And look at Brazil. Brazil is a country which is expanding with little innovations inside, right? That's Asia. Look at here. Asia. For irrigated rice, we are moving up. Area is not expanding, production is increasing. That means technologically things are getting in. Look at dairy in Europe. It's happening both little bit. So this is the kind of scenario that is likely to happen. And what would real technological interventions into the production realm would be what's happened to the Europe. Basically, you want to retain the same area but produce more. That means technology has to be applied heavily on that. So what are if this has to happen, what are the new technological solutions in 4.0, Agriculture 4.0? If you ask this question, 3 general trends where technology has higher potential to disrupt the production system. There are 3. I will discuss on 3 of them. So one is produce differently using new technologies. You are producing right now, but now produce differently using new technologies. That would be one stride. The second one would be use new technologies to increase the efficiency. You need to increase the efficiency in the food production chain. That's second one. The third one would be incorporation of cross-industry technologies and applications. That means what? You now try to bring some technologies which are cross-industry. You are using it for something else. Now try to bring that here and make a big difference. These are the 3 different ways in which a disruption in the system could be brought. So okay, let's get into each one of that. Produce differently using new technologies. What is happening here? What is disruption out here? This is what is disruption, vertical farming. Conventionally, we grow up on a horizontal format. Now the vertical format is on the horizontal format, you have layers and layers. That means you produce more. So that's the vertical farming realm that we talk about. Then the desert horticulture and seawater farming. Desert anyway is not in the production realm right now. Now try to come out with technologies and ways and means where deserts are pushed into production. That means you will be now producing differently. And seawater farming, seawater is right now not producing any food for us. Now you ensure that the seawaters are pushed into cultivate food production realms. Like algae in feedstocks, for example, right? Use algae. Algae, we all know how nutrient-rich it is. Sustainable packaging, bioplastic. This packaging seriously is a big issue. We won't take anything without packaging, remember. Packaging is a very important issue, but sustainable packaging is a very critical thing, like bioplastic kind of approach is very essential. Then, let's look at this vertical farming as we see. This vertical, just to give you a glimpse of it, you all know about it. Vertical farming is a process of growing food in a vertical stacked layers. And vertical farming encompasses a range of growth systems of different scale, users, technologies, location, and purposes. And it is particularly suitable to cultivate horticulture crops such as leafy vegetables, right? And let me give you an example. And this particular process uses 95% less water than conventional production. That's the beauty of this particular approach. And therefore, it is disruptive. We have an example there. Netherlands right now produces about 35% of its country's vegetables using just less than 1% of its farmland. That means they are using vertical stacks to produce more fruit. So there are clear-cut examples in filters. I don't go into details into this. Vertical farming uses, all these relevance, deep water culture, hydroponics, nutrient film techniques, aeroponics, aquaponics. These are the kind of systems that are there. But there's a lot of AI there. There's a lot of interventions that are coming into each one of these systems. These are conventional formats, but there is a lot of advancements that are the cross-industry, what I mentioned to you, those cross-industry technologies are coming in here and it's making a big difference. Now, categories of vertical farming, if you see, stacked horizontal systems are available, then vertical growth surfaces are available. We have something coming up these days called BR, Building Integrated Agriculture, and this is basically this is the waste infrastructure that's available is being pushed into that. Rooftops are being pushed into agriculture, like what we call like on buildings you tend to go from rooftop and kind of thing. So yeah. I know. So horizontal star systems. I know. I completely understand. So these are the kind of things that are there, right? These are the factories that you see, food factories. So these are the considerations that are there in each one of these. Then let me come to use of new technologies with increased efficiency in food. Basically, you have genetic modification, cultured meat, application of 3D food printing technology. These are three disruptive methods. Genetic modification, basically, we talk about CRISPR-Cas. It is very site-directed, completely directed. It's unlike protein. And cultured meat is another thing which is coming up. About half decade ago, there was only one company which was trying to do, but we have so many companies now. Cultured meat, no butcher here. You are not killing the animal. You are culturing meat in the media and then trying to eat. Number of companies are increasing. These are the disruptive technologies that we are talking. The food printing, 3D food printing is likely to make a very big difference because you will make your idli the way you want so that you will not waste at all. So this is going to make a big difference. So incorporation of cross-industry technologies are basically this increases the precision of the agriculture, wherein the trends are like IoT, automation of skilled manpower, farming and robotics, RFID. These are the kind of technologies that are likely to get into. Then data-driven farming, the whole of the data, agri-horticulture systems will be data-driven, be it weather, soil type, any of these factors are going to be thing. Another thing is like chatbots. We had chatbots in our phones previously. Now chatbots are available. So these are the kind of robotic agbots that are available right now for us to do things. And many of our Make in India is quite good into this, and many of these things are coming. And then drone technologies is something that is really making a big difference. It is likely to make a difference as we move ahead. Like be it soil, planting, cropping, any of these things are going to be big things. See here, these are the farms are going to be, and many things are coming up in India as well, but these are farms are going to be. Look at the accuracy, 0.1 meter. That means 10 centimeters is the accuracy that we are talking about, and you can have regulation over this. In 10 centimeter accuracy, you can decide what you want to apply, what you don't want to apply. So that's the sort of thing that is likely to come. And we'll have you see the points there. Differential application will happen using this kind of sophisticated machineries. So you basically map the land and apply what you want to apply. You don't apply fertilizer where you need not have to apply. Even if you are applying fertilizer, you apply very differentially like this. So it's going to be, this is the kind of disruptive technology that is likely to getting into, and it's going to be much more, right? So this and it is becoming reality as we are going. This one particular thing I would like to say and maybe close, like in September 2017, something which is at Harper's Adams University, UK, man never entered a field, land was prepared, sowing was done, crop husbandry happened, harvested, sold. Everything happened completely remotely. That's the beauty of it, and that marked the beginning of real application of digital farming or agriculture 4.0. That's kind of thing that happened. I mean, it's a milestone, and that's spreading heavily everywhere, and that's what is what we. So we have many other technologies in this, but what is very important in this role of government, and we need to have policies that have to be very favorable with us, and then financing and finishing schools with respect to this, and see that wherever there are gaps, there are filled in. So this is what I wanted to bring for you in this session. Thank you.
Thank you. Thank you for giving us a very beautiful keynote address for this smart horticulture farming team. Thank you. Next.
Once again, I thank you, sir, for enlightening us on the smart horticulture technologies for enhancing food grain production. Now, I request Mr. Murthy, sir, Innova Technology Solution, Mysore, Private Limited, Bengaluru. Yes, sir, here you have to come. Sir is the lead speaker for this session on the topic, the aeroponics farm future farming technology for the urban and peri-urban horticulture. I request Mr. Madhav Murthy, sir, Innova Technology Solution, Mysuru, for the lead song. Sir, you are PPT. ಹಾಕಿದ್ದೀರಿ.
ಏನು ಅಲ್ಲೇನ ನೋಡೋಣ ಮೇಡಮ್. ಅಲ್ಲಿ ಆ ಲ್ಯಾಪ್ಟಾಪಿಗೆ ಬರ್ತಾರೆ ಆ ಕಡೆ ಒಂದು ಲ್ಯಾಪ್ಟಾಪ್. No. ABCD. This PC of the phone. This PC of the phone. This PC. ಅಲ್ಲಿ ಕೆಳಗಡೆ ಅಲ್ಲ ಅಲ್ಲಿ ಏರೋಫೋನಿಕ್ಸ್ ಇದರಲ್ಲಿ UHS. Yes. Okay. Thank you. So I want to ask apologies for all of you to be staying this late because we are here to speak. Otherwise, you would have enjoyed the function over there.
Okay, sir.
But luckily, thanks for the university for inviting me to be here, and the dignitaries who are here to moderate this whole thing. Just to introduce myself, Madhav Murthy, working in aeroponics for the last 15 years. We made a statement by 2040 we should have aeroponics as the ruler of the farming, especially in horticulture space, because I should thank Dr. Fakhruddin. He covered most of the points which I wanted to tell. So I'm very happy he has created a ground farming. This is my this one. Okay. So we have this one of our farms. We talk about horticulture 4.0. That is where we see that lot of things are going to happen. So without any much talking about, I'll just go on the things. Now, with the population expansion, whatever Dr. Fakhruddin said, it also means that there is going to be a reduction in the agriculture land. When the urban places increase, it is only encroachment of agriculture land, and the focus on agriculture land will be on agriculture produce rather than the horticulture, because most of the horticulture crops are perishable. Main agriculture crops like paddy, wheat, and pulses and all, post-harvest you can store it. Here the importance is now to look at urban and peri-urban areas where, because it is expanding, it is also expanding vertically and horizontally, how we can utilize this space without the use of soil. That is the whole crux of the matter. Okay. Just to talk about it, horticulture is the practice of growing fruits, etc., within cities and peri-urban areas. Here we integrate a lot of farming technologies, sustainable practices, and innovative cultivation methods, mainly to maximize the food in given space. Okay, Dr. Fakhruddin talked about the agriculture 1.0 to 4.0. Today, looking at horticulture 4.0, it's purely technological progression. Okay. Here, some of the points I want to highlight is that the main driver today, if you look at it, from human and animal labor to mechanization, then with the introduction of fertilizers, now in Agri 4.0, it's all about bringing technologies and automations. And the technology that we adapt here is the IoT, AI, drone, and a lot of things. And the last point I want to highlight on this is on the farming style. Earlier, it started off as sustenance in farming when we started it off, to industrial scale farming, which was monoculture approach, to intensive monoculture approach, to today it is precision farming. Okay. So it's all about digitization, automation, sustainability drive, agricultural innovations. Aeroponics stands out as a revolutionary cultural method. Aeroponics involves growing plants without soil, where the roots are suspended in the air. And along with aeroponics, there are other cousins, which I will come to that. Okay. The biggest advantage you have is that you can adapt technologies for monitoring the plant health. One of the things we have experimented is that when we notice some variation in the behavior of plant, when we give the dosage of their nutrient or fertilizers or any supportive input, it takes about 4 to 21 days for it to be absorbed by the plant. In aeroponics, we have noticed that we can have the reaction within the next 4 to 6 hours. That's the type of direct control you have over plants. Okay. Horticulture, it's all about real-time monitoring, automated nutrient delivery, and data-driven. I also hold my postgraduate in big data and cloud computing and IoT. We manage a lot of things as the data-driven decisions. Resource efficiency and sustainability is what we really need to be focusing on. And my whole intention was that I should speak to the universities to see that these are some of the points we should drive for the students over there. Okay. We talk about water efficiency. We have seen, in what we cultivate with 1 lakh liters, we have done it in 5,000 liters. Okay, because we directly miss the roots, the actual quantum of fertilizer used by the plant only can be given. Okay, and then the energy, here we are also bringing the off-grid power. Okay. So when we talk about the controlled environment agriculture, aeroponics stands the method of doing it. This is another thing, it's about you can have year-round production. This alters the perception of the conventional thinking of farming, especially in India, we talk about monsoon-driven cultivation. Seasonal, the whole climate issues. Here, we can do it around the year. And pest control is one of the easiest things. Very rarely we use herbicides and pesticides and all because it's off soil. Majority of the diseases from the soil is reduced. Okay, then cultivation time is much faster compared to the thing. And the nutrient content, see in the soil, with all the things, depending on the particular geography of the soil, the composition of the nutrient will determine the behavior on the plant growth. Here, because the academicians have done enough and more work to say this plant during this phase of growth, this is the best nutrient that you can feed onto the plant. This can be controlled very easily in aeroponics, because of which you can see the consistency in the plant growth. It's all about how do we support urbanization, is that with the increasing concrete space, now this space will become the future of farming activities. Now when we talk about urban food security, we grow it on rooftops, vertical farms. Aeroponics contributes for a local food production. One apartment can supply for the entire residents over there. Okay. This also helps in carbon footprint reduction because whatever is grown there is consumed within the periphery of 1 to 5 kilometers. And still, it's not that it's the, wow, but it is wow, but it comes with a lot of riders. What are the challenges that we face is that the cost factor is really very heavy. It's not that it's cheap and things like that. But because of urbanization, there is a bit of urban dwellers can afford to pay more because of the things. But it's an issue in the initial setup cost. Then there are a lot of technical complexity in it and energy. This is something we are working with off-grid, like solar and wind. Okay, so what the features are is that you can save the space. Like what I can grow in 1 square meter of soil-based, in that I can grow 20 to 25 times the yield per area. and about 3 to 6 times the production per plant. So this is the okay, and see, because it's so fresh, you don't have to bother much about it's free of pesticides and grown. It's not stored in a cold storage. Okay, and okay, I'm trying to hurry it up. Hopefully, I'll do it faster. Okay, we look at in urban horticulture, vertical farming, aeroponics, rooftop. Okay. These are the different types of, okay. One of the things we talk about is the terrace gardening, which is just, okay. What is the benefit of? One is efficient use of space. You can convert whatever was balcony or, uh, in the around the house in the villages, what people are growing now because you don't have space. You started growing in the front and then now balcony, then it goes to the terrace. Okay, it's all using of the unutilized space. And it creates a lot of environmental benefits. One is that when you grow within the house, around the house, the air quality better, you can get fresh food, you can harvest and eat it. The water management, like I said, that not only that you use less water, plus you can also link it up with rainwater harvesting. So it has a double benefit in this, and it contributes to sustainable urban living and reduction of carbon footprint. Okay, so these are some of the methods. The just benefits like it is safe, pesticide-free, healthy, conducive, it means whatever it's just clarification. It also reduces the heat and cold within the building and the sound pollution. And you can also, we have experienced it. Uh, In fact, that is one of what we should you have the presence of what after my role here.
Yes. So next lead speaker is Dr. Nataraj SK, Assistant Professor and Head HREC Hassan. The topic is new flower crops for doubling the farmer's income through protected cultivation. I welcome you, sir, for this session. Sir. Present.
Yeah, good evening. I know the time constraint. Briefly, I'll rush my presentation. So my topic is new flower crops for doubling the farmers' income through protected cultivation. So I'm specialized in protected cultivation. So I'll be taking you through few new crops which can be cultivated. But all these crops are already being cultivated in and around Bangalore by private farm. The planting material and all they are importing from the other countries. And there are many of these new flower crops are seed born. Okay, so I'll just fly through my presentation. Okay, this is the brief introduction about the floriculture industry in India. So it is 297,000 hectares in 1,000 hectares in production, and you can go to the production. I'll come to directly cut flowers. It is 947,000 tons of cut flowers are being produced, and mainly and our exports are 19,677 metric tons. This is and it is giving our export earning of 717 crores of rupees. But majority of this is consumed, internally consumed. So we are not only the 1% of the export earning is from floriculture. Remaining everything is consumed internally because of change in our this thing. It is expected to presently it is 262 billion and it is expected to multiply by 3 times, that's 714. It is expected to multiply by 3 times, okay, by 24 to 32. So this changes, to achieve this changes, 15% change, 11.5 up to 15% changes, we need several factors. Driving forces are there. The one is technology, it's much talked today evening, and government initiatives and policies, and emerging the new marketing trend, the platforms. So these are the 4 key driving forces, okay. And there is a wide opportunity, as we all know that India, we have temperate, we have tropical, we have subtropical. So these are the all the So nowadays, since 10 years, it is African continents. It's much the many export ventures are taking place, and all the those because of the advantage development, they give land free, they subsidized loans, everything. So the people who are managing those structures are from India only. All the Indian productions have been migrated to the Zambian, South African districts. And
Next, enter.
Okay, so what is new flower crop? New flower crop is nothing but the any improved plant which is having long stalk, attractive flower, and that responds to the protected cultivation principles, and which is having commercial value in the market. That is the new flower crop. So the doubling can be achieved through following both the principles, that is principles of protected cultivation and principles of doubling. That is 4R, which we'll be discussing. And why we need flower crops? See, we have seen enough rose, gerbera, carnation, lily. It's done. We are, market is saturated. People are saturated. So wherever you go, these flowers only. So we need to diversify. Okay, and then whatever these key cut flowers are there, they are highly cost oriented. You need see rose. You need to the economic life is 8 years, and we are exporting the rose only from December to February. So these are the 3-4 months where we are exporting our rose. Rest of the time we internally consume. Okay, and during monsoon maintenance of rose is very difficult. Lot of pests are there. We need to maintain the roses. Okay, so challenges are there, and many climate modifications required. Okay, and I already told perennial in nature, and still unique flowers like blue and black. It's still a dream for an Aam. Okay, so though the genetically engineered blue is there, but that is not reached to the Aam Aadmi. So then, so how to overcome this situation? So we need to focus on so it is not new to India. Already the international market has accepted the word Alzheimer, international production, Alzheimer has accepted new flower crops. The so-called annuals. So we were used to tell annuals, winter annuals. Those flowers are today the cut flowers. Okay. So similarly, we need to explore this thing, and then and most of these new crops are seed propagated. And so if I want to give the example of Auditorium, it is 180 rupees per plant. So 1 acre of Auditorium you want to go, so you need to spend in crores for the initial planting material. So this is how we are going to have advantages. Seed propagated within 10, 20 to 30, 50 rupees max. Seedling can be obtained, and we can do the farming. Then all new flower crops do not need poly house. Okay, you can cultivate open or shade house you can do. Nowadays it is not the high tech poly house. It is the tunnel, just tunnel open both end open. Okay, so it is just giving the protection against the UV rays and other thing. Okay, so those things are there with technology we need to adapt. And then expansion. So Kia is Hyundai is there. Kia they gave new model vehicle, Kia Seltos, Kia. So the only thing to occupy the market. So in this way, we also need to bring these new flower crops into the market. Then this is okay. So these are the principle. I will not go much. Higher productivity, off-season cultivation, good quality, photo respiration, and it comes to the principles of doubling farmer income. It is 4R remunerative prices. These are policy issues which need to be, they are addressed, but still need to be stressed upon on these 4Rs. Then okay, this is another technology concern thing, and recently NBRI Lucknow has made the tulips to bloom in the hot climate. Okay, that such technology need to emphasize, and this is the one of the tulip is the crop, very important floriculture crop that can bring the highest export earnings in due course of time. So this is the image of India, right? So and government initiative, lot of initiatives are already taken through NHM, NHB, MIDH. So complete funding is from NHB at 50% subsidy. NHM is also funding for polyhouse and other thing. And exclusively for new flower crops, it's having a package for 5 lakhs per farmer. It gives So he can import the new plants from the other countries. So then MIDS also several programs are there to fund the new program. This is the marketing facilities strengthened at Bangalore flower market, one of the hub of the floriculture in India. So this online market minimize the middleman. Okay, so the farm, the direct, it's a platform brings the both back end and forward thing, and very good cold storage facility for flowers. And then coming to care market is recently going to shift care market, another flower market in Bangalore, which is going to be strengthened. Infrastructure initiative have been taken. Then online flower trading, that's flowerbazaar.com. So then few more sites are there. They are delivering the flower from one corner to another corner. These are the marketing initiatives available for the floriculture. So these are these are not imagined crops. These are all already being cultivated around the Bangalore and sold. Hydrangeas, ornamental sunflowers, Lysianthus, snapdragon, larkspur, celosia stock, and ornamental amaranthus and Chinas. Then this is one of the images from the World Flower Action Center, Alsmere. So we can see many the new flower crops like amaranthus, lustoma, hydrangea. You can see purple. So many new flower crops are already there in the list of the crop. Okay, this is the hydrangea. So in which we are already work. This is called as queen of flowering shrubs and guest indicator plant. I'll directly jump to this thing. This is the one of the farm around Belur in Assam district of Karnataka. He has grown 13 acres in this crop, hydrangea. So the individual flower cost 200 rupees, and the plant cost, all these plants are imported from other Holland. And he is not allowing. Earlier he was the growing rose and chrysanthemum. We were the buyers of them, but now he is not allowing us. And when we visited, and he just took us to the one of the neglected field and shared very few information. With that information, we started growing the hydrangeas at College of Horticulture, Mudigere. The plants were supplied by rice and share at 25 rupees per plant. This is propagated through terminal cutting again. Then we did some spacing studies. What is the spacing suitable? And the crop is similar to the Auditorium. I need to explain anything. So it requires shade 50 to 70%. It blooms after 6 months, and it's a perennial again. And the crop requirement is similar to the Auditorium. So then we also did some color intensification. It's a pink flower. We tried applying aluminum sulfate to intensify the color to the purple. So you can see the range of this thing. These are the plants applied, and optimum spacing is 45 centimeter by 45 from our studies. And this is a fertigation schedule. These are the agriculture operation pinching. It will give 4 shoots. Then after 6 months we can harvest 4 shoots. This is another researchable issue. So what is the pinching? How many number of shoots to be maintained? Another researchable issue. Then staking, it's a huge growth. We need to support on the parallelly on the sides of the bed. We need to support. So these are the after 6 months we can harvest the student 10-month crop. So our flowers were also sold at 200 rupees. Next crop is ornamental sunflower. This is the Sakata company is having 2 varieties with dark center and the yellow center, where they are doing tremendously good. 20 rupees is the wholesale price of talking. So retail is 100 rupees. Wholesale price farmer getting 20 rupees. So it does not need any polyhouse. We can cultivate outside. 60 by 45 is the spacing. Seed propagated 3,000 rupees, 1,000 seeds. Very remunerative crop, having highest potential. And in 3 months, 55 days crop starts and we can complete. Okay, so similarly for this also, College of Horticulture, Mysore Kirtimala is working on some agri lines. This is one of the identified line with dark center, multiple branching, because we don't, local market, we don't need start 1 meter, just 30, 40 foot. Okay, then this is another line, teddy bear type. This is teddy bear type, another select collection. It is in the pipeline. One more, you can see the petal variation from first one to this one. And this is the collection 23. These are the selections made at the College of Horticulture, Mysore. This is a very important, interesting crop. Rose, it is replacing rose. and it's an annual seed propagated, and pinching we need to do 3 shoots comes. Company claim 2 years, but farmers are practicing 1 year only, taking 2 flush, 2 to 3 flush, 8 flowers in a year. Okay. Okay. So this pinching, support it needs staking. It needs 1 layer. We can lift later. Okay, 1,20,000 plants required per acre. Okay, 15 rupees plant cost, and 10 stems 300 rupees. Present the cost is 300 rupees per 10 stems. It almost rose is also selling at 300 rupees now. So this is the packaging 10 stems pack. Okay, anthurium, another important term. This is also seed propagated. Okay, and these are the diversity you can see. Fertilization schedule. Okay, then celosia. Very nice. Yes, thank you. Yeah, I know. This is the new cut flowers in celosia only, and these are the packed flowers, and this is amaranthus. Very. Yes. Yes, sir. See the growth in protected cultivation. More than 1 meter. One interesting ranunculus. This is there in the Bangalore. My dream crop. It is propagated through calms. So it will change our paradigm of floriculture market. So this is the calms. Yes, sir. Trachelia, another. Yes, sir. Yes, sir. Okay, last slide. Research gaps. We need to strengthen research gap, like production technology. We need to standardize IDM, INM, this thing. Then one more post-harvest technology. So and crop improvement we need to take. This sir, I have one more this thing, sir. Not least oral presentation. I'll complete so that I need to plug in. Yeah. Number 7.
7th, sir. Mandira, I am modeling the ecological niches of endemic terrestrial.
In same speed, I'll not take 5 minutes. Ecological niche modeling. Yes, sir. Ecological niche modeling. What is it? 7, sir. 7. Ecological niche modeling. Ecological niche modeling is nothing but the technique computational model adapted for the endangered species to predict the ecological niches. What is the ecological niche? Ecological niche is nothing but the set of the environmental parameters that favors a species for survival, persistence, and multiplication. So, Avenaria roxburghi is the endangered species. It's a terrestrial orchid, ground orchid in Western Ghats. So, we made an effort to employ ecological niche modeling to understand why it is endangered. So that was this is the Roxburghi, Avenaria Roxburghi, endangered, and it is entered into red book. So I'll not, Avenarias are 835 Avenaria species are there in India, and this is having medicinal uses. Avenarias have, all the Avenarias have root bulb. So this bulb is having medicinal property, because of which it is extensively overexploited, and so that has reached to endangered situation. So this is the and forest fires, climate change, other illicit trading are also responsible. This was the objective. So this is the ecological niche modeling computational approach. Okay. Then so what we do, we go to the field. So generally belt transect is put for the assessing the population diversity. So here, since it is endangered, we make 1 meter square meter belt and count the number of the individuals are there. So we also take a image of angle cam where it gives the latitude, altitude, the coordinates. So with this data, we'll load the coordinates to the software that will give what are all the environmental parameters prevailing in that region. Okay, and it predicts so where all these species is existing, where all the other places same environmental parameters are there. So in that place, this species is prevailing. So what are the strategies to adapt, maybe adapted for conserving that species will be known from this model. So now this was the basic, and I'll just go through this thing. So these are the three occurrence data, what we take, and the environmental parameter it gets from the website, and it gives the these are the softwares used, and I told already this is the results. These are the 19 places where we have taken documented that particular species, and the coordinates, and from this the coordinates were given, this is the picture it gave. These are the places where this species, particular species is existing, and this secondary data also we feed. So with secondary data is nothing but the published data. Already there are reports are there, so those data coordinates also take, and we'll feed into the software that will predict after 50 years what is the position of this species, whether it will regain or it will extinct. So that's what the conclusion is. So what are the, and these are all the parameters actually, environmental parameters that deciding the population of the Ebeneria Roxburgh. So in this case, precipitation of the coldest quarter and isothermality and few more are there. So this is how next 50 years this species is will be distributed. The red color marked is densely available in next 50 years, and the red. Then these are the diversity studies we conducted, and this is the Roxburgh, where you can see the coordinates and the image. So okay, so these are the parameters deciding that existence of that Ebeneria Roxburgh, and this is the future prediction of the species. Thank you for the opportunity.
Thank you, sir. Next, we will move on one more speaker by Dr. G.K. Sitaramu, sir, Professor of FLS UH Bengaluru. And the topic for today's session is photoselective netting for improving yield and quality of flower and vegetable crops. Sir is going to join through online mode.
Good evening, sir. Good evening.
Good evening, sir. Good evening.
So shall I upload?
Yes, sir. Yes, sir.
So slide is, I think still it is there, no? I have to remove that slide. Remove that PPT.
I'll exit. He'll stop sharing. Nimd, sir. Google Chrome. बंद कर रहे हो
Stop share. Our share.
ಅವರೇ ಮಾಡ್ತಿದ್ದಾರೆ. Hello, sir.
Is it visible, madam? Slide?
Yes, sir. Visible, sir.
Share ಮಾಡಿ sir.
Okay.
Slide share.
Sir, you go for presentation mode, sir.
Yeah.
Yes.
So now it is in presentation mode, right?
No, sir. No, sir.
It is visible for me, like.
So once again you share, sir.
Stop ಮಾಡಿ sir.
Whole screen ಅಂತ share ಮಾಡಿ sir ಬರುತ್ತೆ.
Third option ಒಳಗೆ ಎಲ್ಲ screen share ಆಗ್ತಿದೆ ನೋಡಿ sir, ಅದು ಮಾಡಿ.
So it's moving, huh?
ಇಲ್ಲ sir.
Slides are moving, sir, but it's not in presentation mode, sir. Sir, close ಮಾಡಿ ಇನ್ನೊಮ್ಮೆ ಜಾಯಿನ್ ಆಗಿ ಸರ್ ಬರುತ್ತೆ ಸರ್. ಸ್ಟಾಪ್ ಶೇರ್ ಮಾಡಿ ಇನ್ನೊಮ್ಮೆ ಶೇರ್ ಮಾಡಿ ಸರ್.
ಶೇರ್ ಮಾಡಬೇಕಾದ್ರೆ ಥರ್ಡ್ ಆಪ್ಷನ್ ಸರ್ ಎಂಟೈರ್ ಸ್ಕ್ರೀನ್ ಅಲ್ಲಿ ಬರ್ಕೊಳ್ಳಿ ಸರ್.
हो रहा है सर शेयर. हेलो सर.
1 second.
Yes, sir.
Now?
No, sir. Again, same, sir. PPT mode, sir. Continue, sir. Okay, sir.
Okay, respected chairman, co-chairman, or organizers of GCSH on smart horticulture and dear participants. I'm very much thankful to the organizer for providing the opportunity to deliver a lead talk on use of photo-selective nets in commercial production of horticulture crops. As you all aware, use of color shade net is in horticulture or in nursery industry is almost 2 decades old in other countries. But in India, still we are in infancy stage, only in few places and few localities. So people are nursery, people are trying with different color shade nuts. Actually, in 1996 only, that Polysac plastic company in Israel it was a pioneer in shade nut manufacturing. And if you take the color horticulture crops into consideration, why it is important? There are many challenges in horticulture like climate change, the problem of pest and diseases with a lot of physiological disorder, and need for efficient resources is also very important. Maybe region specific and cross specific efficient resources are very much essential. So in order to enhance the crop yield and quality, the current knowledge regarding plant response to light is very, very important in applied horticulture. As a result of recent technological inventions like use of shade net development, the photo-selective nets in current market are it is possible to control light quality. So what exactly the photo-selective nets? As you know, plant respond to quantity as well as quality and direction and periodicity of lights. There are numerous photoreceptor molecules are very essential for light absorption, maybe chlorophyll, phytochromes, cryptochromes, phototropins, and they react with green light also. The term color shade net, it is almost there are three primary colors like blue, yellow, and red. So in addition to that, they are non-visible. They are available. Modify the non-visible spectrum and enhance the light scattering also. The other group is they are called as neutral color nets. So like gray, white, and pearl color. Normally we use all these things in some of the vegetables like capsicum and other crops. So actually netting that alters the spectral properties of light passing through the net may reduce or may replace traditional netting, which merely reduce the quantity of light. So nets are produced by incorporation of various chromatic additives, light dispersive and light reflective element into the netting materials during manufacturing. That is the way how they prepare different shade nets in the factory. So there are different types of shade nets are available in the market, maybe black, then gray, red, white, and other colors. So most of the materials has been made out of polypropylene or unknown polyethylene materials. The fraction of sunshine is always that passes through the walls within the shade fabric and remains unchanged. So it is quietly where exactly the light hitting the thread is specifically modified and scattered on the existing crop. So actually that is the major aspect happening in the plants and that will enhance the yield as well as quality of flowers. So coming to the color nets are concerned, color shade nets have the ability to control spectra of radiation reaching the crops below that are developed to change the spectral transmittance of the radiation. These nets are unique in both spectral modification as well as scattering transmitted light. By using this particular technique or technology, many companies are producing various different color nets which are suitable for different crops. For example, red shade net is always known for increasing the vigor of the plant. Blue shade net is always known for decreasing the plant height or creating dwarfing, and also it is responsible for producing a lot of suckers. And even if you take the black into consideration, that is very good for ornamental indoor plants where you cannot, there is no scattering will occur and good transmittance of light may be possible in that also. So there are different types of nets are available. Few type of nets like green, black, white, and yellow is also available. nowadays some of the companies are combining different colors also, like green, black with other kind of black color, then white with black, green with green only, with the different modification they are doing. They are all suitable for many purposes like crop growing and other purposes. So this is the way. Always above the canopy, always we need to insert the different color nets. So they will actually responsible for scattering of light, and that induces production like yield as well as quality of the flowers. There are so normally in old way used to use only green shade net for growing the plants. So nowadays, according to the crop response, we are using different color shade nets. So this is the electromagnetic spectrum available. So blue at 400 nanometer. So blue light is very active there. Suppose if you want to create dwarfing, if you want to create more suckers for plant, always we need to use blue color. So similarly, if you use green shade net, so that will enhance the vegetative growth as well as color of the foliage, and even it increases the leaf area of many nursery plant. So whenever we want to have vigorousness of the plant, if you want to grow tall plant, for example, in nursery, the growth is very important. In such cases, most of the nurseries they are using red shade net because it enhances the plant height and internodal. So that way, depending upon the purpose, also one can use the different shade nets in different places. So in black shade net, there is no spectrum or light quality modification like spectrum and scattering will occur, and even in clear crystal shade net like pearl shade net, the scattering will occur, but the spectrum remains same, and all other photo selective net like color nets. So that is mainly for improving the crop. So this is both spectral modification will also occur and scattering will also take place in that particular region. So this is the way how the shade net light is visible, available for different plants in blue, red, yellow, white, pearl, gray, and black. So some of the important concepts in finding, like so there are many works available in our country as well as other countries also. So normally we use shade net for decreasing transpiration and increase the biomass value. Then protect fruit from sunburns resulted in high yield and enhanced quality. So similarly, other colors with the different findings like fruit quality may be increased, reduced infestation of pests and diseases. That too, if you use red color shade net. So according to the our study, always if you utilize the shade net for different purposes, it is really boon for improving the yield as well as quality of the flowers. So these are all the useful aspects. Always, if you use photo selective netting in floriculture as well as horticulture, it improves vegetative growth. The plant size may be increased. As I said, red is good for increasing the plant size also. Sometimes it increases the branching pattern. All other physiological as well as morphological characters, no, mainly for improving those things. Always we use. And these are all advantages. So it is very much suitable for raising cut flower, cut foliages. Nowadays, cut foliages are taking a lot of advantages in floriculture industry. So in addition to the cut flowers, we can also grow some of the cut foliage plants under photo selective netting, and even nurseries, mainly for increasing the height of the plant in a short period. Or if you want to create dwarfing, so you can use some of the shade net like blue and red. So it is also helpful in enhancing the yield in summer crop. That means the crop periodicity almost increases because of using shade net also, and it protects from pest also. That is also main component. Always we need to understand. Even it also decreases the bird infestation in that particular place. So these are the microclimatic effects of color shade netting, like radiation control. So whenever we use color shade net, it definitely it reduces the radiation reaching the crop underneath, so that it will modify the humidity, the temperature, and also other physiological characteristics. So the scattering will occur with different light. For example, wherever no netting, there is no scattering at all. But if whenever you use color shade net, good scattering will occur, and that in turn increases the crop growth as well as yield in both flowers and vegetables. So photo selectivity is also important. As I said, red is available at 700 nanometer. So there, wherever we want to grow vigorous plants, we can grow in that particular way. And blue shade net is also responsible for creating dwarfing. And green, normally most of the nurseries and other polyhouses or shade houses, they are using green in a normal manner. The air movement can also be reduced. The wind speed will occur whenever there is a reduced reduced air movement. There is a relative decrease in temperature as well as improving humidity also. So similarly, that like temperature, relative humidity also play an important role. So these can be utilized for growing foliage as well as vegetables. So maybe black color or blue net can be used for dwarfing and all. Already I told you all these things. So whenever you use it for cut flowers also, if you use photo-selective net, so it is always, it would get longer and thicker flowering stem compared to any other photo-selective net. So red is almost very good for improving the growth of the plant.
Last minute, sir, for presentation.
Yeah, okay. So in pomegranate also, so normally earlier we used to use green shade nut and we grow anthuriums and other crops. Nowadays, according to the crop requirement, you can use plants. Even sometime black into green also we can use, and we can grow some of the crops like anthurium in a commercial manner. And even some places they use black also, but their growth is very much limited compared to other photo-selective nets. So I would like to share some of the my case studies, like the work conducted by me and my student also. So in RHREC in 2014, 15 and 16, we conducted a study, like we have evaluated different ornamental plants which are suitable for indoor location, like Aglaonema, Dracaena, Maranta, Nephrolepsis and Philodendron with different color shade nut, tree shade, then black shade nut, then green shade nut, red shade nut and blue shade nut. So these are the crops you have selected, like Aglaonema and all other ornamental plants. And this is the general view. We have created the shade house in in RHREC, and you have placed all the plants inside the polyhouse also. In addition to that, as a control, you have tried all these plants in under shade also. So if you see the result, just you can see the both. You have recorded all morphological and leaf characteristics of all those plants. Just I will take the Aglaonema into consideration. Wherever we placed Aglaonema plants in red color shade net, we could get very good growth, like in terms of plant height and also number of leaves per plant, and that is one of the ideal shade net for growing Aglaonema in a commercial manner. So similarly, other plants like Dracaena, that is one of the important cut foliage as well as ornamental plants. We have tested Dracaena in different color shade nut, and wherever we use red shade nut, no, so we got very good vigorous growth. So similarly,
Hello, sir.
Okay, one second, madam.
Yes, sir.
I will complete this case study. So
Yes, sir.
another plant like Maranta. So wherever we use blue and black shade nut, we got very good number of suckers, as well as the leaves are very shiny, and even the plant foliage is also good like that. So compared to all other shade nut like green and even controlled condition. So this is even we tested fern plants like Nephrolepsis. There also, wherever we use blue shade nut, the growth of the plant is very good, and even suckering quality is very nice compared to other shade nuts. Even Philodendron we have tested. The black shade nut was good for growing the Philodendron compared to all other like green, red, and blue shade nuts.
Thank you.
Yes, okay. So this is another study conducted at Arbavi with different color shade nut by mainly for cut foliages, for raising cut foliages. And there, for many cut foliages, this was the result. Gray color shade nut enhanced growth and yield of the plant. If you take the red shade nut, the Dracaena terminalis and Messengeriana gave very good result in terms of foliage. Nephrolepsis was very good at under gray shade nut. So this is the our own work conducted at UHS Bagalkot, both in Arbavi campus and also Bangalore campus. Even otherwise, other foliage plants like Pitospermum is also good for growing under grey shade net. It gives very good yield compared to all other crops. So this is in fruit crops also there is a good response for many different color shade nets. Okay.
Okay, thank you, sir.
Okay, thank you, sir.
So thank you so much.
Thank you, sir. Thank you so much, sir. So now we have completed all the lead speakers and keynote speaker. Now shall we start oral presentation, sir? Okay, so now. So, yeah,
3+1 minutes.
Sir, Sitaram, sir?
Yes. Hello, madam.
Sir, please stop sharing the slides, sir, so that next participant can share his slides. So the next participant is Dr. Yashwant Kumar. First we will finish offline, sir.
Respected Chairman, Co-Chairman, and dignitaries.
So it is ninth, sir. Ninth, determination of water and fertilizer use.
In the world today, we are facing the scarcity of water, that quality water. So if there is any war in the world, that is third world war, it is only because of water. So in this regard, we are discussing here. I am presenting the fertigation and irrigation studies in English cucumber. So looking to the cucumber is a major commercial cucurbitaceous vegetable. So it's having a importance of like it's having more than 95% of the water content. So it's most suitable for making cucumber ideal for hydrating and dehydrating. So for precision farming, so for fertigation and irrigation in polyhouses, we require a Parthenocarpic or gynoecious line. So gynoecious cucumber can be grown round the year in naturally ventilated polyhouse. In naturally ventilated, due to the indeterminate nature of crop, vegetative and reproductive stages are overlapping, and the nutrients are required by plant even up to the end stage of the crop. So that's why. So application of the calculation of the water as well as the fertilizer is a main thing in this for cucumber for under protected condition. Next slide. So in this regard, we with the objective of to determine the optimum drip irrigation level for cucumber grown under polyhouse. Second one, evaluate the influence of fertigation level with the major nutrients like NPK on growth yield and yield of cucumber under polyhouse. And third one is to estimate the major nutrients after cucumber in soil. So this is the program, detailed program of research. That is a general view. Next. So these are the observations regarding the growth parameters, the reproductive parameters, and the yield parameters. Next. So soil properties, use efficiency, fertilizer efficiency, and this is the first factor. There is a first treatment. The I1 is a 60% of ETCs, I2 is 80%, and the third one is 100%. This is the formula for the calculation of the water requirement. Next. So this is the pan-evaporative data of 2 years, 2 season data. Next. After getting that data, we are calculating the available applied irrigation water. So this formula we can calculate the available irrigation water to that much to supply that much of water. We have to how much time is required. By using this formula, we can calculate the time required for the application of the given moisture. This is the cumulative. This is the water requirement, and this is the next book. So these are the treatment combinations, 12 treatment combinations, out of which in most of the next book. In these cases, with respect to average fruit weight, I2 there is 80% of irrigation ETC and 120% of fertilizer has given a highest yield, which is followed in number of fruits per vine, yield per vine. Next book. next one. Before that. So with this, we conclude that. Okay. Sir, I will tell. So in the combination, I3F4 has and I2F4 has given a highest yield with respect to cost benefit ratio, with respect to gross yield and net return. So with the 80% of water and 100% of 120% of fertilizer application, we can get a better yield and with the highest economy. Thank you.
Copy ಮಾಡಿ 12. So you can copy it here.
Serial number 12. Online films list is there. Okay, start.
A very exhausted evening to all the respected dignitaries present here. Myself, Meghna, second year PhD scholar from the Division of Flowers and Medicinal Crops at ICAR IHR Bengaluru. Now I'm going to present on the topic entitled Optimizing the Growth, Yield and Energy Generation, the impact of agrivoltaics on marigold cultivation in the inter-row as well as inter-panel spaces. So to give a brief introduction, as previously said by some of the lead speakers, the population is going to swiftly increase in the near future. And to meet the basic requirements of this growing population like energy and food, which are very vital, we have to, it is one of the challenging tasks for scientists as well as the scholars. So, and one more target set by Government of India is to achieve the net zero by 2070. So for energy, we can go for solar energy, which is the most available source for human consumption. So combining this and keeping this brief history in point of view, like meeting the energy and food requirements of the growing population, we have to come up with a solution. And again, one problem is that both of them require large areas of land, which is again a limiting factor because of urbanization and because of industrialization. With that, this agrivoltaics is one of the sustainable solution where we can grow for cultivation of land, I mean crops, as well as harvest the energy on the same piece of land. So it is a dual land use technology. And it is also considered as a smart farm technology. And when we see the geographical location of India, it holds very immense potential because of, especially in the western and northern states. And it also aligns with various SDGs. And the research gap, what we have found is that the impact on crop growth and yield is lacking. And especially in countries like India, it is still in nascent stage and very pilot projects are being taken up. Whereas in advanced countries, it has been reached at a next level. And limited studies are done in ornamental crops. So with this research gap, we have formulated the objective to influence the mounting height and as well as the tilt angle of solar panels of the crop grown in the inter-row as well as the inter-panel spaces, and also influence on the energy generation. So the first we have gone for the fabrication of this agrivoltaic setup, marking of the pits, digging of the pits, and later on we installed the solar panels at 2 different heights at 6 feet and 8 feet, and at 4 different angles, 0, 20, 40, and 60. And for energy generation, we have connected microinverters, and all of them are connected to the energy meter, and we are getting the energy data from each panel at different tilt angles with the help of N-phase application. So these are the different treatments, and the design followed is RBD with the 3 applications, and these are the various parameters we have recorded. So here we can see the field view at different stages, and this represents the inter-panel space cultivation of marigold, and that is the inter-row, so that we can effectively utilize even the interspaces. And marigold, because it is a hardy crop, we have chosen this crop. To talk about the results and outcome, among all the treatments, the treatment T9, that is the inter-panel space between the panels which are erected at 6 feet and 8 feet height and oriented at a 40-degree angle in the north-south direction, it has proven better with respect to vegetative parameters like plant height and as well as plant spread as compared to other treatments as well as control also. and other yield attributing characters like secondary branches, because marigold flowers on secondary, so it will be directly influencing on the yield. Even it is also reported like more number of branches have been observed in the same treatment T9, and because of this and highest number of like highest leaf area, it has more number of photosynthesis, so it has early flower bud initiation as compared to other treatments, and also it recorded highest yield per plant around 58.65 grams. And T1, T11, which is control, has recorded less as compared to other treatments, because with respect to solar panels, the plants are exposed to more of diffused light rather than the direct light. So the PAR has been equally distributed along the crop canopy. So efficiency of even the lower leaves has been increased. And this we have correlated with the PR incidence. We have recorded this at 3 different duration per day and weekly, and we have pulled the data and calculated meanwhile. With respect to energy generation, the overall energy generated during the crop period, that is from August to October, is around 469.5 kilowatts. And among all the 4 oriented angles, 20-degree panel like orientation has recorded maximum energy because it aligns a little with the latitude of our location, which is around 23 degree. So it has recorded maximum. So with this, we would like to conclude that marigold intercultivation under 40-degree tilted angle oriented in north-south direction has proven better with respect to yield and as well as other attributing characters. And with respect to energy generation, as I have said, 20-degree has performed better, but second better performance was given by 40-degree. So with a little adjustment with the yield and energy generation, it can be recommended to the farmers. Thank you.
Only 30 we have.
Good evening, everyone. My name is Ritima Tripathi. I am from Chandrasekhar Azad University of Agriculture and Technology, Kanpur, and I am presenting. The title of my presentation is Impact of PSP and Vermicompost on Vegetative Growth, Yield, and Quality of Strawberry. And my cultivar is Chandler. And now, strawberry family is Rosaceae, and in subtropical region it is an annual fruit, but in temperate region it is an perennial fruit. And botanically, it is an aggregate and edible portion is succulent thalamus. In strawberry is cultivated in plains and hills, and it is very rich fruit in vitamin C. And now I am talking about why I am take PSB. PSB is a very basic biofertilizer for horticulture crops. It uses increases phosphorus uptake by the plant and crops, And now I'm talking about why I am taking vermicompost. Vermicompost is widely used in horticulture crop production and has the character of being one of the most valuable organic manure, and it is a rich mixture of major and minor plant nutrients. And there is a material matter. My cultivar is Chandler and treatments are 9. The applications are 3 and design is RBD, spacing 45 centimeter to 45 centimeter. And there are some treatments. I am taking vermicompost and PSB, and the quantity is sometimes less and sometimes more. And now I am talking about the growth parameters. T is the best parameter. It's a very significant parameter, and T9 is like a less significant parameter. Now I am separately talking about plant height. The maximum plant height 16.21 centimeter and number of leaves planted 60. And number of crowns and runners per plant, the most extreme number of crowns is 6.09 and number of runners is 4.97 in 35 tons of vermicompost and with 7 kg of PSP. And the number of flowers and fruits is also very significant is T8 and less significant is T9. Then I am talking about increase in the number of flowers and fruits as per plant may constantly be a direct result of the way that PSP and vermicompost application and created the improvement of inflorescence and leaf of numbers. And also in flowers might have also resulted because of the increase in the number of crowns per plant. And now I'm talking about yield. The expansion in yield might be a result of the expanded fruit set. Plant berry length and width as well as berry width and yield might be because of the way that nitrogen fixers and biofertilizers not simply expanded the accessibility of nitrogen and phosphorus through the plant, yet additionally expanded their movement from root to flower to plant foliage. Now I am talking about quality parameters. TSS, all biofertilizers significantly increase the TSS and total sugar of babies. The greatest total soluble sugar, total soluble solid 9.21 degree bricks, and total sugar content 8.31% were recorded in the treatment number 9 vermicompost with 7 kg of PSP and 25 tons of vermicompost. And now I am talking about tritable acidity and ascorbic acid. The maximum tritable acidity 0.69% and ascorbic acid 57.47 milligrams per 100 gram of pulps are recorded in treatment number 8, 7 kg of PSB and 25 tons of vermicompost. Now I am talking about conclusion. In the presentation investigation, it is concluded that plant fertilizer with 25 tons of vermicompost and 7 kg of PSB per hectare significantly increase the highest number of plant leaves, runners and crowns, everything. And based on these observations, it can be suggested that to get a sustainable higher yield of quality berries with more propagation material, the plant of strawberry cultivar Chandra should be fertilized with vermicompost 25 tons per hectare plus 7 kg PSP in the plains of central Uttar Pradesh is good. Thank you, sir.
So last 13th one, Deepak.
Very good evening to one and all, respected chairman, co-chairman, rapporteurs, my dear colleagues, and my dear friends, and all the participants. Myself, Deepak S. Kore, PhD scholar from Department of Food Science, College of Horticulture, Bangalore. This is the other part of my continued part of the work of my research project. That is the conservation of allied wood apple genotypes in Karnataka through softwood grafting. Earlier, as I conducted more than 201 genotypes across the Karnataka, that is covering more than 12 districts from Karnataka. So based upon the GPS survey, all the physical parameters, dust guidelines, and all the biochemical parameters. So the best 28 genotypes were selected. So among the 28 genotypes, I am just presenting here the 10 best genotypes, which are good for further releasing of the variety. So my crop is wood apple, as it is a minor food crops. As we all know, this origin is from India itself. So as we know, It is a minor food crop, we know it is having the most importance of nutritional and all the properties. So coming to the research, it was CRD design, the best 28 genotypes were grafted. So among the best 28, 10 were selected. So for the further data, we can see from seed collection to the graft maintenance. So this plays a major importance role. So coming to the data, we can see these were the observations recorded from like percentage of graft success, graft survival percentage, so and so. So coming to the results, as you can see the germination percentage, it is range from 100% germination was observed in most of the genotypes selected, and we can see the survival percentage up to 70%. And graft height, we can see that from 30 days after grafting, we can see the plant height from 4.06 to 26.43 centimeter. Uh, and similarly, the number of sprouts and number of leaves, we can see in the genotype 3 and 8, it is showing that maximum around 225 number of leaves were obtained. So coming to leaf width and leaf length, we can see that genotype 6 showing around 0.65 centimeter, and at 120 days after grafting, we can see the leaf length around 9.74 centimeter. So I would like to conclude that so preserving the all the essential genotypes which were surveyed across the Karnataka. So as Karnataka is as wood apple is origin is from South India itself. So conserving the most important allied genotypes would further improve, and we can get the best genotypes for further releasing of the varieties. As there are very less number of varieties so far released, so these were the operations, cultural operations which have been followed since from procuring from the rootstocks to grafting success and maintenance. Uh, these were the after grafting. We can see that good number of leaves, good number of sprouts were also around like we can see 100% of germination were germinated. Thank you.
You have the grafting product? Yes, sir. Okay. You have the grafting product?
Yes, sir. It has been maintained in in the university headquarters and in the College of Horticulture. So both locations, the best 28 genotypes have been maintained.
Shruti L.
Yes, sir.
You start your.
Yeah, okay, sir. Thank you.
First one, Shruti. Then Shruti. Print and printer is there. So will you share your presentation from there?
Yeah, I'm sharing, sir. Just a minute.
How's it? Next, Pooja Sri, just get ready. Okay, she started. Yeah, you started. Shruti.
Yeah, is it just a minute, sir? It's not showing for me, sir. Just a minute. Yeah. Okay, sir. Is it visible?
Yeah.
Yeah. A very good evening to all the dignitaries who are present in the conference. So today I'm going to present my research of PhD, that is evaluation of yield of strawberry varieties in vertical farming. So as we know, strawberry, it is Fragaria ananasa. It is a natural hybrid between two strawberry species, and it belongs
Your
to
slides
family
are not
of.
moving, Shruti.
Sir.
Your slides are not moving.
For me, they are moving, sir.
Third option is electronic. Third option is electronic. Great.
Can you just check now, sir?
The screen will get slide show. Entire screen while sharing the stage. Entire screen. Once you just close the shop sharing option. So
Okay.
One second. Just now share screen. Here will be the share the entire screen. Third option will be there.
Sir, is it okay, sir, now?
You see that few options is there. In few options, you can see. You can also change. What is the continue. Yeah.
Is it okay, sir?
Yeah. Good. Continue. You have 3 minutes time.
Yeah, wait, just a minute, sir. Is it visible, sir?
Yes.
Yeah, okay, sir. Strawberry. Strawberry, the scientific name of strawberry is Fragaria ananasa, and it belongs to family Rosaceae, sir. Sir, strawberry is one of the most popular fruits around the world because of its desirable taste and flavor. And as we have seen after COVID-19, the consumption of vegetables and fruits have been increased among the people. Mainly the fruits which contain high amount of vitamin C. So one such fruit that falls into the category is the strawberry. And why I've chosen strawberry for this vertical farming is because strawberry it has very shallow root system that extends only 20 to 30 centimeters, which can conveniently grown in vertical farming. As other speakers, they are mentioning about vertical farming of hydroponics, aeroponics, and all different. So here I have tried with a vertical farming with the PVC pipes that are vertical towers. We call it as vertical towers. So the research objective is to evaluate the yield of strawberries using vertical towers in different growing media. So this is a vertical tower what I have used for this research. So this is a PVC normal water pipe. So in this PVC pipes, I have molded the pipes into the pouches where we can hold the plant. So the height of the tower I took is 6 feet. Of the 6 feet, 0.5 to 1 feet I have cemented in the ground, and the diameter of each tower is 6 inch. And number of pouches means the pockets which you can see. These are called pouches or pockets where we can hold the plant. So each tower consists of 24 pouches, so that one tower can hold 24 plants. So spacing between the two towers is 3 into 3 feet. So in my research, the main work I've done is of these 24 plants, I've divided this whole tower into 3 tiers. As you can see, red line here, the 8 plants in the top of the vertical tower is called as first tier, top tier, and the next is middle tier and the lower tier. So accordingly, I've calculated all the yield parameters in the 3 tiers respectively. So these are the different operations in the research. So first is cementing, then sterilization of the towers and filling the towers with the respective potting media and arranging the drip on the top of the tower, one dripper line, and then planting. So this is a methodology and technical program of the work. So I have done this whole experiment at Center of Excellence GD Metla, Telangana, with a factorial CRD. Factor 1 is varieties and factor 2 is potting media. So among the varieties, I have chosen 4 varieties. That is 2 shorter varieties, Nabila and Flaminia, and 2 ever-bearing varieties, Murano and Vivara. So among potting media, I use one is control, that is cocopeat, and other 3 are cocopeat and vermicompost, cocopeat and FM, cocopeat and red soil. So with respect to yield,
slide is not moving.
Shruti, your slides are not moving.
Sir, for me, they are moving, sir, here in my screen.
Okay, conclude.
Okay, while coming to yield per tower, the highest yield per tower is found in the variety V4, and in the potting media P2, that is cocopeat and vermicompost. Coming to benefit cost ratio, the treatment V4 P2, that is Vivara with potting media, cocopeat, and vermicompost have shown highest cost benefit ratio when compared with the other treatments. So with this, I conclude that among all the 4 varieties which I took, the 2 short day and 2 ever-bearing varieties, among 4 varieties, the ever-bearing varieties are shown highest yield in the short-bearing varieties. Among the potting media of all of 4 potting media, the potting media P2, that is cocopeat and vermicompost, have shown maximum yield. So among all the treatment, V4P2, that is V4, that is Vivara, when grown in potting media with cocopeat and vermicompost, have shown highest benefit cost ratio. So as I've said, vertical tower I have divided into 3 tiers: top, middle, and the lower tier. So the top tier have shown a review than the other two.
Yes.
So this type of vertical farming we can grow easily, not only in the polyhouse but also with on the terraces or the urban farming or the balconies. We can grow, which can suffice the family requirement of this strawberry. Thank you, sir.
Thank you. Next, Pujashri.
Hi, sir. Shall I share, sir?
Shrutish, close madam nenu. Stop share. Pujashri, sir. Serial number 6. T306. Serial number 6.
Sir, it is visible?
Sir? We are saying our slides. You just see the slide and you present. No need to share from your side.
Okay, okay, sir.
Okay. We can start presentation.
Okay, sir. Good evening, everyone who are present here. Myself, Pujashri, and my PhD scholar from the Division of Flowers and Medicinal Crops, ICR IHR Bangalore. Today, I am here to present my oral presentation on the topic differential effects of color shadedness and flowering and foliage quality of Pathiphyllum, that is peace lily. Coming to the general introduction of the crop and work, its Pathiphyllum, it belongs to the family Araceae and its origin is Central America, and it's one of the dominant selling plant in the Florida foliage industry. And also NASA had identified it as a capacity to elevate the effect of formaldehyde, carbon monoxide, and benzene. And there is the response of various ornamental crops to the change in the light quality. So there is extension of internodes in the chrysanthemum, and there are also maximum petiole length in the anthurium. And so there is need of systematic understanding the systematic knowledge regarding use of color shading. So we have designed this work with the objective of to study the effect of color shading and flowering and foliage quality of peace lily, that is Pathiphyllum wallacei. And coming to the material and methods, this experiment was conducted in College of Horticulture, Venkatramanagodam in Andhra Pradesh, and with the 4 applications we did. And here the 4 different color shades that used that are red, black, white, and green with a shading intensity of 50% each. And on control, that is 100 shade was used. And all the flowering parameters are taken periodically and analyzed statistically. And this is the general view of the mixer. This is the general view of the crop which are raised in different color shades. And coming to the results, this effect of this table shows that effect of color shaded and flower characteristics of peace lily. Among the plants grown under different color shaded, black color shaded recorded the maximum flower stalk length, flower stalk diameter, spathe length, spathe width, and vase life also. And in 1 day taken for the flower appearance is maximum in the black shaded. Due to low light intensity measured in the black shaded, the number of days taken for the accumulation of ETN is more. So there is a high maximum days for flower initiation, flower appearance. Next slide. And next, this is the figure shows that the number of flowers. Here also we can see that the flowers which plants grown under black shaded have a more flowers compared to other color shaded. And this table 2 show that the effect of color shaded and foliage quality of peace lily. Here we can see that the plants grown on black color shaded have a darker green compared to other color shaded. Maybe this is due to the better protection of the plant's leaves compared to other color shaded. And this table 3 shows that the effect of color shaded and root parameters of peace lily. At the end of the experiment, we uprooted the whole plant and take these readings. In this also, number of roots per plant, root spread, length of the longest root, and dry weight of the roots are recorded maximum in the black color shaded due to more photosynthetic rate and photoassimilate accumulated in the black color shaded plants. And this is the plant picture to shows that the root length which is influenced by the black color shaded in peace lily. And from this, we can conclude that the plants grown under black color shaded perform better for all the flower and quality parameters. So we can replace the generally used green color shaded, which have the higher maximum yield compared to the black color shaded.
And black color shaded also found to be superior and viable in improving the peace lily growth and quality. And it is profitable, so we can recommend this practice for the nursery people as well. Thank you, sir.
Okay, thank you, ma'am.
Thank you.
Next, Pratima.
Yes, sir. Yes, sir.
Okay, so we are showing the slide here. You just inform us whenever you need to change the slide. Okay.
Okay, sir.
Okay, we can start.
Good evening, all. I, Dr. Pratima H., Department of Botany, Karnataka State Academy Women's University. Now I am going to present a paper, Responses of Capsicum annum to Heavy Metal Stress. Next slide. Next. Heavy metals are naturally occurring elements in the earth crust that can be toxic to humans and the environment. Soil contaminated by heavy metals as a result of various anthropogenic activities like uses of fungicides, bactericides, and fertilizers. Some of the essential metals like iron, copper, and manganese, zinc, they are play important role in the normal metabolism of the plant, but at higher concentration, it can be toxic. Next slide. The effect of copper on plant. Copper is extensively used in agriculture in the form of fertilizer, growth promoters, and bactericide. This can create copper toxicity in the soil. The copper stimulates many enzymatic activities at very low concentration, but copper is rapidly taken up by the plant root system. It can inhibit the enzymes. So effect of the next is effect of zinc on plants. The zinc also in lower concentration it enhance the plant growth, but in high concentration it inhibit the growth. So capsicum annum, it belongs to the family Solanaceae. So it's commonly called as in English chili, and in Hindi mirchi, and in Kannada it called menchinkai. So this plant is originated from the South America. So we use two varieties. One is the badgi and another one is long. So this capsicum annum is one of the important agriculture crop in the field of Vijaypur district. So the fruit is used as vegetable and spices. Soil contaminated with heavy metals such as copper and zinc are released to soil by their excess use like bactericide, fungicide, and fertilizer. The aim of the project study to assess the interaction, different concentration of copper and zinc on the morphological, physiological, biochemical changes in the capsicum seedlings. Next, please. Next slide. So materials and methods. So we collected the two varieties of the capsicum annum. One is long mirchi and another one is badgi from the Agriculture Research Station, Hitnali, Vijaypur district. So we conduct the Petri plate experiment for the seedling, growth of the seedlings. We use the two elements, copper sulfate and the zinc sulfate at different concentration, 10, 50, 100 ppm for 8 days. Next is, so we observe the parameters like the growth percentage of seed germination, root and shoot length, and fresh and dry weight, and also we observe the biochemical effect like chlorophylls and primary metabolites like protein, carbohydrates, and secondary metabolites like phenols, flavonoid, alcohol, ascorbic acid, and the proline.
Hello. Hello, Pratima. Hello. Pratima.
Yes. My slide is not visible, sir.
It's visible. Your slides are.
Now, yes, sir. Now visible, sir. So there in different concentration, the effect of copper in a low concentration at 10 pm, it enhances the growth, but in high concentration, it inhibits the shoot and the root length. Compared to the badgi, the long varieties is more affected. So the in chlorophyll pigments also in a high concentration, the chlorophyll pigments are decreased, but in decreased when in compared to the badgi, the long varieties is more affected. Next, please. The effect on the primary metabolites like protein carbohydrates, they also inhibited at a high concentration of both bad and the long varieties. Next, please. The secondary metabolites like the phenols, flavonoid, and alkaloids, they are enhanced when concentration is increased. So, in next, please. The proline is one of the stress amino acids. They enhanced in the under stress condition and it protect the plants. The overall result shows that 10 ppm copper was more toxic to long variety than bad gain compared to zinc. The low concentration of the metal treatment enhanced the low plant growth, but in high concentration, the secondary metabolites of the inhabits. One of the important metabolites like the alkaloids, that is capsaicin, it gives the pungency to the chili foods. So further work suggests that effect of metals on capsaicin content in the different growth stages of the plant. Thank you.
Okay, thank you, Pratima.